And for all the main talks, it is going to be 20 minutes. There will be a bell and then 10 minutes for the discussion. I just request everyone to stick to the time. Thank you. Yeah.
All right, thank you for the introduction. So, as Anindya mentioned, my name is Vidya, and I'm a recent hire here at the NBRC. I just came here about 2 months ago. Previously, I was an assistant professor at the Jackson Laboratory before I moved here. And so I'll be continuing some of our work that we've initiated at JAX and talk about a new project that sort of emerged as I was preparing to shut down my lab to move here. All right. So we are largely interested in the progenitors of the cochlea because very much like the central nervous system, the auditory cells in the cochlea don't regenerate. And so here is a cochlea that's sort of stripped out, it's and dissected, and you can see its coil structure. And along this, the length of the cochlea, there's a tonotopic gradient that responds to different frequencies of sound. And so because of this regenerative problem, we study everything from the progenitor stage to how it differentiates and attains this really complex organization of specific organization of cells. And of course, how the sensory epithelium then goes on to mature and adopt this highly specialized cytoarchitecture. And all this is very important for its function. Now, obviously, we look at a lot of gene expression during these progenitor stages. I'm going to skip over all of our transcriptomics we've done in the bioinformatic stages and just sort of dive into some of the biology on a select gene that we've become interested in. But the overall goal is using this gene information is to assemble a gene regulatory network that both uh, to better understand its function and also in a spatial context to see how you can differentiate or specify certain cell types, in a spatial manner. All right, so here is an anatomical drawing by Bodel, a very famous drawing of the inner ear. It's a very complex organ, highly specialized membrane labyrinth that's fluid filled. And there are two portions to it. You have the vestibular half and the auditory half. So there are overall 6 sensory epitheliums. There are epithelia. There are 5 in the vestibular system, and they're largely responsible for balance and motor function. And then obviously you have this auditory portion where which is essentially this coil structure, the cochlea in mammals. And this is responsible for hearing. Now, a lot of hearing and vestibular disorders are related, but overall there's a much higher incidence of auditory disorders just because the auditory cells are much more highly susceptible to damage. If we were to take a cross section through the cochlea, you'll sort of see how it reveals this very complex organization, several chambers within this coil structure. And in red here, this is where the sensory epithelium sits in the scala media, and then you have 2 other fluid-filled compartments above and below it. And then in this blue signal is sort of marking the neurons in the spiral ganglion that will go on to innervate the sensory epithelium. If we take a closer look at the scala media, you'll start to see that it actually is differentiated in several other epithelia. But the sensory epithelium is what houses the auditory cells or the mechanosensory hair cells that are in green over here. And if you were to take a closer look at the sensory epithelium, you'll notice that the hair cells in green are intercalated by these glial support cells. And this organization is extremely important for auditory function. So how is it developed? How is it maintained? And a lot of the images that I'll be showing you will either be a cross-sectional view or a whole mount view of the sensory epithelium like you see here. So it's basically a top-down view as if you were looking down at the epithelium from here. And so here's the sensory epithelium labeled for the SOX2 glial cells as well as the hair cells. So you'll notice there's one row of inner hair cells, and that's responsible for detecting sound, and 3 rows of outer hair cells, and that's responsible for amplifying sound. But of course, you also have the spiral ganglion neurons coming in to innervate these hair cells, and that's how they transmit auditory signal to the brain. Now, as I mentioned to you before, even though the inner ear is actually a peripheral organ, we have a lot of the same problems that the CNS shows, and that is that once these cells are gone, that's it. They don't really regenerate very well. And so here's a dissected cochlea, and you can see that is the coil structure is well maintained, even in an in vitro setup. In the base, that is usually responsible for detecting high frequency sounds, whereas in the apex, that detects low frequency sounds. Now, ironically, the cells in the apex are actually born first, last to differentiate, but they also hold their regenerative potential the longest. And even then, they sort of disappear within the first postnatal week. By P3, you will start to see the last, vestigial evidence of regeneration when stimulated. But regeneration in the base is very, very difficult to achieve. And in fact, those cells are actually the most susceptible to damage. So we have a very difficult problem on our hands. Now, over the years, as a field collectively, we've identified a lot of pro-proliferative factors. We've also identified a lot of key differentiation genes that are important to get these cells to first re-enter the cell cycle and differentiate. But another thing that I've sort of gotten interested over is, well, yes, we've got to regenerate these cells, but what's important is that we generate healthy cells that will stick around and that are viable. And if we don't take care of this, it can lead to quite a few debilitating genetic diseases. So because I'm interested in regeneration, the cell cycle becomes extremely important. So all cells will go through different phases of cell cycle. The growth phases are where the cell sort of prepares itself for the next phase. Now, during the S phase is where DNA replication happens, and mitosis is when the cell divides and gives rise to 2 daughter cells. But we also have this important growth arrest phase, where cells are just paused momentarily. If they see a pro-proliferative signal, they will re-enter the cell cycle, and if they see a pro-differentiation signal, they will start to differentiate. And so we know we can follow these key phases of the cell cycle by either using or marking for pCNA or BRDU. BRDU is a thymidine analog that gets incorporated into DNA as cells will divide. For the mitotic stage, we can label cells with Ki67 or phosphohistone 3. And for markers of cell cycle exit, we can look at P27, P21, P19, and there are several genes of this class. And there is now, this is quite well-known old data that is well established in the field, but there is a nice correlation between what cells are what cells are not proliferating and what cells are expressing this cell cycle exit marker, P27. So in red, you have cells labeled with BRDU, so they're still proliferating, whereas P27, they're in that G0 growth arrest phase. And as I was saying before, P27 signal starts in the apex and moves to the base. So the cells in the apex are the first to be born. They're the first to exit the cell cycle, and yet they're the last to differentiate. Now, can we use signals like P27 to sort of induce them to re-enter the cell cycle? And so a lot of people in the field have been playing around with manipulating these factors. And so, for example, if you were to look at a cochlea in the first postnatal state, there is no proliferation going on at this point. So there is no BRDU, EDU uptake. EDU is just a similar analog. Now, if you were to knock out P27, that's that cell cycle exit gene, you can stimulate a few cells to re-enter the cell cycle. So that's pretty exciting for the field. However, the sad fact is about 4 weeks after you've regenerated these cells, they all die. So how do you get them to survive? So there's something about the cell cycle we still yet don't understand. Here's another example, a cross section of a cochlea. This is the control, so you can see your 1 row of inner hair cells and your 3 rows of outer hair cells. And when you knock out P21 and P19, which are the other cell cycle exit markers, you'll actually start to see cells, they do re-enter the cell cycle for 1 round of proliferation, but then they will start to die off. So clearly this is a problem. We've got them to maybe re-enter for 1 round of proliferation, but how do we keep them around longer? So to sort of look at this holistically, I sort of went into very early development to try to understand, well, what is the embryo doing that is right in the first place? And I'm sure most of you are familiar with this from your basic biology classes, but the Wnt pathway is very well known for specifying this stem cell population or progenitor population in any organ. So again, I'm going to skip over all of our transcriptomics, but we identified this gene MYBL2, and this has been specified in several stem cells. If you were to knock out this gene in early development, you wouldn't even get past the inner cell mass stage. So it's very important for those early stages of development. But if you were to look at its spatial expression of the progenitor population in the cochlea, here's porcupine, which is an enzyme which is important for Wnt secretion. So it's enriched on this side of the cochlea, on the medial side. And you can see this is where you have your pool of progenitors. So they are Ki67 positive here. And very nicely correlating with that, you have MYBL2 expression. So what we do know from the stem cell studies is that MYBL2 is important for the DNA synthesis phase. So it comes on about late G1 through S phase until about early G2 phase. So we got the mouse model and we knocked out MYBL2. And not surprisingly, we saw, here's our progenitor pool in the control. We labeled it with both Ki67 and PCNA. So green labels Ki67. Not surprisingly, when we knock out MYBL2, you see a decrease in both the fluorescence of Ki67 and the number of cells that express Ki67. So I'm just going to skip over the graphs, the quantifications, and just give you the gist of our finding. Now, what was strange is that so you've reduced your progenitor pool, so automatically you expect to see fewer sensory cells. But that's not really what we found. So when we took out this organ to E18, so I have to stop and just remind to tell you that this is E14.5, so mid-gestation. Now at E18.5, they differentiate quite well. Here you're looking at the hair cells, 1 row of inner hair cells and the 3 rows of outer hair cells. But in the MYBL2 conditional knockout, you actually start to see more hair cells. So it's a bit counterintuitive when you think about it. How do you remove your progenitor population to end up with more cells? So there's something about this gene that we don't really know about that has roles other than cell cycle regulation. So to first develop a hypothesis, we compared its spatial expression. So utilizing ChIP-seq data, we found that JAG1 is a target of MYBL2. And JAG1 is a major gene in the cochlea that is essential for specifying the sensory epithelium. And if you just look at JAG1 expression here versus MYBL2, maybe it doesn't really seem exciting, but we have to look at it a bit quantitatively. So here is a single trace of this example here. MYBL2 is expressed on this medial side, and JAG1 is maybe slightly a bit more centrally, and you can see the JAG1 peak in green. But what you'll see is that JAG1 expression is increased just where in spatially where MIBL2 expression is lost. So so MIBL2 is a transcription factor that, it can both regulate the expression of genes, but it can also repress the expression of genes. It has both functions. So how do we determine what its role? We actually use spatial expression to help us here. So wherever MYBL2 is expressed, there is no JAG1 expression, and where you lose MYBL2, you start to see that JAG1 peak. This is just a single trace, and that was consistent when we looked at several examples. So likewise, you expect, how does this relate to influencing patterning of the sensory epithelium? So here's JAG1, which is the predicted target of MIBL2, and SOX2, which is the specifier of the prosensory domain. And from work done from several years ago, we already know that Wnt can regulate JAG1, which also regulates SOX2. Now, when you look at the MYBL2 conditional knockouts, you actually start to see an expanded JAG1 domain. And you also see, and because of that, you see an expanded sensory epithelium. And this was, we quantified this, and it was quite significant. So what happens in a more mature cochlea? That was E14 mid-gestation. So again, at E18, you have your one row of inner hair cell and 3 rows of outer hair cells. Now in the MYBL2 conditional knockout, you start to see additional inner hair cells. So we quantified this to death in all ways, shapes, and forms. We measured every single domain. We looked at the levels of gene expression. But the one thing that I wanted to point out is that in all cases, when we looked at SOX expression, along the entire tonotopic gradient, we saw an increase in SOX2, both levels as well as area. So the loss of this progenitor gene is somehow resulting in an expanded sensory epithelium. So how is this exactly happening? It's happening in a spatial context. So within that progenitor pool, it is suppressing JAG1. And it's establishing where that boundary of that sensory epithelium is going to be formed. So when you remove MYBL2 in this progenitor pool, you start to relieve that repression on JAG1 and actually expand that sensory epithelium. So it's sort of like a catch-22 problem. So if you want more cells, you have to transiently increase MYBL2 to get them to proliferate, but then you have to shut it down so that they can become sensory later. So this is, what we very recently published. But then after this, while we were doing these studies, we found a lot of strange data that we couldn't quite explain. And this is what is sort of leading down to this new path. So I already showed you that Ki67 expression was decreased in MYBL2. Not very surprising. It is affecting cell cycle regulation. PCNA, which is a marker for S phase, there was no real remarkable change in fluorescence, at least. It's hard to talk about numbers because you've obviously decreased the size of your progenitor pool, so you're going to have fewer cells. But this was sort of stumped us a little bit. So we wanted to use BRDU to actually look at the S phase in particular. So we moved to an in vitro setup where we wanted to see what was happening to DNA replication in particular. So we can culture these organs in vitro, add BRDU, and see what happens. So just to make sure that you can inhibit the cell cycle, here's a control cochlea with BRDU incubated for 24 hours, and CPT, which actually inhibits BRDU uptake and proliferation. Now, weirdly enough, when we looked at the MYBL2 knockout, we actually see an increase in BRDU incorporation. So again, what is going on? This just confused us like crazy. So what was happening? We see an increase in BRDU, but yet we're seeing a decrease in Ki67, and yet no real change in P27. And this needs to be properly quantified, but the data wasn't really quite making sense. But what it seemed to suggest is somehow the cells are being stalled in the S phase and not really progressing through to the rest of the cell cycle after that. So what could be happening here? We were worried about the DNA integrity if there's so much BRDU uptake when there shouldn't be any, or so we thought. So during cell proliferation, or just during a lifetime, you can have a lot of insults to DNA. So you can have single strand breaks, you can have oxidative problems, cross-linking lesions, mismatches during DNA replication, but you can also have double-strand breaks that can happen to irradiation, drugs. Now, imagine an embryo. They're protected from UV light. They're not really exposed to radiation, so that's clearly not going to explain what can cause double-strand breaks. But potentially, replication for collapse could be an explanation here. And when DSBs occur, you will start to see an increase in gamma H2AX signal, and the signal is sort of helps recruit DNA repair genes to the site of the break and repair the damage. So could that be what's happening? Because there's just so much BRDU incorporation during this S phase, we wanted to look at the DNA damage status. Oh, and I should also mention, although there are environmental factors that can cause DSBs, there are so many genetic factors that we know of today that cause really debilitating diseases. Myelodysplastic syndrome, Fanconi anemia, and even progression, adding insult to injury. Glioma, the progression of gliomas is just that much worse. So what do our organs look like? So in a normal progenitor pool of a normal cochlea, you will see some double-strand breaks occurring in the system. But the machinery to repair these double-strand breaks are also present. But when you knock out MYBL2, you start to see that much foci, H2X foci, indicating that much double-strand breaks. So MYBL2, yes, it's important for cell cycle regulation, clearly plays a role in boundary formation, but it's also important for recruiting or regulating, it's a transcription factor, or regulating the expression of DNA repair genes. But also MYBL2 has been linked to this syndrome, myelodysplastic syndrome. And so, as through the progression of disease, even in cancer cells, where this is most well studied, as cells are proliferating, you're going to constantly get these DNA strand breaks. Your telomeres are going to be completely unstable, and that just goes on in a much aggressive state of disease. Now, in myelodysplastic syndrome patients, there's a high incidence of cancer, and there's only one reported case of sudden hearing loss, but I have to admit this is in general very poorly reported in the clinic. However, MIBL2, according to ChIP-seq data, is regulating BRCA1, which is a major DNA repair gene. And BRCA1 has been linked to Fanconi anemia. And this, we know, has been linked with hearing loss. So clearly, MIBL2 is playing an important role in DNA repair and maintaining DNA integrity, both the developing cochlea, and if not, it can lead to debilitating genetic diseases. So we can use MIBL2 and the BRCA1 mouse models to start studying this. Now, previously, it thought that, once you have a double strand break, that's it, you would halt cell division and gene transcription and everything. But that idea is changing now. Double strand breaks can play an important role in actually the regulation of genes, especially when chromatin is so tightly wound at certain stages. That double strand break might be essential for turning on gene transcription. So this idea that DSB kills off cells may not really be true. So what really happens in our case, because we clearly do get hair cells, even despite the double strand break that happens at E14, they still clearly go on to differentiate into hair cells. So taking a closer look at these hair cells, we just wanted to look at a closer look at how they look. So in a control, you have your one row of inner hair cells, 3 rows of outer hair cells. It looks pretty good. But when we knock out MIBL2, we start to see a lot of defects. Cells that are trapped in a hybrid stage that are trying to become a hair cell and don't quite make it through. We have some cell division defects. We have cells with micronuclei, and a lot of morphological defects in general. And I'll just end there. So there's a lot of interesting work that we'd like to do going forward, but it's sort of an open-ended space. So this work was initiated at JAX, and a published study was spearheaded by Carol and Emily in my lab. And now we're sort of moving on to potentially look at DNA repair pathways.
Time for a few questions.
Is MIBL2, is it a basic helix loop helix protein or DNA binding protein? So how does it sort of recruited? How does it get recruited into these cells? I see. Hmm. And in your system, you said, initially you started with the Wnt signaling, and that seems to define the proliferating field. So is there a connection between the MIBL2 recruitment and the misregulation of the Wnt signaling? So how does these two connect? Right. But the recruitment process is, you don't know the recruitment mechanism.
Questions for Vidya? All right. If there is no more question, thanks, Vidya. Our next speaker is Somia Anger. She's going to talk about Songbird model system to study vocal deficits in neurodevelopmental disorder. Over to you, Somia.
Slide is also not showing. Okay. Yeah. So I got to the point where these birds will pick up their song from their fathers during a sensitive period. First, by hearing the song, creating a copy of it in their brain, and then by practice, which is auditory motor integration or sensory motor integration. And by 120 days when they're adult, they're producing an almost perfect copy of this father's song. I'm saying almost perfect copy because no two zebra finches sound exactly alike. Each zebra finch has its own signature tune that it sings in adulthood. So this is again this a little bit more on sensory and sensory motor phases. The sensory phase starts at about 20 days after they hatch at the time of fledging and goes on up to 40 days. So this is the time when they're listening to their father and creating that copy of the song. 25 days onwards to about 70 to 80 days is when they practice their song. And during this time, initially, they produce a very soft, wobbling kind of song, which is similar to a baby's babbling, followed by plastic song where the song is still malleable. It can be changed because they're rehearsing the song. And by adulthood, which is at 120 days, they produce a fixed song. So after 120 days, the song doesn't change. Here's a visual representation of this song, frequencies along the y-axis, time along the x-axis, and we have different notes in each song or motif. Each note or syllable is of a different type. So for example, you can see frequency modulated notes, which are sweeps of frequency. You can also see these notes which have harmonic stacks. Now the harmonic stacks are simpler to produce. They are called like notes.
whereas the frequency modulated notes are considered to be more complex. And zebra finches, to attract females, should produce more of these frequency modulated notes because females like them. Now, the motif itself, you can see it's composed of these syllables, and this is repeated over and over again in the song. And there's a small gap between each syllable, which is called the intersyllable interval. This is the song control system, or the set of neural circuits which are important for controlling the song. This is a sagittal view of a zebra finch brain. Here's the cerebellum, and the beak would be on this side for orientation. Field L is the auditory cortex, and input goes into this area, which is called HVC, then to another nucleus called RA, and then to another nucleus called the 12th nerve nucleus. This particular pathway is important for controlling muscles of the syrinx, which is therefore this is the vocal motor pathway. This particular pathway is also important for controlling temporal features such as syllable duration and intersyllable interval. HVC projects to an area of the basal ganglia in birds, which is called area X. Area X projects to a thalamic nucleus DLM, which then sends projections to a cortical nucleus lateral MAN. And lateral MAN forms two loops in the brain by looping back onto area X and also onto RA. This particular pathway is important for vocal learning during development. So if it's damaged in any way or injured or lesion in any way during development, during the sensitive period, the bird will not be able to sing properly. In addition, area X ultimately projects to the ventral tegmental area through a couple of synapses, and the ventral tegmental area sends dopaminergic projections back to area X and also to other areas like, for example, HVC and RA. So what about the opiate system? The opiate system consists of different kinds of opiate receptors. Although there are at least 5, I'm going to mainly focus on mu and delta opiate receptors, which bind the endogenous peptides, and these are the endorphins and enkephalins. Now, there is no strict one-to-one binding of these ligands to the receptors. Endorphins can also bind delta, enkephalins can bind to mu. And these are some of the antagonists that I'll be talking about later in my talk. CTPA is highly selective for mu. Naltrexone binds mainly delta, and naloxone is not very specific. It mainly binds mu, but at higher concentrations will start binding to delta as well. So these are the opiate receptors are inhibitory GPCRs, and they are generally expressed by GABAergic interneurons. That means if these mu and delta opiate receptors are bound to their ligand, then what will happen is that they're inhibitory, and the inhibition will be removed because you're basically inhibiting GABAergic interneurons. The mu opiate receptors are mainly known to modulate pain, neuroendocrine functions, food and water intake, etc. But what really caught my attention was the fact that they modulate vocalization, social behavior, and motivation and reward. And delta opiate receptors have a role in anxiety, threat situations, and enhancement of drug-seeking behaviors. So a little bit more about opiate receptors. In cognition, mu opiate receptors are, of course, well known for the fact that they're involved in opiate addiction. And recent studies have shown that this, in opiate addicts, you see deficits in verbal intelligence and speech, slurring and stuttering. So clearly there is a role for it, for the mu opiate receptor system in speech and in speech itself. In utero exposure to opiates, mothers who are addicted to methadone and heroin, their children have impaired mental development and also language development. And I will skip for delta opiate receptors. Both are involved in different kinds of learning and memory. I'm not going to talk about this too much. But for example, in a condition in ASD, what happens is that these are some case studies which have shown that naltrexone and buprenorphine. Naltrexone is an opiate antagonist, whereas buprenorphine is an partial opiate agonist. Both of them ameliorate symptoms of ASD, and they also cause increased social and affiliative behavior in children and adults who have this condition. Also, recent studies have shown that mice where the mu opiate receptors are knocked out, This leads to what is called a monogenic model of autism. They display symptoms which are seen in other kinds of autistic mice models. So recent work in my lab, what we have done, some of my new students have shown this, and this is that both opiate receptors, the genes for opiate receptors, mu and delta, are connected to neurexin 3, and neurexin 3 is very well known to be involved in autism. So orthologous genes for mu and delta are present in zebra finches, and also recent studies have shown that this opiate receptor, mu opiate receptor gene is found, deletions of this gene are found in ASD patients. So in fact, copy number variations of this opiate receptor, the mu opiate receptor, were detected in low functioning ASD subjects. So all leading to the fact that besides studying the mu opiate receptors for addiction, you could also link it to autism and study it from that point. So any other connections between opiate receptors and uh, the song system? Well, it turns out that both HVC and RA, which I have shown you earlier, there is an increase in proenkephalin mRNA after singing. And this happens both in adult birds and juvenile birds that are practicing. These are studies by Wada et al. So both of these regions have more of enkephalin being, and the gene for that is proenkephalin mRNA increases after they sing songs. So when we started our work, there was not too much known about the opiate system in zebra finches. So we started initially by staining for these receptors. We have found the mRNA for opiate receptors, that this detects both mu and delta, found in all song controlled areas, like for example, lateral MAN, area X, and HVC, all of them have opiate receptors. And recent studies, we've used immunohistochemistry to detect opiate receptors. This is for mu, staining for mu opiate receptors in HVC and RA of adults, and also HVC and RA of young birds at 35 days, that is during the sensitive period. Studies done by my student Sandeep and some old work from Nazia when she first started. Nazia showed that in area X, and I'm only going to describe one set of these experiments, the area X is unlike striatum in or basal ganglia in humans, in the sense that both striatal and pallidal neurons are present in this one region. Normally in mammals, you have both the striatal and pallidal neurons are separated. Now, if you block this area by putting a cannula like this and injecting naloxone directly into the striatum, both striatal neurons and pallidal neurons, these larger neurons which are pallidal and the smaller ones that are striatal, both express mu opiate receptors. Now, what happens is, as a result, we have certain changes in the song. And what are the changes in the song? I should also point out here that there is a connection to ventral pallidum. This is also an inhibitory output going to ventral tegmental area, which is dopaminergic, and these dopaminergic neurons loop back onto area X. The projections going to area X from lateral MAN and HVC are both glutamatergic. What Sandeep found when he did these cannula implants and injected naloxone directly into area X, he found that there was an increase in the number of songs. So the highest dose, he tried out many different doses. The highest dose caused an increase in the number of songs that these birds produced. But on the other hand, dopamine showed slightly different pattern. So for this 100 nanograms dose, there was an increase in dopamine, but for this highest dose of 200 nanograms naloxone, which we directly infused into area X, this level was not so high. And this is where the birds sing more. Now, recently, Mukta has shown, and this is by making a lentivirus against mu opiate receptors and knocking down mu opiate receptors specifically in area X, she sees something very similar. This is basically preliminary data, but before the lentivirus injections, they were singing at this level, and after the infusion of this lentivirus carrying the and used for knocking down new opiate receptors was injected, they sing much more. So these data are kind of similar, whether we do naloxone infusion or we do lentivirus knockdown of new opiate receptors. And this is what I was talking about in terms of quality of song. We've done analysis of various kinds of features, like for example, syllable length. This is a temporal feature. We've also looked at frequency modulation, amplitude modulation, mean frequency. Vena entropy measures the noisiness of the song, pitch goodness. All of these varied, either became higher or lower across the different doses of naloxone. And there were therefore changes in the acoustic property of these vocalizations that these birds produced after injecting naloxone directly into area X. This is supported by earlier data showing that dopamine infusions into area X led to changes in the spectral features of song. And what we think from these results is that probably what is happening is that when we flood area X with naloxone, which blocks new opiate receptors, we don't, at this point, what will happen is that these neurons, the pallidal neurons, which are the output of area X, this output will be blocked. As a result, what will happen is that there will be no output going to DLM, and this output will be also reduced. As a result, your ventral pallidum will be activated, and ventral tegmental area will then start secreting less of dopamine back into area X, which is what we found with the 200 nanogram dose. On the other hand, if this output is reduced, then DLM will start to fire. There will be more firing of lateral MAN, and there will be more of vocalization, which is exactly what we found with the 200 nanogram dose. So it is possible, and this is data shown by others, is that birds may be singing more in an attempt to increase the release of enkephalin, which now can no longer bind to its receptors. So all this work was done in adult birds. But what happens to young birds, and how do we link it to ASD? Yes, we tried CTAP also. CTAP is highly selective, but we haven't tried it in adults. We have done it in juvenile birds, which I'll be showing you. So I can skip over this data. This is just to show that just as we had theorized, pallidal neurons are more active with 100 nanograms and less active with 200 nanograms. And this is basically staining for ARC, which is linked to, which is an immediate early gene, which is linked to increase in activity. So what about young birds? We decided to look for effects of mu and delta opiate receptor neuromodulation on song learning. And we did this by using a similar paradigm for both mu and delta opiate receptors. We waited for the birds to become about 35 days. Now, 35 days is a time when they're actively learning their father's song and also actively practicing. So at this time, there's a lot happening inside the brain. We have two pathways which are being constructed literally in terms of pruning and refinement. One is this pathway going from the thalamus to the lateral MAN, and this is a thalamocortical pathway. Also, lateral MAN itself, those neurons project to RA. This is also being refined. So extra processes are being pruned off, specific connections are being made. And interestingly, the pathway going from HVC to RA, which is a major component of the vocal motor pathway, this is not fully even formed. Those axons are just growing into RA, and they will be pruned, and they will settle and make connections following the 35-day period. So this is the time that we started making these antagonist injections. For new opiate receptors, we use the CTAP, which is highly selective for new opiate receptors. And we also used the similar paradigm we use for the Malton rule. What we have done is we've used siblings, since both siblings will learn from the same father. One bird was given the vehicle and the other bird was given this treatment. And this went on for 10 days, so between 35 days and 45 days. At the end of this period, this is at about 120 days, actually between 80 and 120 days, birds' songs were recorded every 10 days, and then we compared the songs. So for mu opiate receptors, what happened? We found that birds were singing much more. The treated birds sang much more than the controls at every age that we tested. We also have changes in the timing. Intersyllable intervals were longer. They were prolonged as in terms of singing. And this is in fact something which is similar to, at least in some cases of ASD also, there is, kids will stretch out their vocalizations. There were changes in pitch, frequency modulation, and mean frequency of these songs. So these are all spectral features, and in all cases, there was a decrease in these spectral features in treated birds versus control birds. And finally, we compared learning. So what we did was we compared the father's song with the control and the treated bird. And here what we found was we looked for copied syllables and non-copied syllables. So this is Mukta's work. What we found was with birds which were treated, they produced many more of what are called non-paternal syllables. So they were not copying their father's song very well. They were actually copying more, or they were producing more of song notes which were not belonging to their father's song. Where exactly these had been copied from, whether they were from neighboring birds, whether they had produced them, improvised them, or they had picked them up from their siblings, sometimes they do that, is something that we still have to study. So this was as far as new opiate receptors were concerned. So you can see here the treated bird produced only one of his father's songs, notes, but all these were copied from the father in the control. In the case of delta opiate receptors, what happened was we did not find any changes in the number of songs, which I'm not showing here. There were no changes in syllable duration or intersyllable intervals, so no change in timing. The spectral features, pitch, mean frequency, and frequency modulation all changed in new opiate receptors, and the same way we had changes in delta opiate receptors. There was significant decrease in all these spectral features of the song. And finally, when we compared the songs of their fathers with the control and the treated birds, what we could see was here, treated birds sang whatever the father was singing, so that was not the problem. What happened was that the kind of notes that these birds were producing. This is a control, and I told you that frequency modulated syllables are more complex. So birds like to include more of frequency modulated syllables to attract females, and call-like notes or harmonic stacks are simpler. What was happening was that these birds, control birds, were producing more of the frequency modulated syllables, which are more attractive to females, but treated birds were producing less of these. Instead, they seem to be replacing most of their song with these harmonic stacks or call-like notes, which you can see here. So mu and delta opiate receptors have overlapping but slightly different functions and can affect songs in different ways. So I will skip over this comparison and go over to the last slide. Just to summarize, the endogenous opiate system affects both the motivation to sing and quality of song in adult male zebra finches. And this system also plays an important role in song learning. Blocking opiate receptors with specific antagonists during this period leads to long-term changes in the song, which you can see when the bird becomes adult. So just what is important here is that we just used a 10-day block. After that, there was no block. We left the birds back. We put the birds back in the aviary. They had normal social interactions with their other birds in the aviary. It was just during that 10 days when they were taken out, injections were given, and they were put back into the aviary. And mu and delta opiate receptors play overlapping but slightly different roles in song learning. The last is a conclusion that I want to draw from this. Male zebra finches can be used to study deficits in learned vocalizations associated with various neurodevelopmental disorders such as ASD. What we plan to do is, and which we've already done in the lab, we have a lentivirus against mu opiate receptors. I showed you some preliminary data in adult birds. Now what we plan to do is to knock down opiate receptors specifically in area X of young birds and compare it with the sibling and the father, which would receive, the controlled sibling would receive scrambled shRNA in lentivirus injections. And in this manner, we can also look for other genes and how they would affect the vocalizations and whether they would produce vocal deficits, which are similar to those produced in cases of ASD. Thank you. Stop there.
Time for a few questions.
Yeah.
So the first part is, do you think that most of
they probably are, but I can't rule out that because these are systemic injections. So if there are any direct effects, that also I can't rule out. Literally, the entire brain is full of opiate receptors, so it's actually very hard to tease out where it could be dopamine. And on with systemic injections, at least, the opiate system not only interacts with the dopaminergic system, but all other neurotransmitter systems as well. So one would literally, for a systemic administration, I can't say too much. Dopamine we've checked out. Others there could be also.
so sort of very you think it's like the direct effect locally where gather
Yes. Yes. Right, because in this experiment that I showed you afterwards, that what we had done was we injected specifically in area X. Now, this naloxone is not going to other parts of the brain, it's going specifically to area X. As a result of a lot of naloxone being released in area X, you would have ultimately shutdown of the pallidal neurons. If the pallidal neurons are shut down, and that's a GABAergic output to ventral pallidum, those ventral pallidal neurons are allowed to fire. They are also GABAergic, and as a result, downstream you have the ventral tegmental area, so there won't be any release of dopamine. There would be a reduced release of dopamine anyways. Any other questions?
My question is, do you think that encoding decoding system of autism, which is actually less in temporal lobe, has this particular training has any effect or positive effect on that, or positive influence on that encoding decoding system, for that what they are actually hearing, they can't decode it or they can't encode it, whether it has any influence on that?
You're asking whether it has effects on auditory system
Whether
as well.
not the auditory system actually, the when it is on temporal lobe and then interpretation is going on, whether that particular auditory system has any auditory system, auditory
Whether it's
canal is okay, that I know.
interpreting it right or wrong is something that is difficult to say, because all said and done, the vocalizations are only for one specific purpose, that is to attract females. They will still produce the song. The only thing is that the song is different
Interpreted or
from
not.
controls. However, the one thing that we do want to try out in the lab is to try to look at calls. Calls have meaning.
That
There are
I am
different kinds of
asking.
calls have meanings.
Yeah,
We
yeah.
have actually not studied calls yet. We've looked at song, but not
Okay, okay.
calls. So I will be able to answer your question after Mukta is done with all her experiments.
Okay.
Yeah.
cannot absolutely do it, then I have this listing. I think it's going to work. Yes, perfect. Okay, that's fine. I'll just control it from here. I'm audible, right? Yeah. So today we will discuss one of our work on how non-coding RNAs regulate synapse development. So Anindo is right, because most of my talk will be how non-coding RNAs are regulating synaptic plasticity and how that regulates memory. And we have started working on non-coding RNAs in synapse development and its impact in autism like 1 or 2 years back. And then now there is a significant data that I could share with you. And just to make it more coherent and fit it to the symposium theme. So this is almost like all live slides, you can say. So our lab is primarily interested to understand the RNA-based mechanisms of synapse development. That is very important for many aspects of developing brain, including the social behaviors, social interactions, and also the adult brain, synaptic plasticity, and memory. For interest of the time and based on the theme, I'll be talking about how synapses are developed, and we focus on one of the most important brain area is hippocampus, and this is one of the CA1 neuron, and these green dots, the white dots are actually each of these synapses. And we will discuss how these synapses develop and how they mature, and we are not quite yet to the autism, but we are getting there, trying to understand how synapse development is regulated by a class of non-coding RNA called long non-coding RNAs. And eventually we try to understand how the dysfunction of long non-coding RNA activity impacts synapse development, and that could be involved in autism spectrum disorder using some of these mouse model that we have, particularly in 16 chromosomal deletion mice. So this particular cartoon is from published quite a few years back in 2006, very powerful review by Erin Schumann. And what she described is that like every other, all the RNAs that are transcribed in the nucleus, they're transported through the neuronal dendrite and then localized at the synaptic compartment. And in response to the synaptic activity, in a very input-specific manner, a subset of the synapses are structurally modified, utilizing the activities of the RNA locally at the synaptic sites, and then they can make structural and functional modifications that are important for plasticity. So what we want to understand is that what are the role of these localized synaptic activities in terms of developing brain, in developing synapses. So what we know, how many RNAs are present at the synapse? So there are many non-code, there are coding transcript that are present in the synaptic compartment, identified by Erin Schumann and Kelsey Martin. Erin used transcriptomic analysis, and Kelsey Martin used the microarray analysis, and they estimated to be like more than 2,000 RNA are enriched at the synaptic compartment. There are non-coding RNAs, there are regulatory RNAs, and one of the most important one are the microRNAs. Gerhard Schratt and Ken Kosick, they used two different techniques. Schratt used the microarray, and Kosick used laser capture microscopy to take out the synapses, the dendritic spines, and then analyzed the distribution of RNA at the synaptic compartment. There are a lot of splice isoforms, like some isoforms are present in the synapse, but they are devoid in the soma. So those large-scale studies were done by Erin Schumann's group. So now today, I'm going to focus on another class of a regulatory RNA, called long non-coding RNAs. They are longer than 200 nucleotide. They're transcribed by RNA polymerase II, and they are polyadenylated. They can also be non-polyadenylated. And most of the polyadenylated long non-coding RNAs are more stable, and they are present in the cytoplasm. So some of the examples of the long non-coding RNAs are that are so far known is the, say for example, this particular long non-coding RNA called PANTER1. It is enriched in the CA1 region of the hippocampus and some of these cortical layers, whereas its isoforms are present only minimally in the CA3 region, and it's not present in elsewhere, except some amount in layer 2 and layer 3. So this is the in situ hybridization data from the Allen Atlas, and then it was analyzed by John Mattick and Tim Mercer. What they have shown is that these long non-coding RNAs are expressed in specifically in the cerebellum also, and they're present in the soma, they're present in the nucleus. So there is a subcellular distribution, and that you can appreciate from this particular photomicrograph. And you can see there are some small little puncta along the neurite, so they are present at the dendritic compartment as well. So they are cell type specific, and they have subcellular expression. So with this note, so we wanted to understand what are the long non-coding RNAs present at this synaptic compartment. So what we did a synaptoneurosome preparation, which is a synaptic prep, a biochemical prep that captures the synaptic compartment. And then we did it in Western blot analysis to know whether there is an enrichment of the synaptic compartment. And we see there is an enrichment of PSD95, CAM kinase 2, synapsin 1, all synaptic proteins, the devoid of histone and GFAP. And this shows that this is a pure synaptic enrichment. And then we did transcriptomic analysis from the hippocampus of the mouse brain, 3 independent experiments, several bioinformatic pipeline analysis, and followed by FTR analysis. And this shows this particular plot. On your right-hand quadrant, the red dot long non-coding RNAs, they're enriched in the synaptic compartment. By saying so, I mean that these long non-coding RNAs are more present or much more abundant at the synapse as compared to the soma. Whereas this little quadrant, this in blue, they're more enriched in the soma than in the synapse. But there is a large number of long non-coding RNAs that are present on the both side of the compartment, both in the soma and in the synapse. So there are many isoforms. I'm not going details of these isoforms, but just to appreciate the fact that a lot of these isoforms are very specifically localized at the synaptic compartment. For example, for this long non-coding RNA, which will come in my talk today, this we call this an NO1 RIC. It is this particular isoform is enriched at the synapse, and whereas this isoform is not present at the synaptic compartment. Similarly, PVT1, these 2 isoforms are very much enriched at the synaptic compartment where others are not. There are many isoforms of many long non-coding RNAs. The highest is the Gas5. They have 40 different isoforms. And we did an in situ hybridization to show that they are indeed localized at the synaptic compartment. So this is synapsin 1 is a synaptic marker, and this is the long non-coding RNA. Similarly, the other long non-coding RNA that will come to my talk is the PVT1 long non-coding RNA. They're also local in the synaptic compartment, and these numbers represent the colocalization coefficient. So they are localized at the synaptic compartment, and we are trying to understand how these long non-coding RNAs regulate the synaptic development. In order to understand that, we did a technique called in utero electroporation into the CA1 region of the hippocampus prior to synapse development. So we want to knock down the long non-coding RNA before the synapse development and ask what happens if this non-coding RNA is not present in the CA1 neuron. So this following in utero electroporation, you can see this particular CA1 neurons in this photomicrograph as well as in this photomicrograph. They are green because this particular neurons are expressing both GFP and corresponding gRNA or shRNA for their knockdown. So first we'll discuss the role of long non-coding RNAs in the excitatory synapse development. So we've screened many long non-coding RNAs and their expression profile. So we focused on two such long non-coding RNAs. One I will talk about PVT1 for excitatory synapse development and NO1-RIG for inhibitory synapse development. They express during the window of synapse development. And based on that, we selected this particular long non-coding RNA PVT1 and knock it down in the CA1 neurons in hippocampus. And we see the knockdown of PVT1 significantly reduced the excitatory synapse development, whereas there is no change in the inhibitory synapse development, both in the inhibitory synapses that form onto the soma or onto the dendrite. So it is one is the GABA alpha receptor, the other one is the GAD65. So we do the co-localization. Yeah, so we are not counting only GABA or only GAD65. We do that. We're doing the only GABA and only GAD65 count of co-localization between the GABA alpha receptor and GAD65. If they are overlapping, so we are counting each of them as a synapse. No, that is not done manually. We after the image and then image analysis and custom written algorithms for that. And we captured the like per micrometer how much synapses are formed. And then we did a transcriptomic analysis to understand what is the mechanism of synapse development. So what we observed is that, so we did a knockdown of PVT1 in CA1 region of the hippocampus, 3 independent experiments. And on your right, these are the transcripts that are upregulated following the knockdown of PVT1. And you'll see this particular one, Philip1, they actually negatively regulates the synapse formation. That is known from the recent literature in the PubMed. And there's a large number of transcripts that are down-regulated following the knockdown of the PVT1. And we did a gene ontology analysis. And here is the plot that shows that many of these important aspects of synaptic function, like for example, modulation of chemical synaptic transmission, the glutamatergic synapses, transcripts encoding synaptic signaling, chemical synaptic transmission, and also the presynaptic functions, the snare complex and the neurotransmitter transport, all of these are severely affected when there is a knockdown of this particular long non-coding RNA called PVT1. And then we wanted to see whether if you do the knockdown of the PVT1, whether that can regulate the synaptic activity or not. So what we did, so we knocked down this particular long non-coding RNA called PVT1 only at the synapse by a CRISPR-guided system that is expressed only at the synaptic compartment due to this dendritic localization signal called CAM3. And that will take this particular pin endonuclease guided by respective gRNA to specific transcript, either PVT1 or other long non-coding RNA SYNLAM or any other one. And then knock it down specifically at the synaptic compartment. And then we did a electrophysiology, that is whole cell patch clamp recording from these neurons. And what we observed is that the knockdown of PVT1 significantly reduced the, this is done in the neuronal culture, because we need to know which neuron also. And we see there is a significant reduction of MEPSC amplitude, but there is no change in the frequency. What it means is that, so there is a significant reduction of the postsynaptic function, but not the presynaptic function. If there is a presynaptic function changes, then we should have seen some of the presynaptic activity should have been changed. But if we do the global knockdown, like for example, both pre and the post, we see also the changes or reduction in the frequency. Which one? This one? No, not here. I have, like for example, another slide if I can share. So here it is not significant, but this is significant. Yeah, but so what we did the knockdown of the PVT1 only at the postsynaptic site of the neuron, but not at the presynaptic site. So there is no change in the frequency. But what I'm saying is that if you knock down globally, then without this SART system, then you will see the change in the presynaptic activity as well. There is a reduction in the presynaptic activity also. So indicating that these SARTs can specifically knock down the function of the long non-coding RNAs at these postsynaptic sites. And then we wanted to know if there is a change in the postsynaptic activity, how the long non-coding RNA is regulating the synaptic activity. So we looked at the surface expression of AMPA type glutamatergic receptor. There are two different types. One is the GLUA2 and one is the other one is the GLUA1. They're the two major subunit of AMPA receptor. So when synapses are matured, initially the developing neuron will have more of NMDA receptors, and then as neuron mature, then it will be more of AMPA type glutamatergic receptors. So most of the neurons that are mature, that we get the AMPA current from them. So then we measured the AMPA receptor subunit distribution following the knockdown of PVT1. What we observed is that the knockdown of the PVT1 significantly reduced the surface expression of both GLUA2 as well as the GLUA1 on the synaptic surface. So then, so what, in summary, if we do the PVT1 knockdown at the synaptic sites, it regulates the excitatory synapse development by regulating both the pre and the postsynaptic proteins. So now we would look to focus on that how long non-coding RNAs could regulate the inhibitory synapse development. So one of the long non-coding RNA that we focused here is the NO1-RIC. So this particular long non-coding RNA is similar to PVT1, also expressed during the window of synapse development. And we reduce the level of NO1-RIC specifically at the CO1 region of the hippocampus. And we see the significant reduction of NO1-RIC level in the CO1 neurons. Then we looked at the excitatory synapse development, and what we observed is that there is no change in the excitatory synapse development. Here we used PST95, which is a postsynaptic marker, and synapsin1, and we compute the co-localization of both PST95 and synapsin per micrometer of dendrite. And then we looked at the inhibitory synapse density. What we observed is the knockdown of NO1-RIC significantly reduced the inhibitory synapses that form onto the CO1 soma, and also in the dendrite. So there is a clear-cut reduction in the inhibitory synapses that we measured by GABA alpha receptor co-localization with GAD65. Then we did a whole cell patch clamp recording. First, we looked at the EPSC measurement, and there is no change both in the amplitude as well as in the frequency. But when we looked at the IPSC measurement, we see there is a significant reduction of MIPSC amplitude, but there is no change in the frequencies. So then we wanted to know how the NO1-RIC long non-coding RNA could regulate the inhibitory synapse development. We did NO1-RIC knockdown in hippocampus in CO1 region, and then did again a transcriptomic analysis. And here is the transcriptomic data. And what we observed is that there is a significant number of transcripts that are downregulated, and particularly the one that was very interesting because many genes are transcripts that regulates the ciliary functions. There are small protrusions in the cell that regulates the cellular signaling are downregulated following the knockdown of NO1-RIC. So why? Okay. So right now what we are doing is that we're knocking down one of the most important candidate, that ADMT16, that is also downregulated in one of the 16 chromosomal deletion mice, where this long non-coding RNA is also downregulated. So we're trying to do the patch following knocking down of this construct. So that will answer like how this ciliary function could regulate the inhibitory synapse development. Plus, we have initiated several cell biological experiments to understand how ciliary structures are regulated. So cilia, as I mentioned to you, are small protrusions that originates from the soma, particularly just exactly on the top of the nucleus, and the single cilia which is present in the hippocampal cell. The neurons can be multiciliated also, and these cilias, the single cilia that are present in the hippocampal cells, are also non-motile cilia. And currently, in collaboration with Sadhin Jana at NCBS, we are trying to understand how these non-coding RNAs are regulating the structure and the function of the cilia. So the interesting how we connect these particular aspects of non-coding RNA-mediated synapse development is this particular data from Ted Abel's lab. What they did, they took 16 chromosomal deletion mice and then did a spatial transcriptomic analysis. What they observed is that in the chromosomal deletion mice, there is a significant reduction of expression of this non-coding RNA NO1-RIC, both in the hippocampus as well as in the striatal region. And we are trying to understand how this ciliary function is regulated by NO1-RIC long non-coding RNA in two distinct types of neuron that are present in the striatum. That is, there are spiny projection neurons, D1 receptor expressing, D2 receptor expressing, and how that regulates the synapse development and how it contributes to autism. So this is in progress. So this is just some of the preliminary data that I have shown to you. And with the interest of the time, I'll stop here. And before I stop, so I'll acknowledge the students and the funding agencies. So Sharbani will be speaking in the symposium tomorrow. She will describe more on our work on how long non-coding RNAs are regulating synaptic plasticity by regulating the localized translation in the CO1 hippocampal neurons. And Balakumar Srinivasan, who did all this patch clamp recording for this study, I think today he'll be talking about his work on how microRNAs regulating the synaptic scaling. Kamakshi Garg, who did this all NO1-RIC study, how NO1-RIC is regulating the inhibitory synapse development. And Dipanjana will be presenting a poster on how early life stress regulates the long non-coding RNA function and how RNA modifications are important for early life stress that is have a direct impact on anxiety and memory. And Karthik, who is currently working on how long non-coding RNA regulates synaptic plasticity and how sleep deprivation impacts that. Sania is also working on the 16 chromosomal deletion mice, using 16 chromosomal deletion mice, how this synaptic development is regulated. And the new entries in our lab, Rina, Lakhong, they both working on this synaptic development using two different mice models. Pallavi is also working on synaptic plasticity and sleep deprivation, how it regulates the memory deficits and energy metabolism. And Gauri and Shreyas, their master students, they are developing in vivo imaging for long non-coding RNA transport at the synaptic compartment. And funding sources and collaborations, a very insightful discussion with Ted, Tim Brady, recently with Barbara Bartoni, that we had this CP grant to work on how FMRP and long non-coding RNA interactions regulate autism. And with Sadhin, we are looking at the ciliary function, Dasarathi, all the transcriptomic analysis, and with Sharbal, we are trying to set up some of the 2-photon imaging experiment to study how this synaptic development is affected in vivo. So with this, I would like to stop, and I thank you for your attention.
Very interesting. So I'm wondering how you're thinking about how the link RNAs are actually controlling this. Are they making packages with other mRNAs? Are they directly attaching to the ribosome and regulating translation? Or so it could be any of those. I wonder if you have ideas about this.
Yeah. So there are a number of ways the long non-coding RNA can regulate the function. One, Sharbani will talk in detail that where the long non-coding RNAs can function as a decoy for RNA binding protein. So this particular RNA binding protein called FUS that she'll be talking about, they are translation repressor. In response to activity, it can repress the repressor. So it activates the translation, that is one. Then Karthik in my lab, who is working on long non-coding RNA and the translational control and sleep dependent translational control and memory plasticity, he identified one particular long non-coding that could regulate the sponge for the microRNAs. That is one. They can in Dipanjana's project, what we observed is this long non-coding RNAs could be regulated through the RNA methylation, and that could regulate either the stability or the translation. So there are many ways long non-coding RNA can function, and one of the predominant role of long non-coding RNA is they regulate transcription as well by regulating the chromatin modifications. So one that we characterized is the RNA, the postsynaptic compartment. We characterized very well is the RNA binding protein decoy. So this long non-coding RNA in response to activity, so the mode of it binds to the mode of this translation suppressor. So the translation suppressor was earlier bound to the coding transcript. In response to activity, this most we haven't worked on the calcium dependence or not, but most likely it is dependent on NMDA activity, and that involves the calcium response as well. Yeah, so basically we haven't tested that, but based on hypothesis, it should be calcium dependent. Yeah, sure.
For the GABA synapse phenotype, have you looked whether specific subclasses of GABAergic synapses are preferentially affected? Because the effect per se is
Right.
modest, but you have sort of a 15% reduction. But if this was happening for PV cell synapses selectively, or for which could be because of the soma where you count it, so have you looked at subclass markers?
No, this is a very interesting question actually. So that is in our list to do. Sure, yeah. Mhm. Right. Right. Okay. So what controls the timing of the expression, which we don't? Yeah. Yeah. So one of the one that is known little bit about that, it's the BDNF expression regulate transcription of certain long non-coding RNAs. But what is the upstream regulation at specific time point? That is not. That is definitely an important regulation. It could be couple of different regulation. One, it could be definitely transcriptional regulation. Second, it could be their transport, their localization. Third could be their stability. Sonika Charan, right? So they need to connect it actually. Kedar, you need to connect it. It is connected, okay.
Good morning, everyone. Today I'm here to present my work on effect of antibiotic treatment on vocalization and vocal learning in zebra finches. So I'll start with the just brain-gut microbiome axis because this axis is mainly affected in various neuronal disorders. So here in our study, we are planning to disrupt this microbiome and see what happened at the brain-gut microbiome axis. So there is one study in zebra finches also where they have seen where they have seen there is a connection between the microbiome gut brain axis in the songbird also, where they have seen the cognitive abilities with the relation of the cognitive abilities with the microbiome of the birds also. So my main objective is to study how these antibiotics disrupting the microbiome affects this brain gut microbiome axis. So uh, for in our lab, we mainly works on zebra finches because these are the natural vocal learners. These are the excellent models to study vocal learning, vocal behavior. So these are the natural vocal learners. They have well-defined neural circuit and they exhibit behavioral flexibility, and even they have sequence genomes. And we can use this study as comparative and translation approaches also. So there both vocal learning shares similarities in humans as well as in songbirds. So there is as both of these take place during the sense, all these things have ma'am already told you, but I'm just giving the recap to get into the flow of the experiments also. So all these things occurs at the sensitive phase of the period, similar to human beings also. All this vocal behavior and all this relies on the social interaction. Moreover, the brain circuitry of the song circuit in birds and the human circuit also share similarities. So, so we are using all these capabilities of the zebra finches makes it an excellent model to study vocalization. So my aim is to study vocalization at the two levels: learned vocalizations as well as the vocal learning. Learned vocalization means the birds has already learned the vocalization. And in the vocal learning, there are different phases. All this I ma'am told you earlier, but I just want to get into the flow, so I'm repeating all these things. So this day one is the eggs. This day one is considered when the eggs are hatched, and this there is a sensory phase and the sensory motor phase. During these sensory phase, the bird listen to the songs, and sensory motor phase, they are listening to the song as well as they are practicing the song. So at the age from 19 to 120 days, they are considered as a crystallized songs. Crystallized songs means the song is not going to further have changes in the songs. So I'll be for the learned vocalization, we are using adult birds, this adult zebra finch birds, which are 120 days. So here the main thing is that the song is already crystallized.
This means their song, whatever the bird has learned from the parent, is now the plasticity has been achieved. So with this, all this background, the main objectives of my study is to study the effect of antibiotics in the learned vocalization as well as in the vocal learning in zebra finches. So this is the phases of the vocal learning in zebra finches. So I'll be starting first with the learned vocalization that's in the adult birds. So to achieve these objectives, we used three different approaches. We want to study the at the metabolite level, at the microbiome level, and at the behavior level, what changes are occurring. So we start with this adult male bird. We give treatment of antibiotics. In this study, we used a cocktail of antibiotics because our aim is to deplete the microbiome. So these studies, there are studies which have shown that this cocktail of antibiotics cause depletion of microbiomes in rodent models. So we started with this study in our zebra finches, and we gave the treatment of these antibiotics along with the amphotericin B, which is antifungal agent to prevent other contaminations. So we collected, we gave the dose orally, and then we do for the metabolomics, microbiome, and behavior studies. So for doing metabolomics and microbiome studies, we collected the stool samples before starting the treatment and after the treatment. For these stool samples were collected in this autoclave partition box. The bird was kept in the upper chamber and the stool sample was collected in the lower chamber. So after all these were UV sterilized for 30 minutes. All these things were done to prevent contaminations. So we collected stool samples for metabolomics and microbiome studies using this approach. And for the behavior part, this is a setup showing how we recorded the songs. So this is the female directed song. Ma'am has already told you about all these female directed songs. Here there are females, and this is the song, adult male singing the song. For this way, we have recorded the audios and videos of the songs for the behavioral studies. Coming to the result part, for the metabolomic studies, we collected the stool sample, and the sample were given to THSTI for the processing for doing untargeted LCMS. Untargeted LCMS, these results show us what are the changes in the what metabolites are altered in the treated birds. Once we know the metabolites are which metabolites are altered, we do the KEC pathway analysis. From that, we came to know about that this is the alanine aspartate glutamate metabolism is the main pathway to be impacted by the treatment. So this similar pathway is found in the autistic children also, with studies done by Wang et al. So this is the first section that showing that at the metabolite level, this pathway is impacted. Then we go for the microbiome level. In the for the microbiome, we collected the stool samples before and after the treatment, and these samples were given for 16S RNA sequencing. And from that, we found that in the treated birds, these are the control birds before treatment and after treatment. This is the treated one before treatment, before starting of the experiment, and this is the after treatment. And we found significantly decrease in the relative abundance of firmicutes, and in the case of bacteroidetes, it was increased. The relative abundance was increased. So from these results, we came to know about that this cocktail of antibiotics results in the increase in the relative abundance of bacteroidetes and decrease in the firmicutes. Then for the behavior study, how all these microbiome and metabolomics changes are affecting at the vocal learning, vocalization. So this is the adult male. So this x-axis denotes the time in milliseconds, and frequency is in the y-axis. So this is the song. This is the particular motif, the whole motif. And generally bird used to sing, this is the introductory note, which is bird used to generally sing this before the motif. So there are, in the motif, there are different syllables. These ma'am has already told you, but just going through it again. So the gap between the two syllables is known as the intersyllable level. So for the behavior level, we used to study all these parameters. For the syllables, we do study at the motif level, syllable level, introductory note level, and the number of songs also. So with the from the I just want to show how we do the recording of the female directed songs, so that this was the sonogram, so that you can correlate this sonogram with the song also. So you can clearly see these are the female directed songs. The bird is coming towards the female and then singing song. And just there is a pause between before introductory note and this the whole motif. Once we see sonogram and this video, we can easily interpret also that this at this time of point bird is singing, or it's introductory note, or it's giving a pause. Everything can be correlated with the sonogram also. So we used to analyze all these things. So from the effect of, we get this the number of songs get decreased, as well as introductory notes decrease with the cocktail while giving this cocktail of antibiotics. So this is the one study that I have done. Furthermore, this is the conclusion part that I have already told you. Then we found that we study on another drug called lincosamide. A case study was done in the autistic girl, which shows that there is administration of this antibiotics results in the abnormal movements, as well as there are several studies which have shown that this drug has known to cause autistic like features. So we also did the same study, this the revision part. Then we did in the adult birds for the learned vocalization, and then all these results are the same as we did for the cocktail of antibiotics. This is for the specific antibiotic which can cause autistic like features. So this is the adult part. I'm just skipping through it because I need to come to the more interesting part also. So this is the we started so for all this I told you is about the vocalization part. Now I'm coming to the vocal learning part. For the vocal learning part, we started treatment at 35 days. Ma'am has already told you the reason behind taking this 35 days. Then we have given treatment for this long period of time. And for the metabolomic studies, we did the stool samples at this 35, 80, and 100 and 120 days. Song recordings were done at 80, 100, and 120 days. Then this microbiome and metabolic sparks is yet to complete it. We have given sample for the process for analyzing them. Once we get them, we can correlate this with this behavior also. But we have completed the behavior part, where we have seen that yes, indeed, this antibiotic has resulted in the repetitive behavior, which is the characteristics of the autistic patients. So this is the father song. These are the syllables. Here, this we can see that in the treated bird, this syllable is repeating continuously. There is this is, I'm just showing you the results of one bird, which is showing the repetitive behavior. But we have done n is equal to net 10, and in all the 10 sets, we have seen that this syllable is repeated several times. So this is the behavior part we have completed, and we check the linearity and consistency also of these syllables of these songs also, and we found that we found the decrease in the linearity as well as consistency of these young birds treated with lincosamide for this period of time. So from the whole study, this is for this for the vocal learning, we have studied lincosamide, which have shown repetitive behaviors in terms of the vocal behavior. And in the adult birds, we have I have told you earlier that these are the main pathways impacted. We did all the microbiome, metabolomics, and vocal studies at learned vocalization level. Also, for the vocal learning part, we are left with the microbiome and metabolomics part, which is going to complete soon. So finally, acknowledgement. I am thankful to my supervisor for providing me guidance and support all the time. Whatever I want to do, she has helped me a lot doing that. And for the funding and BRC, DST, and our collaborators, which have given valuable input all throughout our experiments and discussions. And I'm thankful to all my lab members for their support throughout the project. And I'm especially thankful to Pooja, Anjali, Risna for helping me during the dosing of the birds. And I'm thankful to Shri and Isha also for analyzing the song data. For this, I conclude my presentation.
So did you, is there any, sorry, is there any record about, what happens to the brain structure after
So,
the treatment of
so
these
we
drugs?
have, once the experiments are over, we have collected the, we have done the perfusion and we have collected the brain and kept in the minus 80. We
So
need
you
to
still
do
don't
the,
know.
we, for the neuroanatomy part, we have to work
Check.
on it. But we have the brain that we can be, that can be done.
I guess it's a very general question about all microbiome analyses. How do you think the communication actually happens? Do you think it's to the brain, right? Do you think it's small molecules that are traveling? Do you think it involves some nerve activation that then feeds back? Is there any way to disentangle that? I assume the zebrafinch is not the easiest system to analyze this, but what's your view on how this might be working?
So for the microbiome part, we are just focusing how we are depleting the microbiome, and then there are several studies in the autistic behaviors, autistic children's also, where they have shown that the particular species of bacteria is increased or decreased, which has they are either correlated with the short chain fatty acids. The metabolites are involved in all these things, which are connected to the brain also through the vagus nerve system and all these things. So these things changing in the microbiome have resulted in difference in the at the brain level also, and that brain level is reflected at the social behavior and in the repetitive behavior also. So once we by depleting this microbiome, I have not shown you the this adult part. When we do deplete the microbiome depletion for 10 days, then the vocal behavior and the vocalization is impacted less as compared to when we deplete the microbiome throughout 120 days. Then we found different results, and this there is when there are different bacterial species, they have different roles. They they work in such a way that they inhibit the growth of some other bacteria which is useful for their brain to normal function. So this way they are connected. I hope I make you understand the question. Thank you, sir. Thank you.
Save some time. But we have plans to look at synapses and also neurogenesis in the brain. But then it will have to be done by other people because Sunita has decided to leave.
Oh,
she got a job.
Okay.
So
Congratulations.
To hold this. I don't have to move. It is there. You can hold it like this. Okay. How to hold? Like, is this on it? Yeah, it's on. On. Let's remove the slides. No, it will not work for your slides. You have to. You want slide movement through pointer? You want to. Otherwise, is okay. No, no, no. I don't want now. You don't want now. After, yeah, after. I will buy me sleep off. I have a connector. I have a connector with me. That connector with me, I will take it. Neurology. Neurologist. Yeah, pediatric neurologist. Neurologist.
Neurologist.
Bluetooth. Bluetooth. Okay, where's this pointer? Okay, thank you. This is working, right?
Everybody, after 45, we have a patient for Shomer Sridharam. She's a pediatric neurologist, and I think I was very good access to the possibility of autism, the ASD. So we get to hear from you. And the time is 30 minutes to tell and 20 minutes and 10 minutes for interaction.
Okay.
And the way is there.
So just to confirm, my time is 20 minutes for talk. Okay. Thank you, ma'am. And first of all, I would like to thank Dr. Saurabh for inviting me to deliver this talk. Okay. So I will be speaking about the clinical aspects of autism spectrum disorder, mainly on diagnostic evaluation and therapeutic strategies. So I will not be touching much on the research in this particular area. I will be sticking towards the clinical features, a brief introduction of autism, clinical features, how to evaluate and come to a diagnosis, etiological evaluation, and some of the treatment strategies. So coming to the introduction, autism spectrum disorder, as you all know, it is a highly irritable and heterogeneous neurodevelopmental disorder. So the clinical manifestation composed of mainly the deficits in these four core areas. They will have social communication deficits, cognitive impairment, repetitive behavior, and various sensory anomalies. So why we should know about this particular disorder and why we are holding such a symposium on this autism spectrum disorder is because the prevalence of ASD is actually increasing. There is a debate whether the number is increasing or whether we are diagnosing it more. So as per now, the recent survey which is conducted in India, around 1 to 1.4% of children in the age group of 2 to 9 years has autism spectrum disorder. And in the North America, the rate is around 1 in 36. And it is 4 times more common in males than in females. It's maybe because that in the females or the girls, it is not well diagnosed. That is one reason, or the second reason is because of some protective effect for the female gender. So clinical diagnosis, it is basically a diagnosis based on a set of clinical criteria. So there is no objective marker for the diagnosis of autism spectrum disorder. No imaging, no blood markers. So the clinical symptoms vary depending upon the age, cognitive ability, language ability. So these three factors determine how a child presents to you. So we widely follow the DSM-5 criteria in our institute, and some other places they follow the ICD-11 classification. So this is a table which shows the main core features of autism spectrum disorders. Mainly two aspects are there. One is a social communication deficit, and second is a restrictive repetitive behavioral patterns. So among the social communication and this language ability, there are three criteria has been defined under that. And the child should satisfy all the three. That is social emotional reciprocity. Deficits in social emotional reciprocity. That means two back and forth conversations, the ability to set a back and forth conversations, sharing of emotions. The child may not be able to share his or her emotions with the parents or others, and may not be able to understand others' emotion as well. So they have not only their verbal ability or language ability is compromised, they have a difficulty in the non-verbal, that is showing the gestures as well. So they will not be able to understand the body language of the other person and may not be able to show the body language to communicate, to supplement their verbal communications. So they have a deficit in recognizing others' emotions, showing emotions, etc. So the difficulty in adjusting behavior to suit various social contexts. They may not be knowing what to do in when they're attending a classroom or attending a marriage ceremony or any other funeral function. They may not maybe behave out of proportion to that particular context. In all these individuals, all these features may not be there. These features may vary. Sometimes they may have very good verbal ability, but they may not be able to follow the rules. They may not be able to have a good disclosure, that is proper conversations between each other. They may interrupt or they may talk like a little professor according to their own interest. So in this slide, you can see our therapist is trying to engage the child with ASD, but he is not even attending to the name call response. The child is not keeping eye contact. The child has poor social engagement. Here you can see child is playing by himself with the toys and not attending to the therapist. So there is no shared interest in these kids. So lack of eye contact is one of the earliest features with which we can recognize a child with autism. Another important second criteria, that the core criteria that the child must meet, is the presence of restrictive repetitive patterns of behavior or interest or activities. So there may be the repetitive behavioral pattern could be motor movements. You may have just seen the children with hand flapping, hand wringing movements, or doing this hands like this. Or they may repeat whatever you are saying. But if you ask the child, what is your name? The child will tell, what is your name? So that is repetition of others' conversation, which is termed as echolalia. They may have insistence on sameness or inflexibility or adherence to routines. They may have a fixed timetable. They may walk on particular aspect in the house. They may want to present their food in a particular way. They may eat from the same plate or a same color. They have preference for some color. So they have insistence on sameness, inflexible adherence to routines, highly ritualized behaviors, etc. Fixated interest. Out of many things, they may be fixated to one particular thing. I have a neighbor who is mild autism. He's fixated about mainly the places, geography. He knows the capital names of all the countries. Some others, it might be cars. And for others, it may be dinosaurs. Another problem is the sensory issues that they may be facing. So the sensations we feel and we respond to various sensations like vision, hearing, smell, this vestibular system, proprioceptor inputs in a different way. So they may have a sometimes very much sensitivity to these type of stimulus, or they want these stimulus more. So they have either a sensory avoidance behavior or they may show a sensory seeking behavior. So sensory avoidance behavior is when they hear a loud noise like the sound of a pressure cooker whistling or the mixie, they may not like it and they may close their ears. Or sometimes they may use rattles to make the sound and try to hear it often. So these are types of, or they may see a child with frequent mouthing behavior or goes on smelling things around. So these comes under the sensory. So out of these four, at least two of these criteria should be met. And these features should be there during the early developmental period. Sometimes these features may be mild, may not be recognized, may be recognized in adult. But these symptoms should have their onset in the early developmental phase itself. So you can see this child. I think the audio is not working, but he is repeating whatever the therapist is telling, so that becomes echolalia. This is another peculiar behavior is you can see this is another autistic child is playing with the doll. This is part of autism diagnostic observation schedule. This is ADOS. Goes on trying to poke the eyes of the doll. So this eye poking behavior, they may poke their own eyes or the doll's or others' eyes as well. So this is eye poking behavior. This is very peculiar in autistic children. So another thing is the fascination for rotating objects. They may be looking at the fan or any rotating wheels, etc. You can see the child. He is not actually playing with the car or the toy. He is actually lying down and seeing how this wheel rotates. So this is a very fascination for the rotating or revolving objects. This is another child. This child is actually a case of Rett syndrome. You can see the repetitive motor behavior pattern, rocking movements to and fro, and you can see the hand movements, clenching, fisting, and all. This is very classically which is seen in Rett syndrome and other causes of autism as well. This is the one which I was telling about the auditory seeking behavior. This is a rattle. She's continuously actually playing with that to make the noise. So this is an auditory seeking behavior. This is another peculiar way of looking at things by the patient, seeing through the corner of the eyes. Like they may not look straight, they may look like this out of the corner. Sometimes they may look at you directly, which is known as intense eye pointing. So these are some peculiar features which can be seen in children with autism. You can see her, how she is looking out of the corner of her eyes. So once we have to elicit all these history, whether these behavior are present, and look whether it satisfies the DSM-5 criteria or not. Not only we should ask the history, we should actually observe the child for this particular behavior. And then we have to come to the examination. As a neurologist, then usually what we do is we try to do a complete head to foot examination, anthropometric head circumference, look for neurocutaneous markers. Some of the conditions like tuberous sclerosis will have this marker, and we can readily diagnose these patients. Look for facial dysmorphism, skeletal deformities, and neurological examination. So these are examples of various causes. Facial dysmorphism is shown in 3 patients with different causes. One is a Fragile X syndrome with a large head and large ears. This is a chromosomal abnormality. You can see the significant facial dysmorphism like depressed nasal bridge, overhang in nasal tip, low set ears, etc. So by general examination, we could pick up these clues and look whether it will fit into particular syndrome or not. So a detailed interview is required, which requires a history and observation. And short clinical visits may not allow even a skilled clinician to accurately recognize symptoms of ASD. And we require a formal assessment, look for ASD, look for the language ability, cognition of the child, adaptive abilities, and sensory status. So these are the various standardized assessment tools that we do in case of autism. One is the AIMS Modified England Diagnostic Tool for Autism, ASD, which is Professor Shefali has developed that. And the gold standard is the EDOS, second edition, which requires at least minimum 1 hour to administer and half an hour for scoring itself. Then social responsiveness scale, which assess the social domain. Then the childhood autism rating scale, majority of the speech therapist usually does this childhood autism rating scale. Then for assessing intelligence, we have various tests. For adaptive function, we can look into Vineland Adaptive Behavior Scale. Once we make a diagnosis of autism, then we have to mention whether the child has intellectual disability or not. Majority of the parents feel that the child is very special. We use the term as a special kid only. They have, but most of them have intellectual disability. Very few have this high IQ. So they may perform better. And mention whether they have language impairment or not. Sometimes they may be completely non-verbal. Sometimes they may speak, but they may not be able to identify the sarcasm, idioms. They may not be able to narrate a story. They may have catatonia. They may have coexisting medical conditions like gastrointestinal disturbances or other neurological problems like seizures. And mention what is etiology. It's a secondary to some environmental influence or is secondary to any genetic factors. Again, we have to grade severity. We can classify them into mild, moderate, or severe, but it is not a quantifiable scale according to this particular DSM-5. It is based on your objective assessment. So comorbidities, there can be many comorbidities like psychiatric manifestations like mood disorders, anxiety, depression, bipolar disorder. Common comorbidity is ID and ADHD. Epilepsy is seen in 7 to 46%. They can have sleep disorders, which is actually very much problematic for the child as well as for the family. Aggressive behavior and injurious behavior and various forms of eating disorders and food refusal. Sometimes it may not manifest in one point of time. As the child grows, the neuropsychiatric manifestation accrues. So the co-occurring conditions evolve over time. That will lead on to particular selective clinical phenotypes, which will influence the prognosis and treatment. So we have to detect these comorbidities. Next comes etiological evaluation. So children with the diagnosis of ASD should be evaluated for potential etiology. We can use a series of tests. So based on history and examination, you will arrive at a particular diagnosis, and you may think that these particular etiologies are possible, and then you plan your investigations accordingly. So which includes genetic testing, imaging, EEG, metabolic testing, evaluation of vision, and hearing evaluation. So what causes, to know the etiological evaluation, you should know what causes autism. There are two aspects. One is the genetics and other is environment. Tomorrow, I think we have more detailed discussion on the genetic aspects of autism. And also because of the influence of environment and genetics, the child can have ASD. So environmental factors, there are many proven risk factors. So these risk factors cannot be considered causal. So they could be independent or contributory for autism. So these environmental factors act as a trigger by having some epigenetic effects or induce inflammation or oxidative stress or can lead on to hypoxic ischemic damage. So some of these environmental risk factors have strong evidence for causing. In the x-axis, you can see it is a rare one, and this is a more common. Folic acid has a protective influence. Then other risk factors include maternal obesity, preterm birth, older sibling with ASD, increase in paternal and maternal age, and hypoxic ischemic encephalopathy. Evidence is inconclusive for exposure to pesticides, air pollution, and not supporting as a cause for autism spectrum disorder is a vaccination and other assistive reproductive techniques, cesarean section, etc. So coming to genetic factors, the twin and family studies have estimated heritability from around 40 to 90%. So for monozygotic twins, it comes to around 98%. For dizygotic, it ranges from 70 to 90%. And if one sibling is affected, the risk of other sibling is from 4 to 14%. So there are some of the genes which are more than 100 genes which is confidently associated with autism. And some of them very common alleles, common genes, which is small effect sizes. So we can search for these things, but at least Mendelian disorders, at least we have to screen to identify a cause. If you evaluate genetics in these children, around 10 to 20% of them can have rare de novo variants, and it increases, the yield increases to 20 to 30% if it is a female child, those with intellectual disability, seizures, and those with multiple unaffected siblings. The chance of detecting rare de novo variants, heterozygous de novo variants, is more in these situations. So coming to molecular mechanism, so these genetic factors and environmental factors work together or independently to cause some of these abnormalities in the synapses, chromatin modification, regulation of gene expression, or maybe an abnormality in the glutaminergic neurons. Some of these genes have got effects on these multiple functions, which is spatially temporally defined and can happen at any stage of development, and will lead on to the socio-behavioral phenotype which we see in the patient. So it could be an excitation inhibition imbalance where there is increase in glutaminergic system and reduce in inhibitory neurotransmitter like GABA. So the synaptic functions, we can see many genes and the presynaptic and postsynaptic neurons, which is also responsible. For example, we have Fragile X syndrome, that is FMR protein, the TSC1, TSC2 leading to tuberous sclerosis, mTOR, SYNCAP1, etc. Again, the chromatin and transcription regulation, we have the SPEED10, MECP2, UBE3A, which are commonly associated with autism. These genes are not only causing autism, but can cause other neurodevelopmental disorders as well, including epilepsy. So the yield of genetic testing for chromosome microarray is 5.4% to 14%, which can detect deletions and duplications. Whole exome sequencing studies have shown a yield of 8 to 20%. For Fragile X, it is 0.45%. Whole genome sequencing, we are doing mostly in the research setting. Most of these guidelines recommend chromosome microarray as a first-line investigation, and the national policies insurances will give only if chromosome microarray is ordered first. But in our institute, we practice whole exome sequencing because now the newer software can detect the deletions and duplications. And if it is negative, then we go for chromosome microarray. This, of course, it depends on your clinical diagnosis. So what is the potential benefits of establishing a genetic diagnosis? It will provide diagnostic closure. It will help us to prognosticate, monitor certain conditions which can occur in the future if the patient has got an underlying genetic problem. It will avoid ineffective or potentially harmful treatments. And we can give etiology-specific treatments. So since the time is over, I will actually skip on to, we can do MRI, not in all cases, but when there is a typical presentation, when there is seizures. Okay. Okay, fine. Okay, thank you. Okay. So MRI brain is not indicated for all cases. It is when there is a typical regression, when the child has microcephaly or macrocephaly, seizures, or any other neurological manifestations, obviously we should go for MRI. Otherwise, MRI is not routinely indicated in clinical practice. Whereas in research, we can do diffusion tensor imaging, functional MRI, and volumetric analysis. But the MRI has now not yet become an imaging biomarker at present. We can also look for other neuroimaging features like atrophy of the brain, or look for hypoxic ischemic damage, or any congenital brain malformations, or whether it is secondary to a neonatal encephalitis, or any intrauterine infections. So these children will have additional manifestations, any neurological signs, seizures, etc. will be there. Such situations, we have to definitely go for MRI. Metabolic testing is also not routinely recommended because yield is very low. Only very few conditions can primarily manifest as autism and neurodevelopmental disorders. So it is pursued only if you strongly suspect or if you have an MRI picture. EEG is not routinely recommended in case of autism spectrum disorder when the patient has seizures or if you want to rule out an acute epileptic aphasia, then we can do EEG. Qualitative EEG is used for the research purpose and is being explored as a biomarker. And sometimes in Rett syndrome, they are associated with seizures and they may have continuous activation of epileptiform discharges in sleep. So management of these children, the goal is to actually minimize the core deficit, to reduce associated impairment like seizures, neuropsychiatric manifestations, maximize their functional independence, and eliminate, minimize, or prevent any problem behaviors. So in each individual, we have to set an individualized treatment plan, which is developmentally appropriate, should be intensive, and relevant to their treatment goals. If it is a toddler, it is better to place them in a preschool, daycare, or Anganwadi. For a school age, they should have some educational settings and then have their therapy. So types of interventions also differ. We don't have any national guidelines framework for a particular type of therapy, but district early intervention centers are operating under RBSK, but the extent to which that reach the target population is very minimal. So we have comprehensive treatment model where all the multidisciplinary team will come together and work on various aspects like speech therapist, occupational therapist, psychologist, etc. Or it could be focused intervention targeting one particular function, like improving the social skill or sensor integration therapy, etc. Modality of delivery, whether we are giving direct one-to-one, one therapist, one child, or is it a group therapy sessions, or the therapy is provided by a professional or by parent, whether it is school or home setting, or whether it is done in a public, not-for-profit agencies, schools, or early intervention services. So the early intervention program is the one which is shown to have considerably, we have evidence towards their very good outcome, and which is based on either applied behavior analysis, developmental therapy, education models, etc. But these centers are very minimal. They require a very intensive approach up to 24 hours per week minimum. And in our situation, because of the lack of sufficient therapists and infrastructure facilities, not all children are getting these therapy. The focused intervention, again, it could be either speech and language intervention, sensor integration therapy, fine motor and balance training. Now we come to the medical management. The various autism does not have a cure as such. There is no medication per se for autism. But if they have accompanying hyperactivity, neuropsychiatric manifestations, obsessive compulsive disorder, yes, we do have pharmacological treatment. Then the recent articles have come regarding the application of oxytocin, cannabidiol, trofinetide, which is approved for Rett syndrome. So the prognosis, the communication and social skills improve over time. Repetitive behavioral pattern change over time. Social difficulty is more for ASD with language impairment than in those ASD without language impairment. And in those with ID, the social skills and the adaptive functions are very poor. Yeah. Concluding, autism spectrum disorder is a heterogeneous disorder with multimorbidity. The diagnosis depends on the clinical diagnostic criteria by DSM-5 or ICD-11. Till date, no biomarker has been identified. Evaluation is by standard assessment tools and etiological evaluation. These people should require a life cycle approach with an aim of functional independence. Pharmacological therapy for comorbid medical conditions. And in future, we are looking forward for a precision medicine approach. This is our center, and I'm thankful for these therapists and my research fellows. Thank you.
Soumya, very interesting talk.
Thank you.
So my question is, so you have mentioned that there's a increased prevalence of autism, and you also mentioned that it could be because indeed there could be increase in the number of the child, or could be because of increased better diagnosis.
Yeah.
So my question here is there any indeed increase in the number, or is because of the, I mean, which one is the main cause?
Some of this actually, if it is based on a hospital-based study or very small but the population, we may find that the prevalence is increased. But when we go, some of the studies which is population-based has not found a definite increase in the prevalence rate. So probably it's because of the better diagnosis and accurate early diagnosis. Probably the number has gone up. And what CDC has mentioned is probably because of this claim for insurance or other things. Probably they are early diagnosed. And in research setting, probably we may pick up these cases much more rather than a real world scenario. So these are different aspects. So we don't have an answer. And the second possibility, there might be a mild increase in the prevalence as well because of this increase in the genetic mutations and environmental influences also. So both these effects are there.
Yeah, let's say the child is suffering from both autism and the intellectual disabilities. So where will it be categorized? Will it be categorized as ASD or ID?
That is a very good question because earlier in DSM-4, such a provision was not there. ASD, if you have to label, there should not be any comorbid conditions. In DSM-5, we have the liberty to attach as many comorbid conditions. So one of this diagnostic criteria that they mentioned that these symptoms could not be explained by intellectual disability alone. So whatever this deficit is there, it should be over and above what an intellectual disabled child should cause, ID should cause. So they should have much more effect on the social deficit. So the ID is there, which is very common in this, but the social deficit should be more. And then we can categorize that as ASD. But ID can cause most of the symptoms, especially severe symptoms. They may not be socially engaged. So it is very difficult to pick up autism in such intellectually disabled children.
Okay, this last question. In your study, how many of the children are because of the environmental factors compared to the genetic causes?
Environmental is we have only risk factors. We don't have a particular direct cause. For example, if maternal intake of valproate, the child is having during the pregnancy, the mother is taking valproate. There is an environmental risk factors, but we cannot be so sure that is the reason for the autism. So it is a risk factor. We can tell environmental factors are present as a risk factor alone. Then second is intrauterine infections. That we can attribute it as a cause. If it is found to have any congenital toxoplasmosis or rubella or cytomegalovirus infection and the child has autism, of course they have other neurological problems also, then we can attribute as a cause. Again, hypoxic ischemic encephalopathy, we can attribute as a cause. So that is actually less as compared to the genetics as a direct causal relationship.
Thank you.
Thank you for the nice talk. Very naive question from a preclinical person.
Okay.
So it seems to me that the behavioral interventions have some positive impact on the children and that the pharmacological interventions rarely do anything but help with some comorbidities. But has it been tried to actually combine a pharmacological intervention with behavioral therapy to potentially increase the receptivity of the child to that behavioral therapy and maybe also make it better from a cost perspective if you need fewer sessions or something like that?
Yes, definitely. Actually, there are not many drugs which are approved for children less than 6 years of age for the pharmacological management. But some of these children becomes very much hyperactive, disruptive behavior. So we use off-labeling in young children as well. What we recommend initially is the behavioral therapy. But for the behavioral therapy to be effective, they should be sitting and should be receptive, as you suggested. In them, we have to give them pharmacological management, and then once the behavior has settled a bit, then the behavior intervention combined approach will definitely yield a better result.
Have you seen come across how much masking has relevance with ID and diagnosis when it comes to the disparity within the male and female population diagnosed, and also the
I
intervention?
didn't get your question. Can you?
Yeah, sure.
My ears are blocked, so it's
Okay.
difficult.
So my question is, how much of masking that an individual that is in the spectrum, so how much of that have you seen being irrelevant with being diagnosed as a
Masking means those who are not being diagnosed.
no, masking is a person, an individual, and not ASD individual who can learn the rules of the society and just go with it. Like masking,
Okay, that is
most
actually
of
they get adapted to the situation. That is what you.
yeah, like in high functioning
A high
ASD
functioning definitely in
individuals.
our scenario, where the social what expectations
Yeah.
in a case of public speaking or coming in the front, etc., is not that much.
Also in a
So,
conversation, going back and forth.
fourth conversation.
Yeah.
Even if they avoid, if they are good in studies, people don't try to involve them in any of the activities. They will label them as shy or silent.
Introvert.
An introvert, shy or silent. So they usually gets adapted to that situation, and they prefer to their particular life skills. Some of them may not have any problem. They may advance in their life, but when it comes to the socially engaging situations, then it becomes awkward. When the social demands exceeds their tech adaptive functions, then people start realizing that there is something problem. But others may not be able to realize, especially in the school setting. When they come towards us for some other reason, then we will be able to pick up by direct interview that we have actually a late diagnosis, especially in girls
Yeah.
in school age around the high school. When they come, they may be actually the silent observer for their sibling. The sibling manifests with autism, and when we look at the elder sibling, we find that the elder child is also autistic, but high functioning. So they may not come to us with their complaint, but when we screen the elder, they will have. So then again, parents, they are most of them, if you actually interview them, some of them actually fits into either ASD or broad autism phenotype. So these are all there in the individual, in the society.
Thanks.
Hello, ma'am. Rather than any targeted therapies, there are any softwares available to support the target therapy or to like change the target therapy. As you mentioned about precision medicine, like is there any softwares available? So like quizzes or anything like patient is having the same response as targeted therapy in the brain, like plasticity or something.
Okay. This actually, in case of rehabilitation, many softwares have been developed, and also virtual rehabilitation is also coming in the management of autism spectrum disorders. For the language functions, there are many alternative augmentative communication devices. Softwares are already available, which are regularly used by the speech therapist. And for also for the social skill training, the virtual rehabilitation, reality-based rehabilitation techniques are also coming up. So that also being employed. So it is there.
Like according to your opinion, like how much it can like potentiate the targeted therapy, or it can replace the targeted therapy in the future?
That I don't have that much of experience because AAC is definitely alternative augmentative communication is definitely has a great impact. Regarding the VR and other things, I'm not very sure. We are not practicing that in our because we are more focusing on evaluation. We are not providing that much rehabilitation. We are actually providing parent-based home interventions because we don't have sufficient number of staff.
If you permission, can I take one
Okay.
point? First of all, that abnormal fusion, that abnormal being a psychologist, I don't know when to actually we are telling that there is a diverse social attitude. So for the term abnormal fusion and telling is hinted in a identification which is not approved, socially approved way maybe. But when we are telling clinically abnormal, that means particular clinical diseases for that reason I'm telling. One. And second point is in your assessment tool, Indian scale for assessment of autism should be mentioned because it is now for the certification.
Is required.
Must. Even not cards, even not rules for that reason. Please don't mind.
No.
Thank you. I think no instruction. She's not only lady, but we have pediatrician and neurologist and really nutrition in autism. So, again, I'm telling, she need no instruction. To her. Yes,
Nice talk.
Good afternoon, everyone. I thank Dr. Krishanu, Saurabh. Yeah. And NBRC for organizing such a thing and making me a part of it. Guess my journey for looking after autism is more than 3 decades and for these children. And also like being International Society for Autism Research Global Senior Leader for India for many years now, and editor of Autism Journal, I get to see the across the globe what is happening. Most of you are basic scientists, so I will just try to talk in a way, there is a, I mean, in a way which makes it interesting for you, and just to understand little thing. There's only one message per slide. Basically, there's an interface between autism and epilepsy. What is leading to what is not clear. Okay. So what is predisposing to what is not clear. But it goes bi-directional. I have nothing to disclose. I made the talk into 5 points, whether you know the disease prevalence of what is the prevalence of epilepsy in autism and what is the prevalence of autism in epilepsy. To know the challenges, the diagnosis of a seizure in a child with autism and vice versa. Number 3 is to know various genetic conditions that predispose both to autism and epilepsy. So we have to understand that what is leading to what is not clear. And to know the risk factors for epilepsy in autism and autism in epilepsy and their mimics, because there certain mimics are there. And to know the treatment modalities for the overlap conditions as well. So autism spectrum disorder with the current estimates of CDC, March 2023 is 1 in 36. In the INCLIN study, in which I was the network coordinator and the right PI, which we did the data collection in 2011 by population proportionate by sampling, pan India, it was 1 in 89. Mind you, the data collection is 2011, though the paper is in PLOS Medicine 2018. And it is seen more in boys than girls, as Soumya has already mentioned. But let me tell you, initially the ratios were around 4.5:1. But in the CDC of 2023, it is going around 3.5, 3.7:1. So the ratio is coming down. And somebody asked whether it's somebody asked Soumya whether there is it's just that increased awareness and more diagnosis, which is there. No, there is a genuine number of increase. There was a paper in which they what all you can mistake autism as, with intellectual disability, or hearing impairment, speech language disorder, all those total N before 1990s and post 1990s were taken. And so it was not that you were misclassifying or missing them. If you're missing them, you're misclassifying somewhere. There's a dramatic increase in these numbers. And other kind is epilepsy in children. Epilepsy in children, it's 1 out of 150 children are diagnosed with epilepsy during the first 10 years of life. And Indian prevalence rate is 6.99 per 1,000 population in rural areas and 5.48 in urban areas. Now this epilepsy and autism, autism and epilepsy. Now let's have it. So epilepsy and autism, the pool prevalence of epilepsy in autistic children is 7%. And in autistic adults, it is 19%. Prevalence of epilepsy in those with intellectual disability is higher. It's 21.5% versus those who do not have ID. As Soumya mentioned, the current DSM-5 says that you may have autism spectrum disorders with specifiers. Yes, so you can have autism spectrum disorder with ID, and I will tell you about one of my data set, which I'll be coming to shortly. Increasing age, female gender, speech problems, and lower socioeconomic status are other risk factors. And 150 participants in autism, they followed in another study, and 22% developed epilepsy, which was having those with poor verbal abilities. As regards risk in siblings, those who have a history of epilepsy had a higher chance of having autism than those who did not have a sibling with epilepsy. This was the data I was talking about. 1,872 children over about 3.5 years, which we had analyzed. We had 11.5% of our patients had epilepsy. On seeing the other side, we tried to see a couple of our West syndrome patients. Out of 55, we found 24% of these had autism. So West syndrome is a very common epileptic encephalopathy seen in early in infancy. This was another data set which we had. There were two sets of data in this collated. And overall, we found that 1/8 of the children of autism have epilepsy to various data sets. This was another data which we had done and seen sleep issues in children with epilepsy. We had done a CSHQ questionnaire, then we had done a PSG in all of them. Mind, it was a very difficult study. They don't sleep at home. We made them sleep in the hospital. So it was really, really difficult. But in that, we obviously came to know that sleep problems are very common, but 15.5% of them had epilepsy. As we all know, tuberous sclerosis has a lot of TANs you have, and you have a lot of autistic features in that. So we took 26 of these children, out of which 17%, 26 children we saw, then they had 18 children were fulfilling the criteria of DSM-5. Rest all had only autistic traits, they were not fulfilling. Out of 18, 17 had drug refractory epilepsy. So you're looking at the other way around. So what we did was a collected data, did a search from PubMed and Google Scholar, and autism and epilepsy was put. And in 2013 to 2023, we found 527 articles. And then after excluding duplications, title rejects, abstract rejects, we zeroed on 75. So I will be telling you this autism and epilepsy prevalence of 75 articles. So epilepsy prevalence is significantly higher in ASD than compared to general population, yes. And ID is a major risk factor, epilepsy in ASD. This was one systematic review in which they had 74 studies reporting on 2,83,549 patients. And they found that overall period prevalence of autism in epilepsy was 9%, including the syndromic epilepsy and those with developmental delays. But when you exclude them, it became 8.1%. So anyway, it's clear that ID is predisposing. So to summarize, the prevalence has been ranged from 5 to 38%, and autism alone was 2 and 8%. But if you have severe ID, it goes up to even as high as 27%, and those who have GDD ID and cerebral palsy, it goes up to 67%. The summary of this 75 papers was that epilepsy prevalence is significantly higher in ASD, and prevalence of autism in people with epilepsy is higher compared to the general population. Accompanied developmental delay and syndromic epilepsies are major risk factors. And ID is a major risk factor for epilepsy and ASD, which we have very clearly seen. My second objective. Autism and epilepsy, what is the interface and what are the diagnostic challenges for the interface? It's not only that on studies, in clinical practice, we find it difficult to many times. So their diagnostic challenges are there. So there were 140 children who had some staring spells and they came, and then they did a video EEG and they found only 22 had epileptic seizures and rest were non-epileptic. But what happens in general practice, many of those children who are not having epileptic seizures but the stereotypies and all which are common of autism are getting anti-seizure medications which are compounding and causing more side effects. So it's important for us to understand. The most common seizure was an atypical absence. And when we compared the average duration of episodes between epileptic seizures and the non-epileptic ones, we could make out that the non-epileptic ones were longer duration, had more frequent spells, and then they were responding to verbal stimulation and suggestion. What happens is stereotypies are these involuntary, fixed, patterned, purposeless, repetitive movements like head banging, head rolling, body rocking, hand flapping, or they may have some gestures, some postures, or maybe having vocal sounds and utterances, and often occur when the child is less engaged. More maybe consciously suppressed or decreased with distraction in these children, and they're more common in children who have autism or intellectual disability or schizophrenia or Rett syndrome. And obviously it is not includes what is in epileptic automatisms, in which you may have a person who is not regained consciousness and all but is moving the limbs and not remembering the limbs movement and all later on. And the other thing is you have to see what are the mimickers, because some of the frontal lobe seizures can also have these kind of movements. So for you to understand, these are stereotypies. If you see, the purposeless movement is repeating again and again, and that child also. This one. He just wants to rock the chair. And he is going on staring, the right side one. So, these are abnormal movements, and many times practitioners start them on anti-seizure medications, which cause more problem than benefit. Okay, sorry, I missed the one slide. 1 second. Yeah. So autism in epilepsy has a diagnostic challenge, and 32% of children with epilepsy may have autism when you see on their questionnaires. And if you try to see the type of seizure semiology and number of seizures per day were not different if those who had autism with epilepsy and those did not. But yes, the age at onset was earlier, and number of anti-seizure medications required to control was more if they actually had both autism and epilepsy. The common that it depends generally. I mean, when the beyond the neonatal period, generally you can be actually giving, can you giving phenytoin or valproate and all, but nowadays people are generally giving levetiracetam. But valproate is something which is broad spectrum and can be given. And when these focal seizures, then carbamazepine or oxcarbazepine, first line. But levetiracetam is given in everyone. So the common mimickers for autism will be Landau-Kleffner syndrome or SPAS. I will mention about SPAS because there is something about its peculiarity, both for diagnosis and management, and various epileptic encephalopathies, intellectual disability, and many, many more you can see, including autoimmune encephalitis and also. It has a wonderful range. So you need to see the child holistically by a pediatric neurologist. So you may have developed, now what is called as DE-SWAS is a developmental or epileptic encephalopathy with spike wave activation in sleep. That is what is important. Because in that you have various combinations of cognitive, language, behavioral, or motor regression associated with marked spike wave activation in sleep. So what happens is they come with recent onset behavior issues, and then we have to have a sleep and awake EEG both. If you do only an awake EEG, you will miss. Sometimes your sleep EEG will be completely abnormal and the awake EEG is totally normal. Don't have to go into detail. I just wanted to mention that. So in the sleep record of the same patient, you're having so many discharges, right? You have different formula and all that. You see the SWI index and all that. We don't have to get into it. I'm just trying to tell you that you have a sleep record which is having so many discharges, and the same child's awake record was totally normal. So if you were doing only an awake record, you will miss it, right? So this differentiating these SWAS from autism is important. Both have presentation of language regression, although autism is earlier, generally around 18 to 24 months, you see the regression. And it's established by 3 years of age. Autism will have those stereotypies, repetitive idiosyncratic activities, echolalia of repeating the things, what you are saying, and neologisms. And the child with autism may have a seizure, but peculiar EEG pattern of sleep potentiation is not seen in autism. It is seen in this DE-SWAS, which was earlier called ESAS. So we have to understand that there is a generally the autistic regression occurs between 18 to 24 months, as I mentioned to you, and then others it may be later also. So if you have a late language regression, it's more likely that it's going to be a DAEs first and need a sleep and awake EEG to diagnose, and the treatment is immunotherapy actually. So that's why it's important. Now coming to autism per se, there are a lot of EEG abnormalities which may be there only in autism. In autism, just like that, without epilepsy, right? So they have epileptiform EEG may be there. They may be slowing or asymmetry, and epileptiform discharges consisting of spike and wave discharges, sharp slow waves, generalized spike waves, and general polyspikes was significantly more common in the individuals who had intellectual impairment in addition and who had clinical seizures. But they were also present in those who did not have any epilepsy also. So there are 4 categories we broadly see. One is isolated epileptiform discharges when you have no seizure and you having discharges. They may be found in up to 61% in sleep. So that is something we have to keep in mind in the first one. And the other one is subclinical electroencephalopic abnormalities, or both non-epileptiform abnormalities like asymmetry, slowing, bioelectric immaturity, and epileptiform ones like spikes, sharp waves, spike and waves, and polyspikes, and can be seen in 4 to 88.6%, even in the absence of a clinical history of epileptic seizure. So these isolated ones have complete absence of any seizures. In the second one, you may have both epileptiform and epileptiform discharges. So you see the percentage is so high. Third is if you have paroxysmal slowing and interictal epileptiform discharges are which have been shown to be more in autism children. So that is also in normal population is only 1 to 4%, and they are seen in 30% in autism. And the fourth last thing is interictal spike discharges, which is used to denote the occurrence of spike discharges occurring between seizures. It has been it has been seen to be seen in autism patient, and it has long been known that these discharges can interfere with normal neural processing. That's why there is a link between autism and epilepsy, which is caused what is something which has to be kept in mind. So trying to understand the electrophysiological biomarker for ASD, the isolated epileptiform discharges have shown to have more autistic features, lower social functioning, and more severe behavioral problems. And after 2 years of follow-up, 20% developed epilepsy. So that is something which has to be kept. And 3% of those who were in the non-IED are going to ultimately become seizures. So 3% who did not have this and 20% who had this ultimately will develop epilepsy after 2 years. A second one was the subclinical electroencephalic abnormalities which were there. So in that they have shown that those who they had more impaired adaptive functioning compared to those who had normal EEG in this situation. So they have seen that expressive language was, which they found to be more impaired in those who had these who had this subclinical electroencephalic abnormalities. So there are many studies which have shown some EEG abnormalities from 39% to also they have found some EEG abnormalities, some frontal lobe problems, some differences in hyperventilations, incidence of burst suppression was higher in them. When they did with propofol, after propofol you have a burst suppression. They gave a lower dose of propofol for them because the propofol was acting in a different way in autistic children than neurotypicals. So this one, then coming to sleep EEG. In sleep EEG, they have shown that there is decreased activity of sleep spindle, and the sleep spindles the density was reduced. Coming to quantitative EEG, this is they have a cube profile. You see if a healthy one is having, the delta and beta gamma are less and then alpha is maximum. But here it's an U which is there in the alpha, you have this middle densities are lowest and the side densities are highest. So it has a U-shaped profile on the quantitative EEG. So now there is an inadequate evidence to recommend doing EEG in all of them. We don't do EEG in all patients with autism. It is only done if there is a suspicion of epilepsy or based on certain history of regression, staring spells, or any family history. And especially if late regression, most of them have 18 to 24 months autism. These children may have a later on language regression also. You need to get the EEG done. So these are the various guidelines from AN, NICE guidelines. Now coming to this, in the last 10 decades, all these EEG abnormalities, we had done this literature review and we found 220 articles, and ultimately we zeroed down to 34, and what I gave you the information was based on these 34 articles. To summarize this, stagnation, regression, fluctuations, developmental dissociation, acquired sleep problems, and sleep specific EEG patterns such as sharp focal waves and continuous spike wave during sleep suggest have direct contributory role of these discharges for autism, especially the one which I am talking about, the continuous spike wave during sleep, which is now called DSWAS. So that is something important. So that has got autistic features. So epilepsy leading to autistic features is there. And mere presence of an abnormal EEG is not itself an indication for an anti-seizure medication, because the EEG abnormalities are probably most often simply a marker of the underlying brain disorder which is there, which is causing autism as well. The third objective, neurobiology, genetics, risk factors, both autism and epilepsy, bidirectional. Fine. So there is a co-occurrence of autism and epilepsy. There are impairments at the synapse level, the mTOR pathways, the GABA receptor function, and the glutamate NMDA level of synapse. Malformations which will involve neurogenesis and neural migration defects, cellular disorganization, heterotopias, and dysplasia. An altered ratio of excitation and inhibition. Genetic, molecular, anatomic pathways may overlap, and there is neuroinflammation in both the conditions. So you may have something which is causing epilepsy and causing cognition issues and social and non-social issues. So you have, we have specific common biological mechanisms. You have this for something affecting the synaptic structure. There are genes which are there, something which are going to be there for the transcription regulation, something for cellular growth, something for synaptic, all these channels. So different genes have been identified for the same. So various associated conditions have been identified, like Down syndrome, like you have Fragile X, tuberous sclerosis, P10 associated CDKL5 to name. So you have all these associations of autism with epilepsy, syndromic have been there. There are certain genes identified, there are certain syndromes identified with an overlap of epilepsy and autism. So basically you have causal mechanisms which can be there. You can have certain which are having, if you are seeing in this, you have genes which are coding for the ion channels or for the transcription regulation or the mTOR pathway, or the genes involved in synaptic function and structure may cause synaptic dysfunction and will lead to an imbalance between excitation and inhibition, may cause abnormal plasticity and excessive cellular growth and synaptic growth. And you can see the genes which have been associated and studied. Coming to the neural pathway level, thalamus is an important gating sensory input to the cortex. So thalamocortical networks are abnormal in ASD, and abnormalities are characterized by marked prefrontal sensory motor and temporal hyperconnectivity within the thalamus. And the same side, thalamocortical circuits are abnormal in the generalized epilepsy. So there is a transition from tonic firing to a burst firing mode in the thalamic neurons, which cause seizures that rapidly generalize. And thalamocortical function is disruptive in spike and wave discharges. In autism, you may have genetic attributes causing synapse-related genes, microglia, and immune-related genes, functional impairment of brain regions and neuronal circuitry, alteration in GABAergic and glutamic circuits. You may have inflammatory cascades. This is the 20-minute one, right? And then you may have oxidative stress, a lot of problems in oxidative stress leading to seizures and leading to cognitive issues. We also completed, reported in a study, a transmethylation and oxidative defects were shown in ASD pathogenesis. This was a study we published recently. And other thing is epilepsy and brain. Epilepsy and epileptiform discharges, there may be oxidative stress, neuronal loss, maybe inflammatory mediators, excitatory and inhibitory balance, may be an issue. So have an altered neural circuitry, as I mentioned about thalamocortical. You may have functional and anatomical effects on important areas like hippocampus. So all these are affecting the brain because of epilepsy, right? So there are ripples of hyperexcitability in epilepsy, autism, and inattention. There was one study, this study, they have seen that those children of autism who had epilepsy had a twofold risk of having ADHD also as a comorbidity. So all this is interconnected. Individuals with coexisting autism and epilepsy have higher rate of motor atypicalities, more autism severity, more impaired social awareness and communication, higher CAR scores, low adaptive functioning and greater hyperactivity, and more challenging behaviors. And this is a recent paper of this year in which both the Jeff domains of the autism and developmental encephalopathy associated trioprotein are required for a proper migration of the GABAergic interneurons. So that is why it may be there causing both the things together. So they are, they in 117 children with ASD and epilepsy, 88 had gone genetic testing done, out of which 47, that means 53.4%, they identified pathogenic or likely pathogenic. 39 had single gene mutations, 8 had copy number variations, and SCN1A and MECP2 were the commonest mutations in this paper. We have also been doing studies and trying to find out, and we get many syndromic ones also, like Fragile X Angelman, Rett, tuberous sclerosis, NF1. And we are, this is ongoing study as a part of a center of excellence, and we have this preliminary data of 82 children from North India, in which we have MFRP gene was the most frequently implicated, and many other genes have been identified. You may have certain metabolic disorders which associated with autism and epilepsy, of like creatine metabolism. You may have phenylketonuria. You may have some channelopathies and neurotransmitter defects causing both autism and epilepsy. Have to be kept in mind because of the GABAergic disruption and SNC1A and 2A also. There are environmental factors. Yes, you do have exposure to air pollution, exposure to pesticides in our food, exposure has been shown to be associated with autism, along with maternal drug intakes of SSRI inhibitors. So their association of epilepsy in these cases are under investigation. Coming to MRI, abnormal gray-white matter volumes, aberrant brain growth trajectories, elevated rates of focal cortical dysplasia, heterotropia, increased dendritic spine density, pruning defect, all have been shown to be associated with autism and epilepsy. Then you also have mTOR pathway, have been studied very much, which is there, and this has been shown to be associated both with them. There is increased density of excitatory synapses in the brain of children with ASD, with a putative link to the aberrant mTOR dependent synaptic pruning. And fMRI has shown this. They have shown in the TSC2 haplo sufficient mice model also the mTOR dependent increased spine density has been shown with the ASD and corticostrial heparin. Insulin signaling. I have not read about insulin. Insulin like growth factors is associated with Rett. In Rett syndrome, yes, but not I have not seen with not with autism. I've not read it. It may be, but I know that IGF-1 had come as a therapeutic option for Rett. I don't remember reading it, but there's so many, I just don't remember offhand. But this mTOR signaling disruption and associated development has been shown for many things, with so many of these genes and pathways being affected, as Bea mentioned. I'll just be going, and why it's important I'm mentioning. See, all these pathways which are there, you have with the mTOR, TSC1, and all that, you have possible precision medicine which may be there. That is why it was being mentioned. So you have an RCT going on everolimus on neurocognitive symptoms in P10, which has primary endpoints were not met. Although there is an interplay of autism and epilepsy, what is causing what is not clear. So you are having these defects which are there, which are leading it. Now the last thing is treatment option to treat or not to treat these epileptiform discharges. So the subclinical electroencephalography abnormalities, whether it is going to, will these SEAs directly cause cognitive and behavioral impairment in children with ASD? Will treating SCA prevent the onset of seizure later in life? What is the predictive value of the EEG abnormalities in development of later epilepsy? A large percentage of these patients could develop seizure in adolescent and adulthood, but pharmacological treatment of SEAs in order to prevent epilepsy is to date not supported by evidence. So the first step is to ensure that the cause of epilepsy is properly investigated. Ensure that autistic features are not result of ESS. What I mentioned to you, sleep and awake EEG. And gentlemen sleeping, that's okay. Can awake also later on. Once the clinical seizures and ESS is sufficiently ruled out, decision to treat SCA or not will depend on if the IEDs are believed to cause developmental impairment, or drug-based treatment may help, in that situation. That means if you have these IEDs more in a significant proportion of non-REM sleep and an arrest of neurocognitive development has been shown recently, then you may like to give them these treatments. So valproate and lamotrigine has been studied, maximum. Levetiracetam has been given, but I'll get you the next slide. There were some debates about levetiracetam causing hallucinations and mood behavior problem, but the literature says it is safe and effective in most of the studies, and they say that it is pretty safe, no detrimental in this meta-analysis. Cognitive or behavioral defects have been seen. To summarize, there are three distinct scenarios which will come. If you have an autistic regression in a child in a child who has epilepsy, then obviously you'll give anti-seizure medication, right? If then second child is who has an autistic regression after repeated seizures, then also you'll give anti-seizure medication. Third is if you're having epileptic discharges are found during the study when you are doing, then the indication of EEG will depend whether you're having too many discharges in the non-REM sleep or it's causing a neurocognitive impairment. So the literature review which we had done, we had got initially 101 articles, then could prune down to 11, and the summary is valproic acid and derivatives are the most used drugs in treatment of EEG abnormalities. Epileptiform abnormalities are common in autism, and treatment targeted towards EEG abnormality does not translate into improvement of ASD features. And there are, I have 2-3 minutes. Okay, treatment of specific conditions. So you have these potential targets for precision medicine coming up in the near future, just going on. So besides the common ones, now new ones are coming with ASOs and other things. So you have this TS, we all know, SNC2A, you have that PCDH, SNC1A, MECP2, but you know, sulfurin is still debatable, omega-3, N-acetylcysteine, all debatable, metformin, oxytocin, not any conclusive thing. Pre and post biotics also having an issue. I mean, not completely conclusive. We have also done study, and cannabidiol has possibly the dermal patch in the Australian study is showing benefit. So possibly an FDA approval is going to come soon. You have this erbecloflen, which is going to be affecting through the GABA B receptors. It's going to be possibly beneficial. Till now, this trial has not achieved this outcome. Bumetanide, it's a loop diuretic. Very important. These days, we are encouraging repurposing of drugs. So this loop diuretic, it is causing the inhibition, creating a new molecule takes a lot of time and energy and money, lots of years. So if you can repurpose it, so this inhibits the sodium potassium chloride co-transporters NKCC1 and NKCC2, and it has shown to reduce the exaggerated amygdala activation to eye contact in children with ASD. Then this last thing is gene therapies come up in Angelman syndrome. You know, there's a maternal copy of this gene is mutated. So ASO is utilizing to activate the paternal one. There's a control trial going on. But you should not miss something like steroid responsive ESES, which I was telling you, and TSC would be there. I will leave the case vignettes, forget it. I mean, this was the case vignette of ESES, which I have already showed you, and I'll just wind up. We are doing transcranial magnetic stimulation for ESES, and we're also doing transcranial magnetic stimulation for autism also. Gut microbiome is very important and has to be investigated. And the gut-brain axis and now gut-brain endocrine immune axis, it goes both ways and can be causing autism and epilepsy both. GFCSF study trial, I have just finished an RCT with a subset of them having fMRI before and after, and we have definitely shown improved functional connectivity after that. And similarly, we had an RCT on oral probiotics, which we just completing analysis. We have seen the quantitative EEG and heavy metal effect and seen them with children with autism with epilepsy and those without epilepsy. And we found some subtle changes and biomarkers on quantitative EEG, yes, and on fMRI. Just to mention that, I will not give, I'm just passing the case vignettes to come to the conclusion. So to conclude is epilepsy is a common comorbidity in autism. Epilepsy prevalence is significantly higher in ASD and vice versa as compared to general population. The median overall period prevalence of autism in children, people with epilepsy is around 9%, including syndromic and developmental delay. But if you exclude them, it's 8.1%. These two entities share a common genetic pathways and understanding of shared mechanisms with Herald Neville treatment modalities. Intellectual disability is a major risk factor for epilepsy in autism. Although EEG is not routinely used in all children with autism, certain clue like history of regression, recent regression, late regression beyond, beyond 2 years. This morphology, staring spells, family history should prompt additional evaluation. Mere presence of an abnormal epileptiform EEG is not itself as an indication for ASM and ASD because the EEG abnormalities are probably most often simply a marker of the underlying brain disorder responsible for autism. Epileptiform discharges are common in ASD and treatment targeted towards EEG abnormalities does not translate into improvement of ASD features. Treating discharges on EEG without clinical seizures may not result in improvement of behavior, and those without clinical seizures may not prevent the future risk of epilepsy, but stagnation, regression, fluctuations, developmental dissociation, acquired sleep problems, specific EEG patterns like focal sharp waves and continuous sharp wave during sleep, and also having discharges more time in the non-REM and having a cognitive decline Personalized approach required to rule out the de-swas is important. Always look out for mimics. And one thing, neurodiversity and humanism and autism has to be there. And something important, this is our collaboration with THF and providing various development services for them. And you need to celebrate their diverse. And this was the stool I was telling you yesterday. There was one stool additional given for a child with. So talking about equity, this is my team. This is the AIMS, and this is AIMS during Foundation Day, AIMS during Autism Awareness Month. And I welcome you all to the 17th Asian Oceanian Child Neurology Congress next year in Yashobhoomi, Delhi. And I am looking forward. I'll be writing to IAN for collaboration, because I feel, as I mentioned yesterday, for those who were there, need to have collaboration with clinicians and basic scientists. Then only we'll have something of a translational value ultimately. Otherwise, you'll keep having nature and science publications, and we will keep thinking that you keep getting those nature and science publications, but will never do anything in the life of these children. Thank you so much.
Very nice talk, Sefali. So you have mentioned that a valproic acid is one of the therapeutic drugs.
It's the commonest. I actually did a search for 10 years to get that evidence, right? So in that, yes, valproate acid has been most commonly used, and lately now even levetiracetam is being used. But levetiracetam has this, some people have a belief that it causes behavior issues. So that's why I had put that slide and tried to review on the levetiracetam. So, but the maximum use has been valproic acid. Yes.
So can it be given to any child with autism, or it has to be given to the specific set of children with a certain mutation?
No. They were indications for anti-seizure medications, was not based on any particular precision-based thing. Except then when you're talking about SCN1A, then you would like to give something like phenytoin, which you're not giving. But this valproate is not precision. I was just talking about general. Because generally, valproate is the first choice for all kinds of seizures in childhood, but we avoid in adolescent girls for the risk of polycystic ovarian disease, and later on if they will go on having pregnancies, so teratogenic effects. So that.
Excuse me. So the images that you had up in the slide, where you were showing the repetitive motion,
the video.
Yeah, the video. Sorry.
Yeah.
So which had hand flapping and all those.
So, that is a stereotypy. So stereotypy. Yeah, these are abnormal. They're repetitive.
Yeah.
Purposeless movements in children with autism. Yeah.
So did you show that in relevance with with epilepsy, like most of them?
No, that's what I was saying. It is misdiagnosed in epilepsy, so they do not need to be treated. They are not epileptic. I mean, so that's why I put the second thing, objective is a challenge to identify epilepsy in autism, because many people mislabel it as seizures, but they are not seizures. Yeah. So they don't have risk of epilepsy.
How much do you think would that be because of anxiety, or is it an OCD?
No, ma'am, 1 second. That child is having it repetitively, irrespective of the surroundings. It's not that he's come to me and started doing that. Yeah, it can be aggravated in any anxiety situation, definitely. But these are not that he's doing this in front of us. He's doing that at home as well. So these repetitive behaviors which are there, they are not in one surrounding, although they may have a trigger because of something. They may have a trigger
because of ongoing thought process or something. Was there any fMRI study that was done during those kind of repetitive moments where you could see a correlation or an effect on a thought process area or something like that?
I haven't come across it. It can be done. Let me tell you, getting an fMRI done in these children, I've been doing it for my GFCSF trial and probiotics. It's really difficult. For those you need very cooperative, and that's why it's been done in subset. Yeah, what you were saying sounds good. Maybe we can try it here. We're starting something with Saurabh. We'll be doing MEG and fMRI. So maybe we can add another subset of those. Actually, we do have in our project meet or stereotypies. We would be doing as a subset. So that is there.
Thank you.
Thank you.
You very briefly mentioned about utilizing TMS as a means of treating autism, or
It
uh,
was not a part of this thing.
right? You very
We
briefly
finished in
mentioned,
RCT and yeah.
right. So I was wondering, are you targeting any specific areas, or are you just probing for areas where you are going to see an
No,
effect?
we made a protocol. We made a theta burst stimulation protocol. So that is there, and that's based on literature, which I just mentioned. It was not there. So I can share with you if you're interested. But we developed a protocol based on literature because literature was not unified on that. But we'll be taking, Saurabh will be involved. We're taking it further, larger study. But now we have started offering patients beyond the research mode because in the RCT we did find benefit in many things because it was a RCT and it was a RCT with the um, sham control. So like the person administering and the person assessing, it was all double blind in that sense that it was sham control. So we have the TMS machine, we have the one which has the sham also, sham coil. So the person will not know whether the person has actually got the TMS or it's a sham.
Okay, thank you.
I have one question. One point is that do you think that the delivery failing or the conversion disorder, which is not directly tied to autism, actually for high functioning, is it possible for them?
And for what?
Conversion disorder, not epilepsy directly.
Oh yes, I'm handling one patient in the ward. I was trying to discharge. He's not getting discharged because there is a possibility. Any person, and with epilepsy.
In conversion inside must be there.
No, ma'am. But that will be what I'm saying is then conversion disorders they will be having a secondary gain. So obviously that gain, and I'll tell you, if these children are not speaking, it doesn't mean they don't have anything to speak, and they are more complex, and they are finding us weird. Actually, we are finding them a bit not in our social milieu, but they are finding us in a erratic behavior. See, Elon Musk doesn't bother about the world, right? They are looking for him to get our astronaut back.
Okay, and my another question is how IT actually increase increase the percentage for which IT. One of my questions is that autism. I know so much participants who have complete intelligence, but as they don't know how to express it, their IT score is low. But they have sorry, IQ score is low.
No, ma'am.
IQ score is low. As what I'm telling you that IQ they have, but they don't know how to utilize it. So how IQ?
I'll tell you. No, ma'am. No, I'll just tell you. This is not my postulate. This is our various studies. So they have shown if somebody who has intellectual disability or those who have CP and intellectual disability, ma'am, reason pata hai kya hai? The same genes, the biological mechanisms which is causing autism and epilepsy, they are the common genes which are causing. And let me tell you, I don't want to complicate, but let me tell you, there is an issue in one of the CP, autism mimics is hearing loss. But there is also an association between autism and hearing impairment because of common genes involved. So in this situation, we are talking about the genetic IDs who are having this problem. So if somebody, and other thing also, because these children they perform poorly on the social and communication domains, right? So when you're calling them high functioning, you call them high functioning when the DQ or IQ is more than 70. Although in normal population you call it, I should not be using term normal. In neurotypicals, you have more than 80 as normal and 70 to 80 is borderline, right? But for autism, more than 70 is high functioning because they will anyway do poorly on the social and communication domains. So those the deficit on those domains, ma'am, is already accounted for when you are calling them high functioning. The high functioning autism is by definition being called as those who are having a DQ or IQ more than 70, which actually more than 70, 70 to 80 is only is borderline, right? And 80 to above is normal IQ or DQ, right? So we are anyway counting ki social communication to poor ho gayi. No, you got it, ma'am. No, I got what you are saying. But I'm saying those, we are talking about those genetic defects which are caused. हाँ वो उसीलिए होगा ना तो वो ID जो होगा वो genetic. What you are raising a question is right, but we are talking about the causative thing. Those who have a genetic. That's why, ma'am, 1 second. It was severe ID. That table was those who autism with severe ID. It was not with normal ID. You got it, ma'am? No, no, those slide, I know, that severe ID will be more syndromic, will be more etiology. So you got my point, yeah. And I got your point, that's right. I mean, everything.
Yeah. So, good afternoon, and I'm thankful to the organizers for adding me as one of the speakers here. Disclaimer, I'm not going to talk to you about ASD. I work on 3 viruses which have some relationship with ASD, and for that matter, all viruses have been said to have some effect of cytokine storm and immune activation during in the maternal system, which can actually lead to autistic-like behavior or autism symptoms. So my lab is working on 3 different viruses which are causing different disorders of brain. Okay, better? No? I know, but when I look at that side, we have to do something about it next time. So 3 different viruses, as you can see over there, are the HIV, which causes a hand, which is HIV-associated neurocognitive disorders. And Zika virus causes microcephaly. And in Dr. Swamia's talk, we did see that one of the risk factors, or there is some relationship between the microcephaly and autism as well. So Zika virus causes microcephaly in mothers who are infected in their first trimester of infection, and all those from the NBRC lot know what I'm going to talk about. But for the other audience, it actually causes the damage to the in the neurodevelopment because it targets the neural stem cells, and the entire process of differentiation and maintaining that stem cell pool is affected by the presence of the Zika virus, which actually causes the placental barrier from mother to the child. So in utero infections of Zika virus actually lead to reasons which would lead to the neurodevelopmental disorder, which is abnormally abnormally small head size of the infant. And then, of course, the SARS-CoV-2 virus, I think I don't need to tell you what it does, but it also causes neurocognitive disorders, and which we're going to talk about a little bit. So for the first virus, which we started working with when I came back from NIH after my postdoc, I started working with neuro-AIDS virus with HIV virus, which causes AIDS. And the reason we wanted to get into the field of neuro-AIDS, because at that time it was very intriguing to know that the virus, which is actually an immune crippling disease virus, also causes damage to the brain. And the reason it causes damage to the brain is, again, because of unknown reasons, at least in those times, of the cellular and molecular mechanisms which were causing damage to the human neurons. Now, the interesting part again there was that the neurons, although are not infected by HIV, but then they were still dying. So we worked out the entire mechanism. And in this entire field, we try to understand, we means the community of a neuro-AIDS researcher, is that how HIV and its viral proteins actually cause damage to the neurons and the consequences thereof. So one of the major reasons for challenges of this HIV field or neuro-AIDS field was that this virus is a human-specific virus, due to which it cannot, you cannot use the animal models. And that necessitated the need for developing a cell culture-based model system of human brain cells, which were of human origin, so that they can be infected or they can be studied in a much more physiologically relevant model for, at least for this disease. That's when we started working and devising this model system, which started in NBRC in 2003. And since then, we have made significant progress in this model system, and we've used this for different viruses, as well as in collaboration for studying other neurodegenerative diseases. Now, the beauty of this model system is it is derived from human systems. It is a neural stem cell model. It can grow as a monolayer, as an adherent culture, as well as in a 3D culture, as you can see over here in spheres. And now, because they are neural stem cells, it also gives us an ability to differentiate different kind of brain cells from them and study the effect of viruses or any agent for that matter, not only at the stem cell level, but also when they're differentiating or when they're differentiated fully on the astrocytes and neurons. So with those ability, we started working on those with those model systems. And as I will try to show you here with the time-lapse microscopy, where you can see that those spheres are actually not just collection of cells, but are clonal division from one neural stem cells. They divide over the period of time, and they're very rapidly dividing because of the properties of neural stem cells. And over the period of 48 to 72 hours, they make these spheres. And that's our favorite exhibit from NBRC, one of the favorite exhibit from our lab also. So we routinely show these neurospheres to the kids who come and the visitors who come. And the next visit is on 16 December. I hope we started making them because it takes 48 to 72 hours. And these spheres is what we show them because they can be seen by the naked eye also. So they're as big. Now, what is the use of this model system? As neurospheres, you can actually see when they're dividing, you can measure the diameter of these spheres, and that will give you an indication that under normal conditions or in the presence or absence of a protein or test agent, whether that is good or bad for the stem cell division. So that is what one we can do. You can also break these spheres and measure the number of cells or count the number of cells, and that will also tell you that how rapidly they're dividing in presence or absence of a protein. So the other important properties of neural stem cells is that they can differentiate, and we have successfully differentiated human neurons out of them, as is shown over here is the TOJ marker, which is an early neuronal marker. This slide actually shows you these neural stem cells, human neural stem cells undergoing the process of neurogenesis. And the green cells are indicating that these are fully differentiated neurons, while the yellow and orange cells are indicating that there's a co-expression of the nestin, which is a neural stem cell marker, and they're undergoing the process of differentiation. If you leave them for 21 days or a little extra, all of them will turn green, telling you that these neural stem cells are indeed making new human neurons. Now, using this model system, we have studied different cellular and molecular mechanisms of HIV virus, where we started with studying, how HIV is affecting the stemness of the neural stem cells, and then the process of neurogenesis. Then when we differentiated astrocytes and neuron from them, we wanted to give them a co-culture, and we use this model system in other ways for various understanding the details, cellular molecular mechanisms of the consequences of HIV virus in brain. Now, we've also used it for Zika virus studies and the SARS-CoV-2. All this work is published except for this one. Actually, the manuscript is lying with me. Archana has given to me last week, so hopefully we'll submit it soon. Now, we've been talking about neuroaids, and we know that HIV virus actually causes, is a immune system disease. Then how come it is affecting the brain, and how does it get into the brain? This is again a teaching slide or an animation which we developed when I was a postdoc at NIH, where we show that how the virus can traffic into the brain. And as you can see over here, the virus gets into the blood vasculature, blood goes to everywhere, and there's a microcapillaries in the brain where you're seeing a cross section of it. You're seeing a human blood-brain barrier over here, which is having endothelial cells. You see the white cells, which are the virus over there. And here is a monocyte which is loaded with these viruses that traffics, that helps the virus to breach the barrier without breaking it, and it gets into the cell, into the brain parenchyma, and gets a CD4 receptor, which is present only mostly in the microglial cells, and that facilitates the entry of the virus into the brain. And once this happens, it multiplies in the microglial cells, give more viral particles, and the viral proteins also start coming out. Now, what you saw with the animation was this part where the monocytes were carrying or trafficking the viruses into the brain. Once they get into the brain, a lot of things happen. Remember, I said in the beginning that the neurons are not infected because they do not have the receptors which will facilitate the infection by HIV. However, The death of the neurons occurs by two ways. The direct mechanisms where the viral protein actually are neurotoxic, so they are shed off from the virus for their own replication and other reasons. And those are neurotoxic, and we know them very clearly because if you take those recombinant proteins, you throw them in the human neuron cultures, and they die by the process of apoptosis at 24, 48, 72 hours. And if you wash those off, actually you can prevent the death. So that clearly shows that how that is occurring. So we worked out that as well as the indirect pathway, which was more interesting because if the neurons are not infected, how are they dying? Now here is the key factor, which is the astrocytes, which are the most abundant cell type in the brain. Astrocytes support the neuronal function in many, many ways. And they're just not there as a packing material, but they provide trophic factors. They clear up the waste, which is the excess of the neurotransmitter glutamate. If you leave the excess glutamate over there, that causes excitotoxicity. So all that housekeeping function is provided by the astrocytes, but astrocytes also harbor this HIV virus. Once the HIV virus gets into the astrocytes, it slows down the process of their protection or the goods which astrocytes were providing to the neurons, and that leads to an indirect cell death mechanism. So we worked out that as well. And in subsequent slide, I'm just going to walk you through how we did that without showing too much of data. So the first question when we came back and started the lab in India, the major question was why there is so low incidence of neurocognitive disorders in HIV as compared to the Western population. Although India was at that time, even one time it was #1 in the world, which was then downgraded to #2 in the world with AIDS patients, but we were not seeing that much of dementia related, which was caused by the HIV. And the question at that time was, despite the huge amount of population which we have, why we are not seeing that? So we worked on that, and we figured out that in fact the clade which was there prevalent in India, even today HIV-C is the most prevalent strain in India. At that time it was 98%, which was infecting the Indian population. And we've zeroed down to one of the mutation which is there in the TAP protein, which is of the viral protein. If the dicysteine mutation is, the motif is missing, and if there's a natural mutation at the 30-31 position of the TAT protein, we figured out that that is the reason why, despite having so many AIDS patients, the cognitive decline in Indian patients was low. And then we kept on moving forward and trying to figure out different mechanisms which the virus is affecting different part of the brain cells. And we zeroed down the TAT protein because many people were working on GP120, which was easier to work on. And we went for not for the low hanging fruits, but for a difficult protein. And we studied all the ways by which the TAT protein can actually cause neurodegeneration in these neuro-AIDS patients. So the journey continued, and we looked at how HIV TAT was actually causing damage to the neural stem cells, and we worked out that indeed what happens is the cyclin D1, this the cell cycle mechanisms which we're talking about with Vidya lecture. That's that cell cycle step is actually affected by the presence of the TAT protein. If it is there, the cyclin D1 goes down. It brings down the proliferation of these progenitor cells, which was causing the damage. Then Meher joined the lab, and she continued to understand how the proliferation was affected by the TAT protein, because she zeroed on to the TRIM32, which is stem cell fate determinant. And she found that the microRNA 155 was indeed the reason by which the TRIM32 was being affected. And the neural stem cells were not dividing because the cellular localization of TRIM32, which was supposed to be in the cytoplasm, was moving in the presence of TAT into the neuron in the nucleus of these neural stem cells, which was bringing down the proliferation ability of those cells, right? Now then, because we all know that the neurons are not there in isolation and neither astrocytes are there, so they're all together. So we started the co-culture business. And as I said, astrocytes provide all these important functions in the brain for in the physiological condition. So we established a co-culture system where we had a fewer number of neurons and more number of astrocytes. Our brains are designed for every one neuron. There are 50 to 100 astrocytes. So we co-cultured them, and again, remember these are primary cells. They were not immortalized, and that is why they are more physiologically relevant. and challenging as well. So you can see over here one neuron trying to make its connection to the neighboring astrocytes in culture, and lot of things happen at these connections. Okay, so in the presence of TAT, as you can see over here, these neurons which were extending their processes to the astrocytes actually retract them. And if I wash the TAT off from the media before the neurons die, you can actually prevent the damage to the neurons. So this is the co-culture work which we did. Meher continued to work on the co-culture, and she established another important step, that how neurons are dying because of the glial culture. And that was because the astrocytes which were friends of the neurons actually turned foe. And the reason for that is because they start producing a lot of ATP molecules which are released from the voltage dependent and channel 1. And that was governed by microRNA 320A. So HIV TAT was increasing, it was blocking 320A, which was blocking VDAC, and then you can actually have prevent the damage to the human neurons via the glial cells. And as the new students joined, the lab elaborated our level of complexity of experiments. And when Manju joined from NIMHANS, as she was a MPhil in from biophysics department, and she said, "Sir, why don't we measure the calcium and a lot of things in the live cultures?" And I said, I don't have any clue how to do that, but we struggled together. And finally, she established this beautiful system where we could actually zero down on another purinergic receptor, which was important for releasing those burst of ATP which was coming from the glial cells. And she figured out that if you, in the presence of TAT, the purinergic receptor P2X7 goes up, and which actually is ultimately resulting into the damage to the neurons. So by various ways, we had shown that how glial cells were actually contributing to the damage to the neurons, which was new to the field because we always believed that the astrocytes were good for the neurons. But nevertheless, we continued, and then Chitra added to our further understanding using the same co-culture system. And she showed that, in fact, the ephrin A3, E4, an important ligand binding and the crosstalk between the astrocytes and neurons is also affected by TAT. And then she showed that the glutamate excitotoxicity is occurring because of the balance in this and the downregulation of the glutamate transporters on the astrocytes, which is contributing to the neuronal damage. Now, very quickly, I'm not going to talk about the Zika virus today, but move to the our unpublished work on COVID, where we know that the COVID cases, in COVID cases, we are all experiencing there's something called as post-COVID sequelae, or the brain fog, as loosely called. It is merely, you have to focus more, you have lack of concentration, you have some kind of fogging. That is what is a loosely used term. And that's very prevalent because more than 30 to 40% of long COVID patients have this problem. And very early on, we had written a review when the labs were closed, and we tried to gather the attention of the field that by giving an attractive title saying that SARS-CoV is more than a respiratory virus. If you look at the timeline, this was in July 2020. And if you remember, the lockdowns happened in March, labs were closed. We submitted this review in April, but it took 3 months because nobody believed that the SARS can actually cause also brain damage. And at that time, we had proposed this mechanism for the simple reason because CDC and the WHO was constantly adding factors and symptoms to the list of the COVID-19 patients, which was indicating that there's a clear involvement of the brain. Something like loss of smell, sense of smell and taste, increased hemorrhages, and all these things were indicating that the virus is there in the brain. Now the question again was at that time, how a respiratory virus, what a respiratory virus has to do with the brain. And we proposed at that time in the same review that there are 3 major ways by which it can actually get into the brain. It being a droplet infection and the loss of sense of smell was going on, it clearly indicated that there was some path from the olfactory neurons. And we have proposed that the virus is getting from the olfactory neurons and is getting into the brain. The other mechanism was obviously breaching of the barrier, because if there was huge cytokine storm, and if you remember, we were all measuring this CRP, IL-6, and all the cytokines in the blood, right? So if there's huge amount of cytokines and chemokine released, they are known to open the blood-brain barrier, and there's a free access of inflammatory cells as well as the virus into the brain. And then while we were writing the review and finalizing it, WHO added diarrhea into the symptoms of the COVID-19. Remember? So there was again very clearly indication there was uh, eradication, or there was a damage to the gut membrane, and then there's a gut brain axis which was also perhaps helping in the retrograde transfer of the virus. So we had proposed all these mechanisms which were actually supported by studies which came later from the postmortem samples, and unfortunately India lagged behind collecting the brain samples in that time. I was trying to push and convince, and I cannot push, but I was trying to convince ICMR that we should also collect samples, and NIMHANS was ready to do the brain biopsies. Dr. Anita Mahadevan was ready to do the autopsies of the COVID-19, but the government didn't allow us. But nevertheless, there were reports which were pouring in, which were clear indications were there that the postmortem samples were from the postmortem samples that the SARS-CoV-2 was in different parts of the brain, and as is indicated over here. So that supported our mechanism. Then the major review came, which turned the field from Solomon's group in UK. That's 20 minutes, right? Okay, I'll finish. From Nature Review Neurology, where he proposed three same mechanisms. One is the cytokine storm, retrograde mechanisms, through bleeds, which were again reported from the Iowa Brain Bank. They had found that there were microbleeds happening, which was also resulting in inflammation in the brain. So all that collectively was contributing to the damage to the neurons. Perhaps that is the reason why brain fog is occurring. But what is the mechanisms of cell death? The neuron can die by 4 different mechanisms, or any cell can die by 4 different mechanisms. And what component of the SARS-CoV-2 was actually CoV2 was causing the damage to the neuron? To build on the case, and when we were writing grant, we wanted more and more review on human studies. This Lancet psychiatry study in 2021 helped, which involved more than 200,000 patients, where they clearly showed that what we were saying in reports and the hypothesis, this was actually proven to be right when they did, and they found that up to 33%, which actually was higher in people who were spending more time in the hospitals or ICUs, that they were undergoing a lot of neurological cases in that. This was further supported in 2023 again, that indeed COVID-19 is causing damage to the brain and leading to long-term sequelae in these COVID-19 survivors. So the COVID is gone, but the consequences continue, and that is why we are trying to understand. So then we started, Archana took this challenge, and she said that we will try to understand how. She posed 4 different questions, which are also her thesis research objectives, as well as for the grant, where we asked first question, what is the mode of cell death by the SARS-CoV-2? What viral components is causing the damage to the neuron? Is there any viral host protein interactions in neurons? And finally, how do we validate this in the brain sections from the autopsy cases? So the very first thing we did, this required a lot of human neurons, which is not difficult, not only difficult, but very expensive to have the primary cells. So we then relied on SHSY5I cells, which is a human neuroblastoma cell line. You can differentiate them in retinoic acid into neurons and then use them for live dead cell assays. Now, this exercise was done just to narrow down from the 22 proteins which we wanted to look for in the COVID-19 or the SARS-CoV-2 virus, which ones are actually causing the maximum damage, and then we would zero in on those proteins using those components of the virus on primary cultures of human neurons. We started that, looked into all this, shortlisted the plasmids, packaged them with help of Dr. Saurav's lab into lentiviral particles and with our collaborators at Jamia, and then transduced the human neuron and looked into the cell death pathway. So this was the quick wrap up of the experimental protocol. Now then we looked into the expression of cell death markers. We wanted to look at all 4 possible cell death mechanisms for the neuron. So we looked at those markers, and I'm going to just tell you those. First, with a live virus. So we took the full virus and threw them on the human neurons. And first we found that yes, the SARS-CoV-2 nucleocapsid protein was present in the neurons. That gives you a confidence that these neurons are infected by this virus, the live virus. And then we looked at the necroptosis, apoptosis, autophagy, and pyroptosis markers. If you see over here, out of the 4 pathways, only the necroptosis markers were up. Others were more or less not significantly altered, which clearly showed us that it is the neurons are dying because the live virus infection by the necroptosis pathway. Remember, I told you we wanted to know which component of the virus is actually causing the maximum damage. We zeroed down on ORF3, A, 3B, and 6 and found that they are causing the maximum damage. Then we overexpressed ORF6, again packaged them in and threw them in the human neurons and did the live dead cell assay. We found that ORF6 was actually killing the primary neurons as well. We again looked for the markers for necroptosis. We found that out of the 3 ORFs which we used, ORF6 was actually causing the maximum upregulation of phospho-MLKL and phospho-RIP kinase 3, which was statistically significant. And the caspase 3, which is again cleaved caspase 3, which is an apoptotic marker, was not altering. Again, giving us another evidence that the cell death of the neurons was actually because of the necroptosis pathway. Then we again wanted to dig deeper into the mechanisms for that. We looked into the mitochondrial functions. She looked at the MTC1 and the MPTP, which is the mitochondrial permeability transmission pore. And if the spores open at the outer membrane of the mitochondria, it brings down the potential of the mitochondrial membranes, which leads to increase of ROS, increase of calcium uptake. So we looked at all these assays and we found confirmation for those. She also looked at the calcium assay and she found that if you have ORS6, the calcium levels go up, intracellular calcium levels in live cell calcium imaging goes up. If you use an inhibitor of this 4RU360, you can actually, of the MCU, you can actually prevent the uptake of the calcium. Now, all this confirmed our hypothesis and helped us in understanding the mechanism. This is the last slide. We wanted to validate this in the human samples, and thankfully by that time ICMR agreed to our request. NIMHANS collected tissue sections. We could get the sections from there, and what we found was very good, which is not showing up because of too much light over here. But we found that in COVID-19, in the hippocampal region, we did find the same markers for necroptosis going up, which was quantitated over here and is shown. You can see here in the tissue sections of the COVID-19 autopsy tissue. So this was validated from there. We also did a DAB staining and confirmed this, that the phospho-RIP kinase 3 was also up in COVID-19 patients as compared to the normal controls, age-matched normal controls. We are extending this to human brain organoids model, thanks to our previous director, Dr. Kishan Ure, who believed in us, and SAC approved this. We have started, Dr. Ray, you would be happy to know that we have started that with the iPSCs. We have the lab ready, and we are inch closer there. To summarize, again, as I said, the HIV viral proteins cause the glia-mediated neuronal damage. It contributes to the neurocognitive deficits which is seen there in HIV patients. SARS-CoV-2 neuronal damage is the key problem for brain fog. We have worked out the mechanisms, how they're doing. We believe it is mainly necroptotic pathway, and we have looked at the markers. Other markers are not showing up, and it is happening through the mitochondrial dynamics. And all this has been validated. Most of our work with HIV also has been validated, and now the SARS-CoV-2 mechanisms have also been validated in the brain sections from autopsy tissue. All this is possible because the dynamic group I have, Shafaq and Maulik are the latest addition over here. Bindu, Guneet, Krishano Bindu has moved to University of Miami now as a postdoc. Guneet has submitted and successfully defended her thesis last week, and we are doing well. Sorry. And all the tissue for the fetal tissue which we got for neurostem cells was from Civil Hospital, thanks to Dr. Sharma, Dr. Alka Singh, and Neelam Thapar. All the tissues which we do for validation are from NIMHANS Brain Bank, funding from different sources, including NBRC. Thank you.
our next speaker is Dr. Monica.
Thank you. Thank you, NBSC. But I have to hear. I am putting it down. Okay. Actually, autism and intervention, it is written recent trend. For that reason, autism, there are so many talks from morning autism. So I'm skipping some of my slides with that. It is my neurodevelopmental disability as a whole, what I'm telling. Leo Canal, this is the history. I will not go through for that. This is my actually the adjective or characteristics. A, alone, even with others. Unusual play. Twiddle and twirl object. I, for indifferent to other people. S, for strange movements and mannerism. And most have a learning disability. Most have a. And there are so many other characteristics that they isolates themselves from the society. I, that inappropriate social behavior, what I am telling, that don't place abnormal, that is inappropriate social behavior, speech impaired or absent, obsessive routine, rituals. I'm just skipping it, lack of a normal eye contact. All these are already presented by other speakers. What happened? What is autism? I'm skipping it. Some more information that three crucial areas. Okay, this particular has not actually covered from morning. That does very early symptoms by which you can actually say that may have or the probability of autism, does not babble, point, or make meaningful gesture by 1 year of age. Actually, before 3, we can't actually make a diagnosis of autism. This is legal, but the probability should be there. Does not babble, point, or make meaningful gesture by 1 year. Does not speak one word by 16 months. Does not combine 2 words by 2 years. Does not respond to name, and loses language or social skill. Sometimes up to 2.5, there are social skills, but that is the regressive type autism. They loses language, and having any of these 5 leads to parent and professional to have further evaluation. DSM-5, not very important here, but why I am telling that autism spectrum, in particular in DSM-5, with or without accompanying intellectual impairment has already covered, language impairment, associated with a known medical genetic environmental factor, which has not covered, that is with catatonia. Now the point is that here the point is that slow talk, uneven profile of abilities, what I actually asked Shefali ji, that uneven profile of abilities, they may have abilities, high abilities in intellectual intelligence, but they can't actually express it. They don't know how to express it. They have not actually bothered to express it. But IQ, Of course, IQ may be there. Um, I'm coming with that. And gap between intellectual and adaptive abilities, what I'm telling. Motor deficit, self-injurious behavior, anxiety, depressive disorder, catatonic-like motor disorder. Here it is written that not as a magnitude of catatonic episode, like that when catatonia is under schizophrenia, now it is separated through DSM-5. Catatonic-like motor disorder for autism. And it is very much important, last two, anxiety depressive disorder and catatonic. For that reason, I'm telling that I will tell only the recent research on that. These two actually till date, I'm not telling till date, already I have actually published something, but have a very poor research, very faint research on these last two symptoms of autism. But these are very important for their overall growth. Coming, and this is very common thing, social impairment, communication impairment, and restrictive interest, repetitive behavior. I think this repetitive behavior is actually the flexibility impairment. Why they are repeating? They are not flexible. So I want to write it as flexibility impairment. And with that flexibility impairment, my new research is coming. Some special categories: aloof child, passive child, active but odd child. Aloof, withdrawn and indifferent to other people. Passive, will not make spontaneous approaches to other people but will accept contact. And active but odd, will approach other children spontaneously, but this will often be odd or in inappropriate manner. These are special categories. It is not actually within that particular high functioning or low functioning. I'm telling these are the special categories. Differential diagnosis, I will not actually go for that. Differential diagnosis, I'm just skipping it because the time is not here. Now the recent trend in identification of feature. Recent trend, I'm telling 2 points. One is the identification, another is treatment as far possible. First point is impairment in sensory function. It has covered, but very important point is that it is dysfunction. Another point is disintegration. Dysfunction may be high threshold, low threshold. That is when there are high sound, they are not responding. When there are very low sound, they are responding. That is dysfunction. Disintegration means that is they can't through one sense when we are actually making another sense. That is, say for example, you are in a rail gate and someone, a train is coming, whistle is on, and someone actually with that rail gate passing the rail line. How? With auditory input, they think they can visualize how far the train is, and they can assume that with their capacity, with their motor capacity, that they can run, they can cover the rail line. But one can assume where the how far the train is, but can't, because in any point there are three sensation where it is integration. In any point that integration is less, accident occur. In any point, visual through visual, auditory through auditory to motor, 3 points. So there is a high disintegration for the person with autism. And then impairment in theory of mind, impairment in executive functioning, impairment in central coherence. Theory, I mean, I'm coming with these 3: impairment in theory of mind, executive functioning. Actually, the flexibility impairment, what I am telling, that I actually make it as flexibility impairment. Theory of mind, theory of mind, I am coming next. Identification of some clinical features that I'm telling at first. That clinical features are very important, catatonic features. It is not actually like catatonic episode, but catatonic features are there which may actually decrease their ability which they have in both cases, particularly physiological, physical or physiological activities, etc. All things by which their behavior and their all their attitude and expression decrease. It is in negative senses, and these items may related to catatonic feature and that observe features within the, and till date again I'm telling that catatonic features have practically no particular. It is very much important to assess the catatonic feature of the person with autism, which is very much needed for their intervention, for that intervention, for their improvement, but there is less in India and within world, that is catatonic. There are catatonic scale, but not specified for autism. I'm coming with that. And another one is mood swing, anxiety, depression, both. So bipolar disorder is a comorbidity of autism for adult. So when we can, if we can assess from not from childhood, but from adolescent to adulthood, it will be from adolescent, not yeah, from adolescent. It will be a great help for their intervention if we can assess properly bipolar. Now the first point is the sensory integration, that is neurobiological process, neurological. Actually, what I am telling, both cases, it is a neurobiological process, and disintegration and dysfunction, both are there. And praxis, it is a neurological process. There is cognition directs motor action. What I am telling, that motor action, when you are actually crossing the rail line, that is cognition, and it directs motor action if it is properly. So that is praxis, which is actually the motor planning that when intermediary bridges ideation and motor execution, which is less in autism, and sometimes actually even zero in autism. And executive functioning, again, that is, there is a dysfunction in executive function. What is that executive functioning? It is actually not directly, it is related to the IQ level. It is of course cognitive abilities, but control function of brain. Whatever the IQ level, intelligence, level of intelligence, now they are telling emotional intelligence, not directly IQ, etc., whatever, but the controlling of it is executive functioning, which is very less for the person with autism. A higher control process necessary to guide behavior in a constantly changing environment, that is the abilities of planning, working memory, mental flexibility. What I am telling again and again, that flexibility is very, very important for that. And response inhibition. What is that response inhibition? When I want to just to run from here, but I have to inhibit myself according to the social demand. Really, I'm not. That is that is only the example. But actually, response inhibition, impulse control, that impulse control is actually with response inhibition. If you can control your impulse properly, response inhibition will be. And then monitoring of actions of own and others. Own is not written here somewhere. Okay, all these are actually for autism, these are less. There is a deficiency in executive functioning. Next one is the ability to focus on both detail as well as whole, that is central coherence theory, which is very less in autism. And practically weak central coherence means focus on detail rather than whole. The inability to disengage from the obvious. I'm giving you one example, my direct experience in zoo. I have actually taken some person with autism, children and adolescent with autism, And at the cage of the tiger, it is rare seen that tiger is actually roaming in front of the net. Rare seen. Many times tigers sit alone in a corner of the cage like that. All are actually laughing, all are shouting. Particularly the person with autism, which I'm telling, his all attention is on the net. He is just playing with the net, the circle, etc. What is the design of net? With that obvious stimulus, and very important stimulus, obvious stimulus too. It is not actually very common to him also, but he can't actually take a particular in disengaging from that stimulus, that short stimulus. He can't detail, he can't take that whole. That is a very important ED difference in autism. So executive dysfunction, that view vagus, that ED underlies many of the key features of ASD and caused by difficulties. This caused by difficulties initiating new routine, non-routine, tendency to stuck in task. That what I am telling, flexibility disorder. That within that three, executive functioning is less. They can't disengage from one to another. They can shift from one to another. That is actually a high range of dysfunctioning of their executive functioning. And strong liking for repetitive behavior, that you know, it is common. The next point is very common. And now some special clinical features of mood swing for autism. These are features I am telling. I will I think I will complete within time. Mood swing, that is mood fluctuation, sometimes from external world. I am telling that there is no obvious cause is that. Sometimes they are aggressive, sometimes they are very, but of course cause behind within themselves we can't read. That is of course, but mood swing or expression that actually create that tantrums, violence, aggression, and it get worse if you can't actually properly control it. And these are identify which emotion they are feeling. That is difficult. For which emotion they are particular, they went on that. That is important. And impact is conditions may have negative effect on their processing, integrating, and organizing of information that have significant impacts on their communication, social interaction, functional skill, and educational performance. That particular mood. And all these symptoms may not necessarily indicate actually bipolar disorder, but rapid swing may actually after adulthood may create that bipolar disorder in a clinical, as a clinical case, if you can't actually control it in a proper time. Need of an assessment tool. However, no child or teen should be started in medication or any therapeutic intervention. So assessment tool is of course needed. And there is an assessment tool, what I'm telling, under new research, that 14-item scale was developed already. It was published, mood swing scale for the particularly for autism. And according to CURS 2, mild, moderate, all things are there. 126 individual with autism, and 14-item scale was developed, and with reliability, validity, all things are there. Then catatonia in ASD. Here, the extreme form of catatonia for ASD, absence of speech, that is mutism, absence of movement, that is akinesia, and maintenance of imposed posture. Same thing this type, that is catalepsy. These three behaviors are common for autism. Again, common mean how much you are intervening and how much you can assess properly, and then intervention started. So lesser degree of these impairments, other abnormalities, posture, movement, speech, behavior, motor stereotypy are also considered to be catatonic phenomena. And autistic catatonia is a rare type of disorder that affects roughly 2% of all adults with autism spectrum disorder. Roughly 2%, more or less. Actually, in psychology, the term all should not be used properly. Mostly, I am telling, few exhibit stupor and few severe excitement. So researcher developed a measuring scale for catatonic symptom of the present with ASD, and these are the item full 6-point: negativism, mutism, withdrawal, stupor, echopraxia, flexibility, and stereotypy. Actually, there is no time to really discuss about these, but I'm actually giving that information. If anyone wants, he can contact with me to have this particular two scale, which has already published in reputed journals. These are the result of that. I'm not going to that. These are very common intervention. Lova's technique, applied behavior analysis, already it has already covered. I'm going to the training, some recent trend. Sensory integration and praxis. All these what I'm telling, that is only the assessment tool. Now I'm going to the therapy. 20 minutes. I will. Sensory integration and praxis, executive functioning, theory of mind, how much it would be possible. I'm starting. Sensory integration, there are so many, it is not possible to explain. Brushing technique for what is that treatment? I'm not going to. Motor deficit for praxis. This is practically vestibular sensation, balancing. This is also for balancing. Now the executive functioning. Executive functioning, training materials, again I'm telling in a nutshell, that block span for the flexibility. There are 3 blocks, and we are telling that particular person when this is a particular design, that yellow is here, green, and then when it is just intermingled and ask that, please do that, what you have already seen. And then with other blocks, and then it is complex. First, starting with 3, that just repeat, and then with disengaging, and then with other blocks, and up to 6 blocks. That here three colors are there, other 3 colors to actually to point out that recognize the previous color, stimulus colors. And again, the next highest part is 3 color square and other triangle, circle, etc. That is a block span, which is working memory, which actually directly start in working memory. Object permanence, I'm skipping it. This is the last point of maze, that is again the flexibility. Here you see, where is it? In here, one block is given, and here one stimulus, and the subject said that you have to reach the stimulus. Subject here that reach the stimulus. Then in the next time, that block is has put here, and then again the subject has to actually reach the stimulus. For autism, there is a high chance to actually go to that and failed and aggressive, anxiety, aggression, whatever. And this one is the simplest one, and that is the training. That with training, that flexibility we are actually arranging. And this one is the highest complex. You see that it is a complex one. Then theory of mind. It is very important point of theory of mind, that is autism, person with autism see others as their own extension. Theory of mind of these two kind, cognition and affection. Cognition, there is a high level of cognitive work throughout the world, Saliani test. I will not go for that. Autism mind blindedness is very crucial in their particular extension. Autistic persons say other as their extension, So a false belief task I will not here. I have actually made one theory of mind affective tool. That affective tool, description of tool. First of all, what is that? It is a video. I can show it, that from from smile to actually crying, that pleasant to unpleasant for two person, and there is a data, and with that data it was actually established reliability with that these two ultimately for autism they can actually recognize, and that recognition in that way in computer, in laptop, here one camera was camera was fixed, and with that who can't say that this is unpleasant, this is pleasant, with their expression it has actually, we are actually making that difference, that with that pleasant, whether they express their pleasantness with that camera. And this is one example of that child, that a little girl is sitting with others. Today is her birthday. She this is also a video. She received a doll as a gift. She is laughing and she is very happy. And then her friend snatches away her doll, and the little girl is very sad and losing her doll, and she is crying. And then the question is, smile and cry. It is in written Bengali, Hashi Kanna. Now, what the child did, whether he is crying or whether he is laughing, and the question is, and then the question, and with that, from child to adult. This is all. There is also another story. I am not going to detail of that. Child to adult, and then the question is, why is the girl in the story happy? That is the theory of mind emotional training, is that. And I can remember one girl after some training session, she said, oh yes, in Bengali there was a serial. She said, oh, serial, I can't remember the name. That serial, say for example, Mou. Why Mou keno কাঁদছিল বুঝেছি. Why Mou cried? Because her mother rebuked him, scolded. That is the cause. That is a great, actually for me, that is a great achievement that he can say. That is the cause behind. And there are so many things in play therapy, another very important, health and sex education, that I have to tell. Till date, for the stigma, that is when I am telling health and sex education or sex education, they thought that I want to train with sex. For that reason, I have to write health and sex education. The training concept is growth and development, modesty, public and private, respect of privacy, and differentiation among friends, common. I'm telling at first the identification, similarity and difference between gender. And I have not actually put any picture because of that recognition, maybe. For that reason, drawing, and not with, even not with artist. Very common drawing with person with autism. And then adult and peer. Then the specific gender specific dresses. This is my module of that. And then human being, identification of same sex, gender, what I am telling, same sex by name, Onkit is the male and Onkita is female, like that. Then male and the different ages, female different ages, from neonate to grandmother. Privacy, that is for your modesty, privacy, that is for female menstruation, for male ejaculation, masturbation. All things are how the individual has to wash on garments, and that particular change of garments should be in washroom or in a proper place, like that. And cleanliness, and during menstruation, during ejaculation, brushing, cutting nails, all things are under cleanliness. Again, I am telling, when particularly brushing, cutting nails, all these I have to put for that simplification, because when I'm telling only menstruation, ejaculation, there are some particularly stigma for that reason. Shaving, flashcard, this type of flashcard we have used with that video and flashcard, how to make that shaving. And then recognition of emotion, here happiness, sadness, fear, within that. These three actually very important for health and sex education, because of that happiness, sadness, your happiness with that particular, if it is ejaculation, if it is whatever, that you have to put in actually with privacy. And social behavior, interaction with stranger, taking care of younger, taking care of older, taking care of same age, and sharing habit. These behaviors could be taught through role playing, that is social behavior. And art therapy. And the last one, the art therapy, I can't actually, dance movement therapy is a very important therapy, but it is not actually possible. Here, three walls are with mirror, and it has a very good dance therapy unit we have, but it is not possible. I'm just my last, this is music therapy. This is the last upcoming research, that is brain type of ASD. According to Baron Cohen, extreme male brain is type S, extreme female brain is type E, and balanced brain is type B. And empathizing systemizing score by which type A, B, C, ACB formulated, that particular score till date is not actually properly in India, in culture-free situation. So, and another point, fetal testosterone, a biological factor which is correlated with this ES score. So, particularly high male brain is actually, according to Baron Cohen, is a characteristics of autism, high male brain. But we know we have to a balanced brain, and there is a recent research emphasizing on the identification and training part of this ES concept, ASD, where it has immense impact on their socio-emotional development. And again, I'm telling, it is empathy, not sympathy, attitude toward. Then humble recommendation of the present author to the society, more to say to the able person of the society, let us stretch our mighty hand to a pressure to the person with disability, not to help them to walk, but to protect their flame of hope from the storm of rejection. And this is my institution, Pradeep Center. Thank you. I've taken 1 minute.
Ma'am, for the elaborate discussion, presenting, summarizing all about autism. So since morning, we've been hearing how autistic individuals, they have social and communication deficits. So how do they interact with non-humans like pets and so on? And how do they watch videos, movies, and on their own choice?
Practically, if you think there is a spectrum from low to high, I'm not telling that all can do their own choice or all can, but when we are in a training procedure, we are creating some environment by which we will able to actually make them attention with that. If it is card, if it is video, according to the choice and according to the skill of that person who can actually directly pay attention in video, we make that video. Who can't, who actually with object only actually flexible, we are giving that. So training concept is not actually for overall. All things, all materials are according to the need of that particular person, and we are trying at best one-to-one for training.
Hello, Taranu.
Is that good? All right. I'll just keep it off till we start. That's this one.
we'll start our next session, neural activity and autism. The first speaker is Klaus Tomaru, from CSF, and we'll be talking about 3.0 gamma target for restoring.
All right, thanks. Yeah, no, I want to thank Saurav and everybody else for inviting me and welcoming me here. You know, I was at this place actually in 2012 when I was visiting India, and it's really nice to come back and hopefully have a chance to interact more over the next day or two. And you know, my family is originally from Ludhiana, but my wife's family is actually from not too far from here, from like a village near Kortputli and Narnal, and she still has lots of family around here. So it's great to be here. Okay, so as was mentioned, I'm going to talk about gamma oscillations, so patterns of neural activity, trying to understand how they relate to behavior and how they might be targeted as a therapy in autism. And I'm going to talk about you know, I'm going to just start off by explaining why I'm so interested in these oscillations in the context of autism. And most of the work I'm going to talk about was done by Kathleen Cho, who was an amazing postdoc in the lab, who now has an independent position in Paris. And at the end, I'm going to talk about some work in SYNGAP mutant mice that was done by Xiu Zhu, who's a current postdoc in the lab. Okay, so just to start with this background, the basic motivation is that in many different mouse models of autism, in mutant mice that have disruptions in some of these high confidence autism genes that we already heard about today, there are abnormalities in parvalbumin expressing interneurons, okay? So this was, I think, first really well established in these models of Dravet syndrome. So this is showing expression of SCN1A, which is the gene which causes Dravet syndrome, and noting that it's mostly expressed on the axon initial segments of parvalbumin neurons. Of course, then there was this well-known study from Bill Catterall's lab, basically showing that in a mouse model of Dravet, there's reductions of NAV 1.1 expression in inhibitory neurons, and this results in a reduction in levels of inhibition. And then they went on to show how that relates to behavioral deficits. In ARID1B heterozygous mice, you also see reductions both in total numbers of GABAergic neurons and specifically in numbers of parvalbumin interneurons. And then this is a study that my lab did along with John Rubinstein's lab looking at catnap2. And here we did a couple of things. First, Dan Voigt in John Rubinstein's lab looked at levels of catnap2 and showed that they're particularly high in parvalbumin expressing interneurons, both chandelier cells and this broader population. And then he did a nice assay where you would knock out catnap2 in specific interneuron progenitors, transplant those progenitors into a normal background, and look at the properties of the resulting cells. And what you see is a reduction in the number of parvalbumin expressing cells, and no change in other interneuron classes, such as somatostatin expressing cells. And then if you look at the properties of interneurons, you see that the spikes become wider, and sort of the ability to generate high frequency spiking is reduced, again, specifically in parvalbumin expressing cells, but not necessarily in other interneuron subclasses, such as those which express somatostatin. So this is just to show that across many models, right, this is a common theme. Now, parvalbumin interneurons obviously do many things. They're responsible for generating the inhibition that enables cortical circuits to kind of function normally. But one of the things they do is they help to generate rhythmic patterns of activity such as gamma oscillations. And I helped to show this in work that I did when I was in the Dijsrath lab as a postdoc now about 15 years ago in the early days of optogenetics. And so gamma oscillations are rhythmic patterns of brain activity. This is kind of a classic example from Wolf Singer's lab. If you're just recording local field potential activity in the visual cortex, then when a visual stimulus is presented, this is in a cat, you see this high frequency activity that accompanies the onset of spiking in this neuron. And if you zoom in, you see that this high frequency activity corresponds to rhythmic fluctuations around 40 cycles per second. This is this classic gamma oscillation. And there's also spiking that in this case is actually locked to the troughs of this oscillation. So a gamma oscillation reflects this rhythmic pattern of neural activity that's synchronized across neurons. And that's why you can see it if you record local field potentials or even if you record EEGs outside of the brain. And gamma oscillations can be seen in many regions of the cortex when those regions of cortex are engaged. So here I gave an example in the visual cortex when a stimulus is presented, but you also see these sort of task-evoked gamma oscillations appear in frontal cortex during a variety of cognitive tasks. And so this has led many people to hypothesize that these oscillations might play an important role in the performance of such tasks. And so if you have dysfunction of parvalbumin expressing interneurons in conditions such as autism, but of course also in other conditions like schizophrenia, this might lead to problems with task-evoked gamma oscillations, these increases in gamma oscillations that you see during cognitive tasks, and that might impair the performance on those cognitive tasks themselves. Okay, now this is a very straightforward and intuitive idea, but it's been very controversial because the function of gamma oscillations has been very controversial for many decades. And this has been a major kind of debate in systems neuroscience. Okay, and the reason is because almost because it's so easy to observe gamma oscillations using EEG, and this has been done for more than 100 years, people have speculated about what these oscillations might do. But most of these ideas are really not backed up by causal evidence, right? They're ideas that people describe, but haven't been able to show actually happen in the nervous system. Furthermore, anytime you have excitatory and inhibitory neurons connected to each other, right? When the excitatory neurons become active, they'll tend to activate the inhibitory neurons, which will then provide feedback inhibition that silences the circuit for some period of time before it wears off and the circuit becomes active again. So anytime you have excitatory and inhibitory neurons together, they tend to produce these kinds of oscillations. And so many people say, look, these oscillations, of course they happen, but they're just a byproduct that just sort of tells you that a circuit is active. They don't have any function or any role in how that circuit processes information. And the third issue is that when you stick your electrode into two places in the brain and you see oscillations that appear to be synchronized, where the peaks and troughs appear to line up in a consistent manner, that doesn't mean that the neurons underlying those electrodes are actually synchronized, right? So electrical signals will propagate passively through the brain, just like they passively propagate through the air and can be picked up by our cell phones or car radios. And because of this, when you see oscillations that appear to be synchronized, it doesn't mean that those brain regions are actually synchronized. It may just be an artifact of sort of volume conduction. And so for all of these reasons, people have really wondered, like, oh, when you see this kind of synchronized neural activity, is it actually meaningful, or is it just sort of a byproduct of the fact that the cortex is active? Okay. And so in the context of conditions such as autism, it's unclear whether or not finding changes in gamma oscillations and just the whole phenomenology of gamma oscillations, whether this is just a marker that tells you how well the brain is working and maybe how active certain types of neurons are, or whether this is actually a key functional mechanism that performs some important information processing role that we should be actively trying to normalize in order to restore circuit function. Okay. So that's the basic dilemma that I'm going to try to address today. And I'm going to tell you how we've tried to do this in mice and what this might mean for therapies. So, one of the hallmarks of autism, as we already heard, is sort of deficits in executive functions, particularly deficits in cognitive flexibility. And these are classically measured using tasks like the Wisconsin card sorting task. Now, many of you might be familiar with this task. You'd get a card with, say, 2 red crosses on it, and you'd be told to sort it into 1 of 4 piles. And you wouldn't be told, like, what type of rule to use. You'd just be told whether you did this correctly or incorrectly. So you might sort based off of color or shape or the number of objects. Pretty quickly, you would infer what the rule is. And then without warning, the rule would change. And performance is measured by sort of how many rule changes you're able to learn. And individuals with autism are classically perseverative on this task. They learn some number of rules, but then they get stuck on those rules and cannot learn new ones. Okay, so it's impossible to perfectly, kind of model a task like this in mice, but we can at least use a mouse task that captures some of these key features, right? Learning to pay attention to one type of cue, realizing when the rule has changed, and then suppressing perseverative behavior in order to update the strategy you're using for making choices. So in this classic bowl digging task, right, mice have to find a hidden food reward, and they're presented with 2 different bowls on each trial, each of which has a different odor and texture cue. And pretty quickly, the mouse will realize that one of these cues, maybe in this case, the smell of garlic, indicates the location of the food reward. And then once the mouse learns that, then without warning, the rule will change. And they'll have to switch from an odor-based rule to a texture-based one, or vice versa. And we call this a rule shift. Okay, so we want to measure what's happening in the brain during these rule shifts. Are perhaps gamma oscillations and parvalbumin interneurons involved? And can we perhaps use that information to rescue behavior in cases where mice are perseverative? Okay, so one of the things we've done in my lab is to use voltage indicators to measure neural activity and to quantify synchrony. And this is done to get around those issues with the passive propagation of electrical signals that I brought up earlier. So in this case, we're using ACE-M-neon, which is a green voltage indicator. We're expressing it in parvalbumin neurons in the left and the right medial prefrontal cortex of mice. And then we can measure fluorescence both from ACE-M-neon as well as fluorescence from a reference fluorophore, in this case TD tomato, as these mice are behaving. Now, the reason we have a reference fluorophore is that unlike, say, calcium indicators, the signal to noise for voltage indicators is very low. And so you can't really tell the difference between changes in fluorescence that are caused by voltage as compared to changes in fluorescence that are caused by just the fiber bending or even hemodynamic artifacts in the brain. Okay, so we're going to acquire all of these signals from both sides of the brain, and then we'll filter them in a frequency band of interest. So if we want to focus on gamma frequency activity, we would filter between, say, 30 and 50 hertz. So we're focusing on activity around 40 cycles per second. Then I won't go into great detail about this, but basically we take these filtered signals and we take 3 of them and use them to predict the other one, and we compare how well we do to how well we would do if we shuffled one of the voltage signals. And this basically tells us how much information one voltage signal carries about another voltage signal within a frequency band of interest. In other words, it tells us how synchronized are those 2 voltage signals. And so this metric, how much better we do with real data as compared to shuffled data, that'll be our metric of synchrony. Again, don't worry too much about the details. The point is we can use these voltage indicators in order to measure synchrony in freely behaving mice. Okay, and this is what we find. So when mice are doing our rule shifting task, and they're learning a rule shift and they make an error, we see this big increase in our metric of synchrony, specifically around 40 hertz. Now, this is really notable because error trials are very, very important in this task, right? Mice are not told when the rule changes. The only way they know the rule changes is that they're following the old rule. They're expecting to get a reward, but then instead of getting that reward, the trial ends in an error. And that should be a signal to the mouse that its old strategy has become outdated and that it needs to update its behavior. And at that moment, we see this increase in gamma synchrony. This does not happen on correct trials, and it doesn't happen in other frequency bands. It only happens around 40 hertz. So we're seeing that indeed there is gamma synchrony. It's not due to volume conduction. It's happening at the right time. But what we'd like to know is it actually necessary for the mice to learn this new rule? And for that, we turn to optogenetics. Now, most of you probably know what optogenetics is, but I know this is a diverse audience. And so I'll just kind of give a quick anecdote. So I see patients, I'm a psychiatrist, and actually I worked in the lab where optogenetics was really popularized in neuroscience, in Carl Dijsrath's lab, when I was a psychiatrist. And I used to see patients with Carl. He's also a psychiatrist. And one of his patients was a young man with high-functioning autism. And he actually came in one day, this was probably back in, this must have been like 2008. And he said, "Oh, Dr. Dijsrath, you know, I was telling some of my friends about your research and showing them YouTube videos and explaining how you use like genetic engineering to take genes from microbes, put them in the brains of mice, and then control their thoughts with lasers." Right? And so that is a perfect description of how optogenetics works, right? Optogenetics obviously takes these genes from algae that are sensitive to light, puts them into neurons in particular regions and particular sets of neurons in order to make those neurons sensitive to light, so we can deliver light flashes to activate or inactivate those neurons. Now, most of the time when people use optogenetics, they deliver a single train of light flashes, and they might deliver it to, say, the same brain region on both sides of the brain. What we realized is we don't have to do that, right? So we could deliver 40 hertz light flashes in order to stimulate gamma frequency activity, but we could deliver those light flashes either in sync across both hemispheres, so we would actually boost the synchrony, Or we could deliver 40 hertz light flashes, but asynchronously, out of phase, misaligned between the two hemispheres, so that we're still boosting gamma rhythms, but those rhythms are no longer synchronized between the two hemispheres. And so we tried this in otherwise normal mice. And what we find is that in mice that have channel rhodopsin, in which we've made parvalbumin interneurons in the prefrontal cortex sensitive to light, when we boost gamma activity but do so out of phase, it takes those mice much longer to learn rule shifts. And if we look at what they're doing, these mice have become very perseverative. They're stuck on the first rule and having trouble learning the next rule. We can try different patterns of light flashes. So we can deliver misaligned flashes at 20 hertz, so below the gamma frequency range, misaligned light flashes again at 40 hertz, or aligned and synchronizing stimulation at 40 hertz. And it's only this misaligned gamma frequency pattern. It's only when we disrupt gamma synchrony that we see this perseveration in mice. If we deliver the same stimulation, but now in phase across the hemispheres, mice actually perform perfectly well, maybe even slightly better than normal mice, okay? And so this was very powerful evidence to us that gamma oscillations are not only recruited, but actually necessary for this type of cognition, okay? Now, we thought about doing the opposite, and what I mean is trying to normalize cognition and whether or not synchrony would have an important role there. And this actually is a project that started before what I just told you about, when we were studying mice with mutations in DLX5 and 6 as part of a collaboration with John Rubinstein's lab. Now, DLX5 and 6 are transcription factors that many of you might know are important for the normal development of GABAergic interneurons and are particularly important for parvalbumin interneuron development. These mice are not necessarily a mouse model of autism or schizophrenia, but they are a model of what happens when parvalbumin interneuron development goes awry, okay? And so in these mice, as they become young adults, they develop abnormal parvalbumin interneuron physiology. So notably, the spikes become wider and their amplitude becomes shorter and sort of the input resistance goes up. Some of these changes are similar to what we had earlier seen in catnap 2 knockout mice. They also develop deficits in rule shifting. So these mice, shown here in blue, is the performance of the mutant. They learn the first rule, the initial association, just fine, just like their wild-type litter mates, but they're very perseverative when it comes to learning a rule shift. And then when we looked at gamma oscillations using voltage indicators in our more recent study, we quantified the difference between gamma synchrony on error trials versus correct trials. In the wild-type litter mates shown in gray, we again see higher synchrony on error trials than correct trials. But in the mutants, this increase in gamma synchrony that normally happens is gone. And so these mice lack gamma synchrony, presumably because of problems with their parvalbumin interneurons, and they're very perseverative. Now when we deliver the same patterns of stimulation to these mice, which are very perseverative normally, we find that only this 40 hertz synchronizing stimulation, only when we sort of boost not only gamma oscillations, but also their synchrony between the two hemispheres, we're able to normalize the rule shifting performance of these mice. They learn just like wild type mice. And what's quite surprising, and what I'll come back to, is that these pro-cognitive effects are actually persistent. So once we deliver this stimulation and we sort of boost the synchrony of these gamma frequency oscillations, one time we can test the mice a day later, a week later, even a month later, and they will continue to perform normally. Okay, so I just want to pause here and note that what I've told you is that gamma synchrony does in fact increase during rule shifts, and disrupting this gamma synchrony will actually make normal mice perseverative. And if we boost this synchrony in mutant mice that are perseverative, it actually makes them behave normally. So this is a really powerful double dissociation that shows conclusively that actually it's just the synchrony of these patterns of stimulation that are important for determining whether or not you have normal cognition and are important for brain function. That's the only thing that differs between the in-phase and out-of-phase stimulation. In both cases, we're stimulating the same cells, we're stimulating them the same amount, we're stimulating them rhythmically, but in one case that rhythmic stimulation is aligned, in the other case is misaligned. And that's a difference between a therapeutic pattern of stimulation and a pattern of stimulation that makes mice inflexible and perseverative. Now, the last question I want to talk about here is where does this synchrony come from and what exactly does it do? Now, we in my lab had been very interested in long-range GABAergic projections. We had started to find about 10 years ago that in the prefrontal cortex, GABAergic interneurons were not just locally projecting, but actually would also send their fibers to distant regions. And we wondered, when the two hemispheres become synchronized, maybe this is due to projections that go from parvalbumin interneurons in one hemisphere to the other hemisphere. And so we confirmed this using a very simple experiment. We now express channel rhodopsin in parvalbumin interneurons in one hemisphere. We then record in the other hemisphere. We can see the terminals of those parvalbumin interneurons actually arborizing in the contralateral hemisphere. And when we record from pyramidal cells and deliver flashes of light, we see these optogenetically evoked synaptic responses that are indeed GABAergic responses. Okay, so now that we know these connections existed, we wondered what would happen if we try to disrupt them, okay? So we do another simple experiment, which is now we express halorhodopsin, which is sort of inhibitory opsin in parvalbumin interneurons in one hemisphere, and we place a light fiber in the other hemisphere to inhibit just these callosally projecting parvalbumin expressing terminals. And when we do that in mice that have halorhodopsin in their terminals, when we deliver light in order to activate halorhodopsin and inhibit those terminals, these mice now take much longer to learn a rule shift and once again become very perseverative. We thought that this might reflect disruptions in gamma synchrony, and so we decided to measure gamma synchrony the same way we did before. We once again express a voltage indicator and a reference fluorophore in parvalbumin interneurons in both hemispheres. We additionally express halorhodopsin in one hemisphere so that we can deliver light in the other hemisphere to inhibit these terminals, these callosal parvalbumin expressing terminals. And in control mice, on both days, with or without light to activate halorhodopsin, so these are mice without halorhodopsin, we see this normal increase in gamma synchrony on error trials. But in the mice that have halorhodopsin, when we inhibit these callosal parvalbumin expressing terminals, we now see that this normal increase in gamma synchrony is lost. Okay, so yes, these callosal PV terminals, they seem to be important for transmitting gamma synchrony. That seems to be necessary for learning this rule shift. The last thing I'm going to say about this is we thought, well, look, we're neuroscientists, and so we think that when animals or any organism learns something new, that reflects changes in patterns of activity in their nervous system. And so here we think that in the prefrontal cortex, there must be patterns of activity that are changing when mice learn these new rules. And maybe what's happening is we're preventing those changes when we disrupt gamma synchrony by inhibiting these callosal PV terminals. And so we use calcium indicators to measure the activity of many neurons at once. We detect these deflections, which represent time points when individual neurons are active. This raster, each row is the activity of a different neuron over time, and these little yellow ticks represent when they're active. And so we can look at patterns of activity, say the pattern of activity following a correct trial or an error trial during the rule shift. Each line here represents when a particular neuron was active. So bright lines represent neurons that are very active, and dark areas represent neurons that were inactive. This represents the whole pattern of activity. And so what we think is that when the mouse makes an error, like patterns of activity in the brain should change because it should learn to change its behavior. And so we can quantify how similar these two vectors, activity vectors are. And what we see is that when we inhibit callosal PV terminals and we suppress gamma synchrony, that these patterns of activity become more similar. That is, they change less. Okay? We can also look at patterns of activity during the initial association versus during the rule shift. And once again, when we inhibit, we see less of a change. So there's less updating of neural activity. Okay, so just to summarize, there are these callosal parvalbumin projections. These are important for gamma synchrony, and they're also important for learning this rule shift and for updating patterns of activity. And if we inhibit these callosal projections, all of these things are disrupted. Okay, so what turns out to be very interesting about this is that all of the effects I told you about seem to be persistent and seem to reflect plasticity in the brain. And what I mean is that if we do the experiment I told you about before, and we express now both halo and channel rhodopsin in parvalbumin interneurons on one side of the brain, and then deliver light on the other side of the brain, okay, If on the first day we deliver light to activate halorhodopsin and inhibit these callosal terminals, we see that mice become perseverative. This lasts, so the next day if we turn off the light, mice are still perseverative. And if we then stimulate these projections at 40 Hertz, we can reverse that perseveration like what we previously saw in DLX56 mutant mice, and that reversal lasts a long time. And so this makes us think that this might be a way to actually reverse perseveration in mouse models of autism, okay? And in particular, we've been studying SYNGAP mutant mice, okay? So SYNGAP is one of these genes that we heard about before that's very strongly associated with autism. And it turns out that in SYNGAP mutant mice, you also have deficits in parvalbumin interneurons and gamma oscillations. So here are a couple of different papers. Here you see reductions in expression of parvalbumin in mice that lack one copy of SYNGAP. You see reductions in inhibitory synaptic currents. And you also see reduced gamma power in these mice. There's something similar here in this other paper from Shilpa Kadam's lab. And so we decided, we've just started doing this work, and we've been looking at this rule shifting task in these SYNGAP mutant mice. And so we'll test them on day 1, and then we'll try stimulating at gamma frequency, and then we'll measure their performance again. And what we see is that obviously compared to wild type mice, these SYNGAP mutant mice are at baseline very impaired on rule shifts. They get perseverative, they get stuck on the first rule. But just like what we saw in the DLX56 mice, when we stimulate at 40 Hertz, we're able to rescue their performance, and that turns out to be a persistent rescue. We're just in the process of doing these experiments. It turns out we can stimulate just on one side, or we can stimulate just these callosal PV terminals, at least in very small numbers of mice, and maybe that alone is sufficient to rescue the behavior of these SYNGAP mutant mice. So the overall conclusion here is just that gamma synchrony is really important for cognitive flexibility. It depends on these callosal PV projections. These seem to really strongly regulate plasticity, and targeting them might be a way to rescue kind of cognitive flexibility in mouse models of autism, like these SYNGAP mutant mice. And I'll just say, I won't show it. I have another slide showing how we might be able to do this pharmacologically by boosting gamma activity with drugs like clonazepam. That seems to have a similar effect. Okay, so with that, I'll just end, and I'll just go to my last slide and see if you all have any questions. Thank you.
Great talk. The callosal PV neurons, do they synapse only on pyramidal cells or also on PVs? Because I think the PV was actually thought to be one major driver for the synchrony.
Yeah, it's surprising. So normally PV cells strongly inhibit each other. They even strongly inhibit themselves. And these callosal projections, as far as we can tell so far, seem to specifically target pyramidal cells. What's quite interesting is that even amongst pyramidal cells, they're very specific and seem to preferentially target subcortically projecting pyramidal cells, which go, for example, to the thalamus. That's interesting because that's the same selectivity that PV cells exhibit locally. And so yeah, it's a little bit surprising that they don't target other PV cells, but perhaps they target these corticothalamic loops that might be important.
Yeah, cool. And then maybe a follow-up, the synchrony that you recorded, that you showed from, maybe I just missed that. From what layer was that recorded? Where were those cells?
Yeah, so we're basically doing, fiber photometry of voltage indicator signals. Our fibers are in the deep layers, so they should be looking mostly at deep layer neurons. I mean, the other thing to note about this kind of voltage indicator fiber photometry is that most of the signal is going to come from dendrites. And so we probably get, the layer 5 cells obviously have these huge dendritic arborizations. So that's probably what we're picking up. Well, it's just an optical fiber, and it's sort of centered over. Yeah, well, no, in this case, the voltage probe, I should say this, yeah. I don't know why we went off on this dendrite thing. Obviously, in this particular experiment, the voltage indicator was only in PV neurons. We've subsequently done other experiments where we're looking at different subtypes, but all of this is all like PV neurons, presumably mostly in deep layers. Yeah, sorry. I kind of confused myself. Yes. Yeah, so we're looking at gamma synchrony involving these PV cells, and these PV cells obviously target many local neurons, but it seems like these callosal PV projections are particularly important. And these callosal PV projections, actually we compared how well they innervate 5 different types of cells. So we compared how well they innervate other PV cells, thalamically projecting cells, mostly pyramidal cells, callosally projecting pyramidal cells which go to the accumbens, and pyramidal cells which go to the dorsal striatum. And they strongly inhibit pyramidal cells which go to the thalamus. They moderately inhibit pyramidal cells which go to the dorsal striatum. They don't really inhibit pyramidal cells which go to the nucleus accumbens or the contralateral cortex. So that's kind of interesting. All of these are regions you would think might be important, but it seems like for this particular function, gamma synchrony and cognitive flexibility, specifically the projections involving the MD thalamus and maybe the dorsal striatum that are most important. So the PV cells in the prefrontal cortex mostly express D2 receptors and are excited by dopamine through kind of a beta arrestin dependent mechanism. So yeah, we're very interested in whether dopamine is important for recruiting this gamma synchrony. That's something we're looking at. Yeah. Yeah, we have been looking at this a lot, and actually there's another student in the lab, Carlos, who's been looking at what happens during errors and are there kind of prediction error type of signals. And there are obviously signals that happen after errors, but the signals that happen after errors during the rule shift are very different than the signals which happen after errors during the initial rule learning. And in fact, it seems to be important for these signals to be different. So in these DLX56 mutant mice that I told you about, the most consistent thing we find is that the error signals, it's not that the error signals become weaker, it's that the error signals become more similar during the first part of the task and during the second part of the task. So I guess what I'm saying is we think error signals are very important, but we don't think they're generic error signals. They're more like an updating signal that's specific for the strategy that's happening at that time. Yeah. These are like multi-neuron patterns, so presumably driven mostly by excited neurons. So these are, what I'm talking about is like doing multi-neuron calcium imaging, and you kind of like find a pattern of activity that happens after an error, and those patterns of activity seem to be really different after errors during the rule shift compared to errors. Yeah, that's right. Yeah, so we're doing like resolving something like 100 neurons. Yeah. That's right. Yeah. So, this is like this endless thing in neuroscience, right? Like we find that one mechanism, it's like, what drove that, right? So, or a whole, it's like, yeah, no, it's absolutely, we're definitely interested in this. So we've been looking at a couple of different sources of input, right? So the things that we think might be relevant are inputs from, say, neuromodulatory inputs like dopaminergic inputs. We're looking into that. I don't have any results. There are changes in dopamine signaling that happen on error trials, for example. We know that, but I can't tell you, are those changes necessary for gamma synchrony? Two things that another thing we're looking at is input from regions like the insula. Maybe that's important. One thing that is important, which we're just writing up now, we should have that out pretty soon, is astrocytes are very important. We find that astrocytes are really important for these increases in gamma synchrony. Of course, astrocytes also have lots of receptors for dopamine. So we do think maybe it's some neuromodulators, maybe it's dopamine or norepinephrine, and maybe it's norepinephrine or dopamine acting on astrocytes. That's sort of where we're at. Yes. Yes. That's a great question. So what we know is that there are long-range GABAergic projections from prefrontal interneurons that go to many targets, right? You brought up, we've had one paper on the nucleus accumbens. Now I'm telling you about the contralateral cortex. We had another paper on projections that go to the dorsal hippocampus. Are these the same neurons that send these long-range projections everywhere? I don't know the answer to that. In the paper, this is Malik et al. In Cell in 2022. In the supplement, we did some tracing of the cells that project to the dorsal hippocampus, and we showed that those cells have collaterals that go to other targets. I don't know if the PV cells which project to the nucleus accumbens also project to the contralateral cortex. But what I do know is there are multiple targets, and some of the cells have multiple collaterals that go to multiple targets. Yes. Yep.
I have a question. So we don't know that during the developmental stages, the excitatory to inhibitory ratio 2 changes, and during the course of event, did you get to see how the synchrony?
So what I can tell you about this, yeah, we don't know if these long-range projections change over the course of development, but and we haven't done measurements of synchrony with voltage indicators over the course of development. We have done electrical, like intracranial EEG recordings at 2 time points, at 7 weeks of age and 10 weeks of age. And the recordings at 10 weeks of age look quite different than those at 7 weeks of age. There are much larger increases in power in the gamma frequency range at 10 weeks of age. So certainly, as the prefrontal cortex is maturing, it does seem like these gamma oscillations become a larger feature of neural activity.
And did you see how it affects with reversal learning? Did you get to see that?
So at 7 weeks of age, even though there's not as much gamma activity, this type of learning, this is like an extra dimensional shift, right? Is not there. It's fine. Like mice can do that just fine. In fact, there's some evidence from Linda Wilbrecht's lab at Berkeley that adolescent mice are even better at this type of learning in general than adult mice. Sort of, adolescence is time of risk taking and flexible behavior maybe. So yeah, that's we don't know exactly what's going on there, basically. Yeah.
Thank you.
Yep. Yeah, this is a great question. All the experiments we did, we also did in somatostatin-free mice, and we did not see increases in gamma synchrony in somatostatin neurons, and we did not see that optogenetically manipulating somatostatin interneurons had any effect here. We do think that somatostatin interneurons are important for other types of synchrony, like we have another paper on beta synchrony between the amygdala and hippocampus, and that seems to depend completely on somatostatin neurons.
Yeah. Yeah. Great. Thank you, Saurav, Anindo, and Vishanu for the opportunity to present here in the symposium. It's always great to be at NBRC. I recently was here trying to teach some systems neuroscience and hope to be back again. So in today's talk, it's something that going along with what just Vikas presented, but totally from perspective of the stimuli or the external environment, the sensory stimuli aspect. That is, we'll be talking about how surprising stimuli which may be occurring at multiple time scales, how they can influence development of the circuitry. And aspects of these, the things that I will talk about, they are covered by Srishti and Ristika later on in lectures, in talks, or in posters. So I will refer to them as we go along. So the basic idea here is, it's based on a couple of different work that we have done. The basic idea is that if you think of the visual system, the way it is organized, and probably most other systems, but with the visual one is the best example to take. That is, it is organized in a hierarchical manner, which starts with very rudimentary sort of elements like edges and lines, which then get combined to shapes, different kinds of shapes. They get combined to form percept of objects, and then to faces, and so on. And you will see that these rudimentary elements are very, very low probability kind of things in the environment. If you look at the entire wall, let us say, to define the wall, these lines here are what is really defining the wall, and the in-between part, which basically has the same thing all around, is a much higher probability, if you will, sensory input coming in. And it is these lines which are low probability that is defining this whole block. And this can be abstracted to higher and higher levels of objects. For example, this room, which has a few people in there, then if we go focus on a few people, then their faces and so on, and we can break it down backwards. So in a similar manner, if we think of the auditory system, this kind of multiple spatial scales in which we understand the visual scene, this can be broken down into basically temporal scales. That is, different time scales at which we have different events in the auditory world happening, based on which we are combining across time to form the percept of, let us say, ultimately a sentence, which can be parsed to words, which are syllables, then combination of frequencies, and initially just frequencies, which are constantly changing in terms of their energy over time, which is represented here. And this is the actual, for example, a waveform. And we will be referring to this kind of a stimulus representation of the auditory system later on quite a few times. And so this is to just to get you oriented. This is the time axis, and this is the frequency axis. And in color is the heat map essentially is showing the amount of energy that is present in different frequencies at different times. And to relate this to the mouse system, what we will go on to is basically the mouse vocalization. So if you think of mouse vocalizations, these are example spectrograms or the cochleogram that we saw of different types of calls for vocalizations produced by mice. These, each one of them are different types of syllables, which are shown here, which we broadly classify based on very early classifications. Now people do much more sophisticated classifications, but for the purpose of the analysis that we do, we need a very broad scheme of classification, where we have some broadband noise types, single pitch contours, which is the S type, the jump type, and then harmonic types, which are crucial, as we have also seen that there is specificity in terms of processing these harmonic types in the mouse, and then other types which contain multiple jumps or multiple other frequency ranges. So we will, and these kind of vocalizations sequentially are produced by mice, which we will show that is what is important in their communication. So what do we mean by surprise in this thing? That if we have a stream of sounds coming in, which is, as Peter was referring to earlier, the oddball paradigm. So if you have a standard sound sequence coming in, and then a different frequency occurs all of a sudden, then back to the standard, where we define the probability of occurrence of this deviant, the other frequency or the oddball frequency, to be sort of related to how surprising it is. That is, the lower the probability of the deviant, the lower the surprise. That is, the less it is expected. And so this probability of standard being around 80 to 90%, and the deviant being about 10%, we know from a lot of studies and earlier that even in EEGs, we can see that neurons or the activity in the brain is selective
to the deviant by doing controls of swapping the two different frequencies. So there is some important, it could be an important signal that is reflected whenever there is a sudden change or surprise that comes in, very much like learning from a change in rules. So in terms of the single neurons, what was first about 20 years ago, what Ulanovsky and Nelken showed that indeed there are neurons in the auditory cortex, and now we know they are there even down to the inferior colliculus. The neurons are highly selective to the deviant frequency, and so this can form the basis for signaling the presence or occurrence of a surprising sound event. And so does this translate to multiple or longer time scales or sound structures? So to do that, we studied stimuli by using stimuli that are of this nature, where we have standard tokens presented with a deviant token embedded at a particular position within a sequence of 15 tokens. And that same structure is repeated over and over again, versus a case where we have the random sound occurring at different locations within that same sequence when we are presenting it over and over again. So in this case, although we have a surprising sound in there, the overall structure is getting repeated. So in that sense, there is no surprise as we go along. And here, every time the deviant occurring at a different position is highly surprising. And as you can see from here, based on the responses of the auditory cortical neurons, we see a much stronger response to the deviant in the case of the irregular sound sequence than in the regular sound sequence. So even for a sound structure, the response to this deviant frequency becomes extremely strong. And when we have the entire sound structure over which it is adapting, the response is far weaker compared to the response to the deviant over the different trials. However, there are populations of neurons that actually prefer regularity also, which is a different story. We will not get into that here. We see that this is present through two-photon calcium imaging with excitatory neurons and two particular subclasses of inhibitory neurons, PV and SOM. They also show similar kind of selectivity. And based on more functional connectivity analysis within the local network, we find that the different preferences, regularity preferring, irregularity preferring versus non-preferring neurons, they seem to form sub-networks within the local cortical region, which is largely defined by the particular inhibitory neuron that is projecting to them more than the other one. For example, I mean, this is the excitatory neuron, which is connected with, let us say, we have two types of inhibitory neurons here, the PV and SOM. So this non-preferring group, they are highly connected within each other, and it is the stronger connection made to both of them here. And here you will see this is the stronger connection made to the SOM type. Here we will see that the stronger connection is made to the SOM PV type, and so on in the different sets. So this thing translates even to sound structures. And so how do we study this going forward with, yeah. No, this is, so we collect data from multiple neurons simultaneously within the local network. And then we connect, based on noise correlations and other analysis also, in this case, we did Granger causality also, to define which neurons are more functionally connected with each other, more like the synchrony that Vikas was talking about. Not exactly the same, but similar. But within the local network. And is that so? Now, if we now the question is, if this surprise is at all important, does it play a role in terms of how the cortical circuitry develop? And in this case, we have what I'm showing, that after embryonic, after P0, when the hair cell starts to mature and finally matures by P6, and then beyond up to P12 when ear canal opens, and up to P15, which this period is known as the so-called critical period so far. we wanted to see that if such surprising stimuli are what can be causing development what could be the role of such surprising events in driving the cortical circuit even when it develops, or does it do that at all? And this was based on some questions that we had because of the known cortical circuitry development from Patrick Canol's work and other work, where we have a set of neurons called the subplate neurons during development, which are transient in nature, which receive thalamic projections and which project onto layer 4 initially. And the thalamus then sends collateral onto the layer 4 neurons, and this cortical circuitry finally, due to the activity of the subplate and the inputs coming in, the final circuit that forms is from the thalamus to layer 4. And there are experiments done by removing subplate neurons or manipulating subplate neurons that shows a number of deficits in cortical development and final cortical structure. And we had been trying to record from subplate neurons during these periods of time. And what we had found is that subplate neurons indeed show strong selectivity to deviant sounds compared to the standard. And whereas layer 4 neurons actually do not show that strong deviant detection. So that was the sort of the basis for our ongoing investigation. And the idea is coupled with the fact that if you think of a baby in the womb, let's say they're supposed to start hearing from the end of the second trimester, and could it be they are continuously hearing a standard sort of background noise, and within them there are sounds that are embedded, and there are studies sort of showing that the baby recognizes the mother's voice and so on. Could it be learning based on this principle of this surprising sound happening once in a while in a very very low probability manner compared to the ongoing background? And so what we did is we used exposure paradigms like with standard embedded with deviant and multiple controls and multiple exposure ages. And we show that actually after development, when we performed experiments by recording from single units in the adult after exposures at these different ages with standard deviant protocols or only standard protocols, we find that in terms of total neuronal activity of all the data that we had collected, the distribution wise they're same. There is no sort of difference in terms of the total activity that we are seeing based on each of these exposure and the control group. And here, however, when we look at the specific responses to the deviant sound and to the standard sound in the standard deviant exposure case, then what we find is that when it is exposed at just before ear canal opening, P6 to P11, where there is hearing, the deviant sounds become stronger responders. I mean, the neurons becomes more responsive to the deviant sound, the ones that have their best frequency at the deviant frequency in the adult circuit. And basically the standard response actually decreases, and other controls basically allow us to conclude that indeed that low probability sound, which was occurring over the period of development between P6 to P11 in that duration, could lead to this kind of increased sensitivity to that particular frequency. More details about this will be presented by Ritika, and their effects on VPA model of ASD is what will be discussed in a poster. So now this was further corroborated with a model where we have the subplate and layer 4, and by using the spike timing dependent plasticity rules, we could see that indeed a network could be learning based on the deviant, where the synaptic strength for the deviant input increases over development with exposure from standard deviant protocol and not to the standard, although it was occurring multiple times. It simply has to do with adaptation. And this further, we looked into this as to why this could be happening. For that, we wanted to see if it, I mean, that was a basic idea that if the activity required to represent all the sounds, if that is sparse in nature in the population, that is, we can use minimum energy to represent the entire sound statistic, then would it be that the low probability sounds are getting higher representation than the other ones, which is very intuitively very simple to say actually, but it required a lot of maths to actually prove that, because there are many problems in the way we go about it. So anyway, so under conditions, under certain conditions, which we cannot go into the details now, we can show that theoretically also, if a network tried to represent the information about the statistics of all the sounds that are coming in, or the stimuli that are coming in, it would end up favoring the low probability sounds or low probability stimuli and down favoring the high probability stimuli. And if it has to do that with minimal energy or minimal cost or minimal activity, while retaining the maximum information about the stimulus. So does this single frequency kind of thing translate to a different setting? For that, what about sound structure or sequence? For that, we looked at the specific types of sequences, which is vocalizations in mice in different social contexts. And as we know, in the pup isolation calls, it is a socially relevant call for them, in the sense that pups produce the PICs whenever they are away from the nest, which evokes a maternal search behavior. And similarly, of course, vocalizations have been shown to be extremely important for mating behavior and so on. And there are other contexts also. We particularly chose the mating context one, where with under different contexts, we recorded vocalizations where, again, with the N, S, J, harmonic, and O type of calls are there. And based on the sequences of vocalizations that are produced during such courtship interactions, in the three different contexts, when the male is alone, where it does not vocalize at all, but after being in a chamber with the female, where the female would be introduced later, they start to vocalize even alone. Those three contexts, they produce vocalizations, and we found that these vocalizations contain sequential structure, in the sense that they are dependent on previous vocalizations or syllables that are being produced, very much like what we see in speech, where Shannon showed that there is predictability in speech sequences in words when we speak in English, or for that matter any language. And that has been shown for a long time in the bird song also, which Soumya had spoken about. And so based on that, we computed which are the most surprising sequences, in the sense that the sequences that are least expected, yet they would be occurring, least expected from probability of single different tokens, but they were still would occur multiple times. So that makes them the most surprising ones. And so we found that based on one way of analysis, that 8 of these sequences were emerged in groups of mice. And these we termed as natural sequences, which generally had the harmonic content to be a big player in the sequences that were derived. And based on the probability of occurrence of each of those syllables, we could create a set of sequences that are called the random sequences. So these are natural in the sense that our mathematical derivation said that these are the most surprising sequences or most informative sequences. So does that play a role in terms of what the female mice see it as or hear them as? So for that, we did a 2-place preference task, where we played the natural sequences from one corner and the random sequences from another corner. And female mice tended to prefer the natural sequence corner compared to the random sequence corner. And switching the sides of presentation of SR and SN in a session after that showed that they reversed, they actually still preferred the natural side and reversed the side together. And in this study, we also finally showed that actually there is selectivity to the entire sequence, which Srishti will talk about a little, as opposed to single tokens. And so neurons, and with experience, with exposure to a male, the female mice, the neurons change their selectivity towards the natural, and these changes were found only in the excitatory neuron and the somatostatin positive neuron, and not in the PV positive neuron. And to corroborate that further, we did some somatostatin positive inactivation experiments with sequence presentation, which showed similar behavior as the experience dependent plasticity that we saw in selectivity of the sequences, natural sequence. And so with all this, what can we learn more about it other than the other things that Srishti and Ritika will talk about? We have been looking at a very rudimentary level at a theoretical principle in terms of the development of networks. And that has to do with basically, can we have a network that simply learns based on surprise in the incoming stimuli? And so for that, we basically have to do what is an expected stimulus, and for that we use the generative model to learn from sound, from speech and natural sounds, and we imposed some criteria for the cost of or the activity, which is basically an L1 norm right now. And by minimizing the cost and maximizing the mutual information between the input and the output activity, which is the basically enhancing or weighing the surprise more. If you have simply a one layer network of neurons learn and the connections that it leads to, we find that tuning actually develops in these neuron networks. And these are being done at multiple time scales. This is at 1 millisecond time scale with broad tuning at the lower frequency sides, and with at a higher or at a lower temporal resolution, you get much narrower resolution tuning, which is what is expected in which is what we see in the auditory system. And so this is really in the rudimentary stages, and we are presenting it at Cosine hopefully next year. And so I would like to end by thanking the lab members and the people who did the work. Srishti is here, Ritika is also here, and other past members, and of course thank the funding. Thank you.
But it's very cool result that you see this differential response in the condition place preference with the natural and the surprising USVs that you replay. I'm wondering, can you actually train the mice that they reverse their preference for that they get used to the unusual?
Yeah, that is the that is the difficult part. I mean, with a reward, maybe I don't know. That is something that can be done. But we have actually gone to a different direction on that. Not exactly the question that you are asking, but that's something to try.
Really interesting talk. I don't know if you have any data or to just speculate about, obviously different interneuron subtypes have different kinds of dynamics, both from their properties, but also their synaptic properties, the incoming synaptic properties, lens cells, the migrating signals, or changes. I don't know if that plays at all role they play in processing.
So actually, I would refer to some of the work that Srishti will present. We find that this is ongoing work. We find that, so if I go back to the sequence of sounds here, what we find is that the tokens, there is no plasticity in the representation of the single syllables. That's the main thing. The plasticity is in the representation of the entire syllable. I couldn't go over all that data, entire sequence. And so what we hypothesize is that it is activity ongoing in between the two tokens that must play a role, which is basically an off response after the stimulus is over for a single token experiment that we always do. And some neurons play a big role there in terms of that off period response. So we think that that may be helping it. In fact, we have started those experiments, but I cannot share right now.
So good evening, everyone. So as you have already known, I work in IIT Kharagpur and under the supervision of Dr. Saurabh Bandyopadhyay. And you will find some of the slides common. So it's actually a work easier for me. So I'm sorry, like my work is depending on development plasticity of the mice auditory cortex. So we are basically interested in how specific stimulus are actually playing a role in shaping the auditory cortical plasticity at that particular developmental age. So earliest sensory experiences leads to profound changes in developing brain. As my supervisor has already explained some of the part of this particular work, that is a previous work from our lab. So a background of the stimulus that we're using is a oddball paradigm, but there is a background for that, why you have chosen that particular stimulus. So in the real world environment, when a baby is within the mother's womb, it is surrounded by the amniotic fluid and continuously being exposed to that rustling sound of that particular amniotic fluid within the womb. And it's being also exposed to the mother's voice, which is much more frequent compared to the other voices in the environment, but less frequent than the rustling sound of the amniotic fluid that it is experiencing within the womb. So we can actually think this particular background noise as a continuous repeating sound, which is being hindered intermittently by the mother's voice. So this is a continuous white noise in the background that is occurring because of the amniotic fluid. So the developmental profile of the auditory systems shows that so we are now interested in so lines of evidence has shown that in the previous figure, in the previous slide, I just forgot to say that when the baby is born, it actually responds more or shows a particular kind of selectivity to the mother's voice compared to the other kind of environmental sound or any other person's voice. So that means that some kind of change in plasticity is already been occurring when the baby is within the womb. Otherwise, it's impossible for it to recognize it when it's being born. So what is the kind of changes that is being occurring in this auditory cortex when the baby is still within the womb? So we want to figure it out. So in human beings, it's impossible to do because the baby is still within the womb. But we can to answer this question, we can use mice as a model, because in this particular timeline, if you see, this is a developmental timeline of mice from E0 to P15. And if we just map this particular timeline to human, it falls in the week 0 to week 40 gestation week in human beings. So basically, the changes that are happening in between this P0 to P15 age of mice, when the mice is already being born, is actually, we can say that the changes here are actually happening when the baby is still in the womb. So we can use mice as a model here. So at P6, the hair cells matures, which is the peripheral sensory apparatus that is leading to the sensory auditory sensory transduction. At P12, the ear canal opens, starting with the starting of the critical period, and P15 it shuts off. So in order to see what kind of changes are happening within this particular developmental timeline, we are interested on a particular type of cell layer called the subplate layer, which is a key player in this kind of plasticity that is being happening. So any kind of sensory information, as we know, before going to it needs to go to the cortex for any kind of behavioral manifestation. But before going to the cortex, it makes a connection with the thalamus, and then the thalamic afferents, before going to the cortex, layer 4, it makes a transient connection with the subplate layer, which is a transient layer that starts disappearing from postnatal day 21 in mice. Okay, so initially the connection is between thalamus to subplate, and with time this subplate layer disappears, and there is a continuous connection from thalamus to layer 4. So why this subplate layer is important? Because this subplate layer has been shown to be important in this kind of neurodevelopmental disorders, because in the second trimester has been shown to be very much vulnerable to this environmental attack because of the subplate layer. The subplate layer can experience various kinds of manipulation because of the environmental assault during the second trimester, and also, as you can see here, hypoxia, ischemia, oxygen deprivation, and valproic acid can cause alterations in the subplate circuit change. And we also know that valproic acid can cause autistic kind of phenotypes. So concisely, we can say that maybe that valproic acid induced autistic phenotypes could be because of the subplate circuit change. So in NDDs also, subplate layer has been shown to be very much important. And in our study, the previously I have said that that standard deviant kind of low probability and high probability sound that the baby is experiencing, and because of that we are using this oddball paradigm. In that case also, subplate and layer 4 plays a very important role. So this is the stimulus that we are using. This is a standard tone. So a particular tone that is being repeated continuously, which is hindered by a low probability tone that is called the deviant tone. And this is a before ECO data. We have before ECO from P6 to P11. They have taken data from it is work from previous work from our lab. So they have taken data from like P8, P9 mice. I have shown that in before ECO, the subplate in the subplate layer there is an increased D response. So for the deviant frequency, there is an increased response. Here, as you can see here, there is increased response for D frequency, and for layer 4, the standard frequency has a more response. So before ECO, the deviant frequency, like the subplate, is acting as a deviance detector, and after ECO, if you can see here that this deviant response has been decreased compared to the first standard response. Okay. So after ECO, layer 4 is layer 4 is playing a deviance detector role, and before ECO, the couplet is playing a deviance detector role. And CSI is the index by which we can quantify if a particular neuron is a deviance detector neuron, but it's particularly its equation is deviant response minus standard response divided by deviance plus standard response. So the if this particular index value is more than 1, then we will say that that particular neuron is a deviance detector neuron. So in this figure, if you can see, throughout before ECO and after ECO, the CSI value is positive. For sublet neuron, it's positive before ECO, and the layer 4 value is lower than the sublet value. And after ECO, this layer 4 value is increasing and the sublet value is going down. So before ECO, sublet are the deviant detectors and layer 4s are not. And after ECO, this layer 4 neurons are acting as a deviant detectors. So now we know that in the in the mice before ECO and after ECO, layer 4 and sublet are playing an important role. They are the deviance detectors. But are this kind of changes are plastic in nature, or are these particular kind of stimulus have an effect in the adulthood? So to see that, they have exposed mice from P6 to 11 before ECO and P11 to 16 after ECO, and have shown that as my supervisor already said, after ECO, this so after ECO there is an increased response. Before ECO there is an increased. Okay, sorry. After ECO here, so before ECO P6 to 11, there is an increased response for D and a lowered response for standard and the first standard of standard deviant. And after ECO there is an decreased response for D. Okay, so from here also you can see that the before P6 to 11, these sublet neurons are acting as deviance detectors, and that particular deviance detection property is being transferred to layer 4, and all this data are being taken from layer 4. Okay, so now we know that deviance detectors are actually being occurring, but we want to know if in the absence of standard also this particular deviance detection property is still there in the neurons. Why a particular neuron is showing a higher response to that particular D frequency? Is it because of the preceding context of the standard tone, or in absence of that also it will act as a deviance detection, or it will show increased response to that particular D frequency? To show that, my objective is context of the sound important in the earliest experience based auditory cortical plasticity, and also we want to check the data that they found this kind of data that in before ECO and after ECO, sublet and layer 4 are acting as deviance detectors. So that is, the pups are very small and it is very challenging. The electrophysiology results are very much challenging at that particular age. So we want to check that if CFOS in the CFOS immunohistochemistry also we get the same kind of result. CFOS is an immediate early gene and it's a marker of neuronal activity. So are the sublet and layer 4 neurons activities before and after ECO gets reflected in IHE? CFOS expression correlate the observed plasticity. Okay, so in we are trying to see it through CFOS immunohistochemistry. So does the context will play any role? Because so the hypothesis is there is a preceding standard. After that, there is a deviant. So since because of the preceding standard, for the first standard the neuron is giving a response, but as the preceding standards goes on, there is adaptation for the standards, and there is substrate voltage. Their substrate voltage accumulation by the preceding standard tone leads to firing on arrival of the deviant tone, could be an explanation for increased response to deviant tone. Okay, so for the preceding standards, there is the response is being decreased, but it is being accumulated as a substrate voltage summation, which when the deviant tone comes, there is a sudden surge in the firing rate. So to see that, we have done the same exposure experiment like my previous lab members, where I have exposed mice from P6 to 11 before ECO and P11 to 16 after ECO with the deviant only frequency at 2 dB level, 70 dB attenuation, 90 dB attenuation, 70 dB dB level and 90 dB level. And then after this exposure, these mice are being transferred to the animal house, and then we did at P8, P28 to P42 age group, we have did in vivo physiology. And then by playing this particular tone from 6 to 48 kilohertz, placed at 0.25 octave difference, we are trying to see if what are the changes of this particular D only stimulus during adulthood. And we have used 4 groups where exposure before issue and after issue and at 2 dB levels. So here is the result. So okay. So this is before issue and after issue. So now if you just remember the data from there, standard deviant, they have played exposed in standard deviant and I have exposed in deviant only in absence of this particular standard tone. So now the result shows that here if you can see there is an increase in firing rate in, so this is exposure at 9 dB SPL and this is exposure at 70 dB SPL. So this particular firing rate is increased before ECO and after ECO this is being decreased, the same result that we have seen in standard deviant exposure paradigm as well. So for the higher dB level, the pristine context is not playing any role. But for the lower ones, if you can see that the result is being opposite. Here, instead of the higher firing rate, the firing rate has been decreased, and after ECO, the increase of instead of getting a decrease, it's being increased. So we can say that this particular context is important when the when the particular that kind of deviant only paradigm is being played in the lower dB level, context does play a role, but when this particular deviant only paradigm is played at higher dB level, context doesn't play any role. Okay. So next we are trying to see it through the CPOS experiment if the result that you have seen holds for the CPOS experiment as well, because whatever result we have seen, it could be translated to CPOS experiment as well, because CPOS is an immediate early gene or neural activity marker. So here we have taken mice age group of P13 and played this 4 stimulus with a habituation period of 60 minutes, silence of 30 minutes, and silence in consideration of 30 minutes. And these are the 4 stimulus that are being played in like 4 mice with the 4 stimulus, and then we have quantified the data. And this is the control data. And so if you see here, you will see that in layer 4, the standard deviant response. For the standard deviant response, the layer 4 is showing more expression compared to the subnet layer standard deviant expression. Okay, the same thing that you have seen in the electrophysiology results as well. There also after ECO there is an increase in the deviant frequency in the standard deviant protocol and compared to the standard. And here also in the CPOS, we are seeing that in the layer 4 for the standard deviant, the expression is higher than the sublet layer. And also we have quantified the data for inferior colliculus. And here also you can see that for the DIC, or the dorsal cortex of IC, there is an increased expression of CPOS, because from lines of evidence, we already know that this dorsal cortex of IC plays an important role in deviance detection. So our CPOS result also holds true for the electrophysiology as the electrophysiology results that you have obtained from the previous works from our lab, and this is a deviant only result. So if you can remember, so all the stimulus were being played at 90 dB SPL. And here also in the previous slide, as I have shown, that context does play a role in the when the particular deviant only is being played in the lower dB attenuation, but in higher dB, when played at 90 dB SPL, context doesn't play the role. So here also, this is the after issue P13 age data, and the mice is being exposed to D only, and that is the D only is being played in 90 dB SPL. And here also we'll see that increase in expression compared to the SS expression. Okay. So that means here also when that particular D only stimulus is being played at higher dB SPL, the context is not playing any role. So this is the CPOS expression here, and we have quantified for V1, V12 just for the control purpose, and the 3 regions of the auditory cortex. And this is the CPOS expression in the IC, and that is after ECO. So the conclusion is, at lower intensity exposure, the preceding context is important in driving the deviant related plasticity, which holds the hypothetical subthreshold voltage accumulation to be true, whereas at higher dB intensity, the deviant frequency alone is enough to drive the plasticity. Larger CPOS expression in L4 compared to SP in response to SC stimulus paradigm, and stronger expression in L4 in D only stimulus goes in line with the electrophysiology result that I've shown you earlier. And the last one is also in the DCIC. We have seen that there is an increased expression in DCIC, and from the previous evidence, we know that DCIC plays an important role in deviance detection. So that also goes with the electrophysiology results, and also the previous results from other labs. Okay, so thank you.
Hello, Orkarup?
Yes.
Yeah, can you hear me?
Yeah, I can.
Yeah, can you switch on your video just for a second and then move on to the top? Oh, and just share the PPT. Can you
Sure.
hear me? Yeah.
Do you see my screen?
Yeah, I can. Can you make it full screen?
Yep. Yeah.
Yeah. Yeah. Perfect.
The only thing is I need to share sound. So hopefully share sound. Okay, that should be good.
Okay. Okay, perfect. So Orkarup was supposed to be here, but unfortunately couldn't make it this time. And he's from Cold Spring Harbor, and he'll be talking about the vocalization using a very unique type of mice that he has from Ecuador, and his studies on social interactions. So Orkarup.
Okay. All right. So hopefully you can all hear me. It's a real pleasure to participate in this conference, although remotely. As Saurabh said, I was really hoping to meet all of you today in person, but this will have to do for now. And I really want to thank the organizer, Saurabh Anindo Krishanu, for giving me an opportunity to talk about our work in this exotic rodent species of singing mouse, which I'll introduce to you. By the way, this is Cold Spring Harbor. This picture was taken a couple of months ago in autumn, and my lab is somewhere hidden behind these trees. So I'm an assistant professor at Cold Spring Harbor. I've been here for the last 4 years and really excited to share with you our work. Okay, so I don't have to belabor the point that human society involves a lot of vocal interactions, and language allows us to do many of the incredible things that we are very proud of as a species. And especially if we consider conversation, which is the focus of my research, it involves many different kinds of mechanisms that has to do with moving tens of muscles in interesting ways. But especially during conversation, you have to couple all of that motor activity with incoming auditory activity and couple this in extremely fast ways. So this ability to take turns during conversation is something that as a neuroscientist, I got very excited about maybe 4 or 5 years ago. And I started thinking about how to approach this problem. And vocal turn-taking behavior obviously is not unique to humans. It's found across the animal kingdom. Here I'm showing you a few examples of vocal turn-taking that one can study in the lab, perhaps. For example, common marmoset has this robust vocal timing behavior where one marmoset will make a phi call and the other one will respond within 5 seconds or so. This has been extensively studied in zebra finches, who also show these kinds of turn-taking behavior with their calls. So it is quite widespread in the animal kingdom. Now, vocal interactions require flexible sensory motor loops. So it involves passing a message between a sender and a receiver. So the sender needs to use its motor system to produce a message, which through noisy channels is transmitted to the receiver, and using sensory perception, the receiver needs to interpret that message. What is interesting about vocal interactions is that it's actually a loop. It's a feedback process. And in humans, it has been shown that the average latency between one speaker finishing its turn versus the next one starting up is about 200 milliseconds, which is really, really fast. And think of all the things that need to happen in that 200 milliseconds, starting from auditory perception to motor planning to movement of the right kind of muscles and conveying of this message. So I want to introduce to you now the protagonist of today's story. These are these Alston singing mice that we study in the lab. And as I'll tell you, these mice are really good at this particular vocal turn-taking behavior, and they have this remarkable property of singing to each other, but politely. Okay, so little bit of a background on these singing mice. So they're formally called Scotinomys teguina. They are neotropical rodents found in the cloud forests of Central America, starting from Costa Rica to Ecuador to Nicaragua, to Panama. And they are small, they are diurnal rodents. They are about 15 grams. They have this innate vocal behavior where songs are an honest signal used for male-male competition as well as for mate selection. There exists a large behavioral divergence among closely related species, so which is good because now we can explore some of the evolutionary questions of how the song system came to be. More importantly, they can be bred in the lab, they can be maintained as a colony, and I believe they are an experimentally tractable system for systems neuroscience. And people have been working on this species for maybe a couple of decades now, especially Steve Phelps, who's at UT Austin, who's an evolutionary biologist. And we are now trying to figure out how to make this into a model system for systems neuroscience. So I'll first show you a video that demonstrates their behavior to you. Hopefully you'll be able to hear the sound. Okay, so what you see here is a singing mouse in a lab, and this video was taken by me a few years ago. And what you will see is an operatic posture where the mouse will stand up and then make a song. This is human audible. I haven't actually changed the pitch at all. I'm not sure if you actually heard it. Okay, I still don't know. Did you hear it?
You can play it again.
Okay. Okay, I'm not sure if you heard it. So, so these mice make these songs that are about 8 to 10 seconds long, and they have this operatic posture when they do it. Okay, so what's the fine structure of the song? The song consists of a series of down sweeps of the order of about 100 or so that evolve predictably over tens of seconds. So this is one song, and then these down sweeps are single notes, and each down sweep is something that goes from about 40 kilohertz down to maybe about 8 kilohertz. And they evolve predictably because notes at the beginning of the song are really short, and by the end of it, it becomes almost like 200 milliseconds or so. Okay, now what is really interesting is that these animals not only sing, they actually sing to each other. So on the left is an animal that will sing, and then on the right is an animal that actually will listen to it and then respond with a latency of about 200 milliseconds or so. And in this setup, we have made sure that the animals can't see each other, they can't smell each other, and the only thing that conveys this information is actually the sound. Okay, I'll try one more time. Let's see if this works. So the mouse on the left sings here, and mouse on the right actually tries to find where the song is. And it responds within 200 milliseconds. I'm not sure why the other mouse thought it would be good to escape. Okay, so this behavior is extremely robust in the lab, and here is an example 1 hour of such an experiment. I'm showing you the auditory trace of two animals, mouse 1 and mouse 2, in two different colors, and each of these blips are a song that these animals produced over the course of an hour. And if you blow it up, you can see this characteristic telltale sign of turn taking, which is one animal sings, which is this mouse 1, and then mouse 2 responds within a few hundred milliseconds. And in the other case, it was mouse 2 who started this interaction bout with mouse 1 responding, and then back to mouse 2. And this is extremely robust, and they'll do it hundreds of times in a given hour. Okay, so if you align the songs of, say, the mouse 1, and especially the end of it in black here, what you notice is this increased probability, a sharp increased probability of the beginning of mouse 2, which, as I told you, is what this interesting behavior is all about. They listen and they can use the end of a song as sort of the auditory trigger to begin their own vocalization. And what is interesting about it as well is that there's a profound change in the stereotypy of the behavior when these animals go from singing alone, which, as you see here in black, they produce these stereotype songs of about 7 seconds, versus when they engage in social interaction, they can change the song duration by 100% or sometimes even by 200%, where they can sing songs that are really short but also extremely long. And then they go back to being stereotyped when they are singing alone again. Okay. I want to tell you a little bit about their temporal precision. So if you do the same analysis that we did for the singing mouse here, which is on the left, with a marmoset, for example, we can find that, like I mentioned, marmosets will also do these vocal turn-taking behavior, but their latency is somewhat longer compared to what we see in the singing mouse. And I think somewhat interestingly, there's an overlap in the distribution between the temporal precision shown by humans and the singing mouse, where the mode seems to be comparable, although there's a large deviance in the distribution in the singing mouse that typically one does not see in human conversations. And as a disclaimer, I do not work on autism. I have some peripheral interest in it. And I just want to mention a couple of studies that I found recently where people have measured dyadic interactions in humans. And it seems that some of these properties like turn taking are affected in ASD. So for example, on the top here is a paper where they have done some automatic extraction of speech and turn taking parameters, and they can use a support vector machine to correctly predict whether a dyadic interaction comes from non-autistic participants versus mixed participants. And in the bottom here, they have specifically looked at some of the properties that are affected, and they claim that there are longer turn-taking gaps and a greater proportion of pause time and less variability. So perhaps some of the things that I have been discussing with you about the vocal properties of the singing mouse might have a relevance to this. Now, I mentioned that this behavior is extremely unique to the singing mouse, so I wanted to give you a couple of slides about where this comes from in an evolutionary sense. So the singing mouse, they are crescitated rodents. They are separated from lab mouse by about 24 million years. And what we found recently was that the song system is indeed a behavioral novelty, but they also produce ton of ultrasonic vocalizations, especially when they are close together. Okay, so in this case, there's an evolution of a new vocal system without the loss of the ancestral one. And the ultrasonic vocalizations, we hypothesized, are used for short distance communication, whereas the songs, they're actually used for long distance communication when there is visual occlusion in these cloud forests of Costa Rica. And here is a video and a behavioral experiment that actually tests this hypothesis. So here we have two animals that are not that separated. They can actually be allowed to come closer and actually interact with each other. For this particular experiment, for technical reason, we actually prevented them from coming and interacting directly. They can interact through a barrier, which allows us to assign their vocalizations to a great degree of confidence. So what we find here is when they are interacting with each other at close range, they make a ton of ultrasonic vocalizations, and I'll pause the video right here to show you these are very different from songs. These are reminiscent of ultrasonic vocalizations that we see in all other rodents, including in lab mice or rats. Okay, so they'll produce these vocalizations as they're coming closer or further from each other, and they do this interesting mode switch between the ancestral ultrasonic system and this novel song system. There are three key behavioral innovations that are needed for this system. Firstly, the songs are about 50 decibels louder than the ancestral USVs. They have this interesting temporal patterning that can be described with a linear model pretty accurately that evolve over tens of seconds. And they are involved in turn taking with the song system, whereas there does not seem to be any turn taking with ultrasonic vocalizations. So at the level of the circuitry, these are the three things that one ought to be able to explain in the brains of the singing mouse and how they produce these songs that are really unique to them. Okay, so I'm going to give you a glimpse of the kind of experiments that we can do in systems neuroscience in the lab, trying to understand some of these song patterning and turn-taking capabilities in the singing mouse. Overall, in the lab, we are interested in all sorts of questions, both proximate and evolutionary, and thinking about Tinbergen's four-question paradigm. We have begun to look into the mechanism, the adaptive value, even the development of this behavior in the singing mouse, and really interested in understanding how this behavior evolved from these ancestral USVs that are shared between rodents. But today, for the sake of time, I'm going to focus on a mechanistic question about how does the brain enable fast and flexible communication in this particular system. So to begin with, a few years ago, with my colleague Daniel Okobi, we identified a motor cortical area that might play a role in this behavior using electrical microstimulation. So we went around and electrically stimulated different parts of the dorsal cortex while measuring EMG activity in vocal musculature and found this anterolateral portion of the motor cortex that produced a hotspot. So when stimulated, with short latency, it can evoke robust EMG signals in the vocal muscles. So that seemed like a good candidate. So then we went on and electrically zapped the motor cortex in the middle of a song. And if you did that, we could momentarily pause the progression of the song. So as you can see here in between these yellow bars, there's no vocalization. But what was interesting that if you analyze this data that we generated, we could actually show that during this electrical stimulation, the pattern generation actually paused as opposed to evolved. And this is not merely an execution deficit in the production of sound. We went on to actually use pharmacology to try to inactivate motor cortex. So in this case, this was a playback experiment. So I pretended to be mouse one and played back songs, and the singing mouse responded pretty robustly to it. Even if I injected saline as a control, but if I injected mucimol to inactivate motor cortex, there was a profound impairment in the ability to do vocal turn taking. And as you can see in the tiny fraction of the cases where they managed to respond, the latency was extremely long and very atypical of their normal behavior. So if you can quantify this across animals, we found this robust impairment in counter singing, this ability to do turn taking with motor cortex inactivated. Now, in order to understand how motor cortex enables this turn taking behavior, we decided to go to electrophysiology, and I developed, along with my colleagues, method to do chronic electrophysiology in the singing mouse. It required some engineering because they're pretty lightweight, and then we had to come up with designs that would allow them to hold this on their skull and move around and sing freely. But we managed to do that, and now we can routinely do chronic electrophysiology with silicon probes, where we can measure their activity over maybe a month or so while they're engaged in freely moving behavior and singing normally. And if we do that, what we found was that about 50% of neurons in the motor cortex show neural dynamics that are locked to different parts of the song. So for example, here I'm showing you some examples. The song starts here, the song ends at the end of this bar, and you can see some neurons that show ramping activity during the beginning of the song. Those would be the motor initiation neurons. And there are some neurons, conversely, which show a large burst of activity right at the end. But then also there are many neurons that have persistent activity, either sustained in going up or coming down during the progression of the song. Now, we wanted to understand how is it that neural activity can contribute to the patterning of the song in this case. So we leverage the natural variability of song duration in this particular behavior. So sometimes the animal will sing a song that's really short, and then the same animal a few minutes later will sing a song that's really long. So if you have songs of various duration, you can construct a couple of models of what neural activity might look like. So for example, you might have something that follows absolute time, where neural dynamics is constant at the sort of beginning of the song, and there's this additional neural dynamic by the same neuron, or maybe a different neuron shows up for the later part of the song. In contrast, Model 2 would predict that neurons follow relative time, and between songs of various duration, it's basically the same neural activity just being stretched or contracted depending upon the duration of the song. So we tested this formally in a paper that came out earlier this year. And we found that neurons actually have co-vary with song duration in accordance with Model 2. So I'll show you some data to make this case. So here's one neuron that we have recorded over 29 songs of different duration. The shortest song is on the top, the longest song is at the bottom, and each tick is an action potential from this neuron. Now, if you look at this activity of this neuron in blocks, so such that you look at activity for a block of short songs versus long songs, we find that this neuron seems to have this activity that's stretched in time as the song duration increases, and it's not just that one neuron. And we found that it happens for almost every neuron that we recorded and have motor related activity due to song. So here on the right, I'm showing you two other neurons with different activity profiles, but they also seems to do this a particular kind of scaling with song duration. And you can formalize this by saying, let's take one neuron and all the neural activity due to that one neuron for different songs, and let's take pairs of those songs. So for example, here, the longer one and the shortest one, and let's find the scaling factor. How much do I need to compress the neural activity of a long song in order to best match it to a template, which is a short song here? And that scaling factor, which is in the y-axis here, seems to perfectly scale with the ratio of the song duration, which is the x-axis here. And there, for 5 such activity profiles, there are 10 such comparisons, which is each one of these is a pair from this particular neuron, and can show this linear relationship between the scaling factor in the neural domain versus the scaling factor in the behavioral domain. And this was not only true for this one particular neuron, but seems to be the case for 107 neurons that we recorded from 4 animals in the singing mouse. And we found this beautiful scaling relationship between neural activity and the ratio of the song duration. So what we think is going on is that the neural activity during a long song is simply a stretched version of the neural activity during a short song. And it's the change of the speed with self-similar dynamics that actually controls the flexibility of this behavior, which allows those animals to sometimes sing a song that's 6 seconds long and then follow it up with a song that's 18 seconds long within a minute or so. And this requires no plasticity, but only a change of temporal scaling in the motor cortex that seems to be good enough for this. So we can rule out model one, and we think it's basically the scaling of neural dynamics in time. Now, neural activity measurement is simply correlational, so we do not really know at this point whether this is causal, but we can do an experiment to verify that. So in this particular experiment, what we did was we actually used focal cooling, and we lowered the temperature of this particular portion of the cortex, this OMC, or facial motor cortex. And what cooling does, it stretches neural activity. So for example, here is a famous example from Yves Marder's lab in the lobster, where at lower temperatures, the neural activity remains self-similar, except it just becomes sort of stretched. And this is because of all the biophysical properties that are temperature dependent. So we can use this and lower the temperature of motor cortex. And if neural activity in this region is causal for the behavior, we would see concomitant changes in the song as we cooled motor cortex. And that's exactly what we found. I'm just showing you an example data here, where if we cool the motor cortex by a few degrees, we find that the song actually becomes longer and longer, and it becomes longer by increasing the number of notes. So that gives us a hint that this neural activity that I was telling you about is actually causal in generating this behavior. So here's the final model that we think controls this particular behavior from the cortex perspective. The motor cortex sits on top of a vocal motor network that produces individual notes and controls flexibility. It controls when the song starts, when the song stops, and how long the song should be. And these are exactly the kind of things that you would need in order for these animals to be able to participate in social interactions and do turn taking. So we think that motor cortex plays a fundamental role in this particular behavior. And the implication here is that there seems to be a non-primate mammal, which is the singing mouse, a rodent, where motor cortex exerts control over vocalization. And this is not the norm. This is somewhat going against the known dogma, where there was a strong preference to believe that the ability of motor cortex to control vocalization is a primate specific thing. And at least in this one rodent, we find an exception where motor cortex seems to exert robust control over vocalization. So I'd like to conclude by telling you some of the things that are going on in the lab right now beyond what I just showed you. So using this particular paradigm of vocal interaction in the singing mouse, one can study the entire sensory motor arc, from sensory evidence accumulation to the decision of whether the animals will respond or not, to vocal initiation and motor command, and also muscle movements. And today I told you a little snippet of our work on vocal initiation and motor command. I'm also extremely interested in the lab to understand how novel behaviors might be generated in general. And especially using this particular system, we can understand how the song system may have come about via changes in the genome as well as in neural connectivity in the brain regions compared to, say, an ancestral behavior in an ancestral species like the lab mouse. With that, I would like to thank the people in the lab. So I showed you work from my postdoc, Cliff Harpole, and my PhD students, Mike Zeng and Emily Isco. I'd like to thank my collaborators at Cold Spring Harbor and elsewhere, my funding, and all of you for listening. And I'm happy to take any questions if you have.
Soumya has a question here.
Hi, I'm Soumya. That was a fantastic talk. I just wanted to know whether these vocalizations are innate or whether they are learned.
Excellent question.
Work on
So,
therefore.
yeah, I understand. All my friends work on zebra finches, so I understand the question. So we think that vocalization itself, the ability to produce these songs, that is innate. And there's some evidence to suggest that. So these animals have dialects, and even, for songs that come from animals that are really close to each other geographically, they still maintain their original genetically endowed qualities of the song. So we think that the ability to produce the song is actually innate. But we are right this moment doing our first ontogeny experiments in the lab with the hypothesis that perhaps the auditory motor coupling, the turn taking, and the appropriateness of their social interaction might depend upon auditory exposure as a function of development. So we still do not know if their ability to do counter singing requires any auditory exposure. So we are actually doing the classic zebra finch experiment of depriving the pups of auditory exposure as they develop to finally check if there is any impairment in their ability to do turn taking.
Hi, this is Shorbo. We work on mouse vocalizations also, the ancestral kind. And I mean, we do have some data which also suggests that the motor cortex can be involved, but in the aspect that in production of sequences as opposed to simply vocalizations. So I mean, it's not published yet, but uh, if you inactivate the motor cortical areas, they still produce vocalizations, going back to Soumya's point of them suggesting that they are innate. And but they lose their ability to produce the kind of sequences they otherwise would produce in a courtship scenario, the sequence of syllables. Anyway, but the question to you is that so are any experiments with the role of PAG done in these mice?
Um, Fantastic question. So yes, so we have been working on the PAG for the last few years, with the assumption that perhaps PAG would be a midbrain control center for producing these individual notes. So I'll tell you a few experiments that we have done. So we wanted to understand if the phonetic mechanisms of how you couple sound with respiration is actually shared between the ancestral USVs and these songs in the singing mouse. So we did tetanus light chain inactivation of the posterior PAG, and we found that the phonetic mechanism is actually fully shared. Not only that, at the level of the periphery, the songs are produced by a whistle mechanism, which is exactly the same thing as USV. So from the PAG downstream, it seems that the ability to produce song notes is basically co-opted from the ability to produce USVs. We have also done optogenetic activation of the PAG and found that it is sufficient to produce these USVs in the singing mouse, which again tells you that the downstream phonatory mechanisms are pretty conserved across the two species. For the song system, we have found that impairing PAG also changes vocal patterning. So we are very excited to follow this up. We are currently now beginning to do our first silicon probe recordings in the PAG. On the structural side, I didn't tell you about a class of projects that we do in the lab, which is we try to understand the circuit differences in projection patterns between the lab mouse and the singing mouse. And in this case, we found that there is about a factor of 4 or maybe 200% increase in the projection of motor cortex to a particular portion of the PAG, and we are following that up with more functional experiments there. So yes, very interested in the PAG. Happy to talk to you more. The motor cortex results sounds very exciting, and that would be sort of in line with what we think is different in the song system. It's not about the production of individual notes, but actually this patterning. So if you find a patterning deficit from motor cortex inactivation in the lab mouse, that would be fantastic. Yeah.
Orko, this is Krishanu.
Yeah. Hi, Krishna.
Hi, how are you? Good to see you.
Good.
What's
Good to
your,
see you.
yeah, what
Yes.
happened to your head?
The same that happened to you.
Okay, so it's very nice work and very exciting. So I have two questions. The first, which is the continuation of Soumya's question. So how
Yes.
much of the pattern variation did you observe in this song? Is it contextually variable?
Excellent question. So, what is interesting is that when these animals produce songs that are in solo, when they are singing by themselves, it's very stereotyped. And we can actually run acoustic classifiers on those songs and actually show that there is information present in those acoustics about individual identity. Now, that particular stereotype gets completely broken when the animals participate in social interaction, and it gets broken because what they're trying to do is also actually combine their production with the auditory input. So depending upon what the other mouse is doing, they can flexibly change it, and they can change it pretty robustly. So we find that there's this interesting dichotomy between their behaviors when they're producing the songs alone, and they use this really for like long distance pinging the world at anybody around. So they'll sing this in a very sort of continuous rate at a basal level, profoundly shift their acoustics, certain parts of it, not the individual notes, but certain supranote features like how long the song is, what the timing of this is, the tempo of the song in a profound context dependent way. Yeah.
And do you see any frequency power spectrum change when they are communicating close range versus long range?
Interesting. So yes and no. So basically, the ultrasonic vocalizations that these mice make when they come closer is sort of the pitch is slightly raised compared to the song, but they are nowhere close to say 60 hertz, 60 kilohertz, 70 kilohertz, which is typically the range found in say lab mouse. And that is because I think of the laryngeal morphology. So their anatomy is such that, and they're slightly short. So they produce songs and notes of the order of 40 kilohertz down to 10 kilohertz. So there is a slight change in the frequency and the pitch distribution between vocalizations that happen when they come close versus far away. But apart from the change in pitch, the more profound changes happen in terms of loudness, in terms of this patterning that does not seem to be the case for over tens of seconds in the USVs, as well as in the ability to use audio vocal coupling. So those are the three things sort of at the supranote level that we seem to find when these animals come closer.
Oh, thank you.
Thank you.
If there is no further question, let's thank our group one more time. And
Thank you.
our group. We hope to see
Thank
you
you.
here at some time point.
Me too. Me too. Me too. Bye. Thank you.
Bye bye. So just a quick announcement. We'll continue with the talk, and at the same time, I'll arrange some tea and snacks outside. So you can bring tea and snacks as and when you get time, so that we can save some time actually. And the next speaker is Balakumar Srinivasan from my lab. He'll be talking about the homeostatic scaling.
Hello. Awesome, right? Okay, so good evening, everyone. So let me start my presentation on the synchrony between translation and protein degradation, and how does that work in excitatory synapses, and what does it translate into synaptic scaling. So first, to give a little bit background about it, I will explain about the target firing rate or the optimal firing rate that is developmentally encoded in a neuron. So once a developmentally end development stage neuron is fully this thing developed, so what it does is it has its own firing rate, and then which can later be modulated pharmacologically or genetically, which changes the firing rate based on the excitotoxicity or hyperactivity, or in the other way, the hypoactivity. So this scales the activity in which the neuron responds to the stimulus. So this particular target firing rate is the optimal solution in which the neuron tends to reach itself to. So here we are going to talk about two different types of conditions. One in the basal condition in which there is a normal level of firing, and then in a terms of reduced network activity, which is hypoactivity, an increased amount of the AMPA receptors are incorporated into the postsynapse to increase the sensitivity of the postsynapse to gain more information from the stimulus. So in the other case, the case would be the increased network activity, which would be hyperactivity. So what happens is the neuron modulates its expression of surface AMPA receptors and removes them from the postsynapse so that its sensitivity is reduced so that it does not get excitotoxic in response to this particular stimulus. So to summarize this particular theory, what we say is synaptic downscaling decreases the firing rate and then also removes the postsynaptic AMPA receptors. And in the other way, the synaptic upscaling in the synapses increases the firing rate while also increasing the AMPA receptor expression at the synapses. In between all of this, the homeostatic scaling, which refers to the term, it maintains the homeostasis of the synapse and also prevents chaotic misfiring, leading to aberrant expression of AMPA receptors at the synapse. So based on this particular theory, what we are curious we wanted to ask about how does protein translation and protein degradation, which were previously known to influence the synaptic scaling differentially, and how do they play a role, and how does proteostasis come together to bring protein translation and protein degradation? How does the handshake work in the terms of dynamics of bringing the synaptic proteome together? So what are the molecular sensors that senses the stimulus, external stimulus a neuron receives, and what are the final effectors that this particular scaling is actually output in the neuron as? So first we were curious about how a synaptic downscaling induced by bicuculin mediated hyperactivity, it activates this. We see that in response to bicuculin, we see a decreased synaptic amplitude, which shows that hyperactivity leads to a decreased firing rate. And then when we inhibit the protein translation with the anisomycin, we see a similar decrease in the firing rate. Whereas in the opposite term, when we inhibit it with lactocysteine, which is a proteasome inhibitor, we see an increased expression of the firing rate. But when these two were added in combination in the final term, here we see that there has been no change, which effectuates the change in the firing rate. So now this led us to the curious case of whether protein translation and degradation can come together to inhibit the hyperactivity mediated changes which can be induced by this bicyclin stimulus. So similar to the patch clamp recordings, we saw these synaptic AMPA receptor stainings at the synapse to see what is the activity of AMPA receptors or abundance of these AMPA receptors at the synapse. So here this is the subunit of AMPA receptor S-GluA1, which we saw in response to the co-localization with PH95. We saw that in response to bicyclin, though the intensity of these GluA1 receptors were decreased, but in addition to both lactocysteine and anisomycin, we see that this particular intensity has been restored, or in other terms has not been changed due to the hyperactivity caused by bicyclin. So we observe the same thing with the other subunit of AMPA receptor, which is the GluA2 subunit. We see a similar expression pattern. So based on this, we are clear that the inhibition of both protein translation and protein degradation brings about a change which stops the hyperactivity mediated change in the firing rate of homeostatic scaling. So thus we wanted to see what are the molecular mechanisms that happen inside that brings together this handshake between protein translation and protein degradation. So while we were looking at interacting partners of the proteasome, of the translating proteasome, we also observed the expression of RPT6 like proteasomal subunits, but also surprisingly we were also able to see this miRNA induced silencing complexes like AGO, TRIM32 and MOB10. These were also getting pulled down along with the translating polysomes. So what we found out was these two particular molecules of the MIRIS complex, TRIM32 and MOB10, they had differential expressions. TRIM32 was highly expressed in response to bicyclin, and while response to bicyclin, MOB10 expression had gone down drastically. So this led us to this question of whether synaptic downscaling, how does it trigger the redistribution of MI risk factors, and whether does this play a role in changing of the hyperactivity induced changes. So here in basic culture, we tried to see whether the inhibition of translation or degradation can change this particular hyperactivity induced downscaling. So here we noticed that in response to the incubation of translation inhibition of TRIM32, we were seeing that MOB10 degradation is being stopped. So we see that this disinhibition of this MOB10 degradation is probably because of a TRIM32 translation mediated MOB10 degradation inside the MI risk complex. So to confirm that, we wanted to see what is the effect of MOB10 by itself in inducing hyperactivity like changes. So here we tried to induce MOB10 shRNA, and then we saw the expression of S-GluA1 and S-GluA2 receptors. And then we saw that similar to bicyclin, we saw a significant decrease in the AMPA receptor subunits. And similarly, when we try to look at the functional output of MOB10 shRNA onto the synapses, we saw that the firing rate also is reduced similar to the bicyclin mediated synaptic scaling. And thus we wanted to see what happens when you combine the bicyclin mediated hyperactivity with a genetically MOB10 knockdown. And then we saw that there was no particular difference between the MOB10 knockdown and the bicyclin mediated hyperactive changes. So thus what we wanted to see is whether is there a possibility we can rescue this particular loss in firing activity by doing a MOB10 overexpression. So thus we did a MOB10 overexpression where we were able to see that there is a slight protection against this particular reduction in the target firing rate between this gray bar and this light green bar. So and this, from the previous data, we know that TRIM32 and MOB10 have opposing effects on each other. We wanted to know whether TRIM32 knockdown can work in a similar method close to MOB10 overexpression. And while we did the same thing, we saw that TRIM32 knockdown caused a similar rescue of hyperactivity mediated changes in the firing rates. So, and this we were also able to show during the abundance of surface AMPA receptors in the synapses, where they were able to get changed at the synapse. And thus we were able to see that whatever changes were caused by bicuculin can be blocked by the inhibition of TRIM32 RNA alone for both S-GLUA1 as well as S-GLUA2 subunits. And then we wanted to see at this MI risk, how does this TREM32 translation get controlled or is being manipulated by bicuculin mediated changes in hyperactivity. So what once we focused is the activity of translation can be caused by the mTOR complexes. So when we try to see whether using rapamycin, whether we can block the inhibition of TRIM32 synthesis, we were able to see that in response to bicuculin, the overexpression of TRIM32 that was caused can be easily blocked by the addition of rapamycin. So thus we were clear that there is a mTOR mediated activation of TRIM32 synthesis, which then also leads to the protection of MOB10 degradation. So the same thing we were also, when we were curious about whether we can rescue the particular hyperactive changes in the synapses using patch clamp data. We were able to see that the hyperactivity mediated downscaling through bicuculin can be inhibited by using or rescued using rapamycin treatments. So now, how do we make sure that this particular pathway that starts from bicuculin to mTOR pathway to TRIM32 to MOB10, and how does it connect to surface AMPA receptors? We wanted to find the crucial link, and using these Western blotting and luciferase assays of ARC, we were able to see this plasticity-related protein ARC is the one that regulates surface AMPA receptors synaptic abundance. So to summarize it all, I want to say that translation and protein degradation is coordinated simultaneously during the synaptic downscaling process, and the translation machinery, active proteasomes, and the MI-RISC complexes are directly interacting with each other under this particular stimulus. And MI-RISC remodeling takes place through TRIM32 synthesis, which leads to its subsequent MOB10 degradation. And then final MOB10 degradation leads to the changes in the surface AMPA receptions via ARC mediated pathways. So this is the summary, and this is generally how the synaptic activity is being modulated by these particular steps. And this is the theory of how it works. And these are the people I want to thank. So if there's any questions, you can ask, please.
Let's talk. I had two questions. One is, maybe I missed it. What is the timeline of your manipulation? Do you look after 24 hours, or how long does
So this is chronic manipulation of 24 hours in bicuculin and rapamycin, whereas the genetic manipulations were done post developmental stage P12, which we decide as the development of the axons and dendrites are pretty set. So we notice that in some of the cases where we intervene genetically way beforehand, we make changes to the presynaptic activity also. So which does not really talk about how the bicuculin mediated hyperactivity changes are regulated.
And one feature of scaling typically is that all synapses of the cell are sort of shifted to a lower level of AMPA receptors, but the type of mechanism that you're describing could actually also act locally. So I'm wondering if you had tried or thought about experiments to just, for example, express tetanus toxin in a subset of terminals that are forming onto one cell to shut off half or a third of the synapses and then see if these changes happen locally.
So since these were done in hippocampal cultures, there is no specificity among them. But if we do it in ex vivo slices, maybe there is a way of targeting those specific way in which you can choose which particular neighborhood to inhibit those certain mRNAs to see whether you can get any changes. But in my opinion, I think you would get something similar to along what we are getting. where you get postsynaptic changes with a definite reduction in surface amperes. But if there is more of a presynaptic, huh? Okay, locally you assume it to work on a shorter time scale where acute Hebbian plasticity might kick in, because over a long period of time it needs a lot of support back from the cell body. So this gives a lot of time for the entire proteostasis to be stable within the entire cell. So doing it local for a chronic this thing would end up giving similar results, similar to what I think the synaptic tagging and capture would give, with a higher expression of synaptic AMPA receptors in other places and lower in some places. Would that, is that, are you asking like locally per dendrite? Okay, okay. I assume you meant locally, but like per dendrite differences, single synapses. Okay. Then you would see single individual changes in the synapses where only the AMPA receptors are getting endocytosed at one synapse.
Good evening, everyone. I'm Srishti Jain, a PhD scholar at Information Processing Lab. The today's talk is on alteration in coding of ultrasonic vocalization sequences in a mouse model of ASDs. By now, as we know, that social communication impairment and repetitive behavior are primary phenotypes of autism spectrum disorders. To better understand these phenotypes, we turn to model organisms like mice, and we particularly interested in communication impairment. So mice vocalizes in different social contexts, and which leads to specific behavior from the receiver's end. So there are two types of mouse vocalizations which are extensively studied: pup isolation calls and mating calls or courtship calls. So in case of pup isolation calls, when pups are isolated or separated from their mothers, they vocalizes, and which in turn lead mothers to search for their pups. And in case of mating calls, adult mice or produces calls in the presence of females. Our previous lab work has used or focused on mating calls, and they recorded it, they analyzed their structure, and we derived 8 natural sequences using mutual information based on Shannon 1950s work. And to compare, we also designed 12 random sequences which does not have any inherent structure in it. So all the data which I'll be talking about is recorded from in vivo electrophysiology from primary auditory cortex in mostly in female mice. So what they found is basically that when the same syllable is present or is a part of natural sequence, it leads to more neuronal spiking than when it is a part of random sequence. And this held true across all types of syllables. And they also this is just for one single syllable analysis. They also looked into the whole sequence analysis, and there also for natural sequences they show that the selectivity for natural sequences was much higher than the random sequences. Selectivity is normalized spike rate. So that highlights the importance of sequence structure in auditory processing. That's already the sequences were already discussed by Dr. Shorbo. So I'm not yes, and also the data. So what we did next, so these calls are given that these calls are occurred in social context. So we were curious, what if we expose females with male, and that may lead to any changes because of the plasticity induced by that social environment exposure given to the female. And they did find in the data that due to social exposure there was change in selectivity and for the whole natural sequences, but not at the single syllable level. So in our work for in our study for valproic VPA valproic in utero valproic induced mice model, it's a model for ASD. We found out that for the whole sequences, that inclination towards natural sequences were gone in the case of VPA female. That we have seen in typically developed females. So this bar graph, the left side is for females, and the data on the right side, the 4 plots are for the males. In the dashed are Yeah. So the red one is for random and the blue one is for the natural. So you can see the first two the bar plots, which are for the female typically developed mice. The blue natural selectivity is significantly higher than the random ones. But in the case of VPA females, that selectivity is completely missing, and there's no difference in the selectivity for natural and the random sequences. There is indeed the difference in VPA male mice, which is already also there in the typically developed male mice, but the selectivity is reduced in the comparison to the male typically developed mice. So, but in the case of single syllable, again, there was no changes. First of all, they couldn't even differentiate between the random and the natural single syllables. And even after okay. So when we tried with exposure, even after exposure, there was no change in single syllables, even for the VPA female mice. These okay. So when we looked into the data for the VPA female after exposure, what we find is that there is some amount of change, or there is some amount of increase in selectivity for natural sequences, but it's really very less with comparison to the typically developed female mice. So basically, what we can conclude from here is that VPA mice model or VPA females are struggling, or most likely they're struggling to leverage the structural information of sequences which typically developed female mice can effectively can use. But here again, the question still one question still remains unanswered. That is, even after exposure, we can see the change in selectivity in the entire sequences, for the entire sequences, but not for single individual syllables, even though these sequences are composed of those syllables only. For the sequences are same, just we are taking single syllables, and in the whole sequence analysis we are taking that complete sequence. Still, there's no difference in for the after exposure, before exposure, no difference for the single syllable. So what makes those sequences so special? So to answer that, we did gap analysis. So what we did is, we just took out the individual syllables, and then we computed selectivity. That's without gap analysis, without gap, and we also computed selectivity, the bottom panel, where we computed selectivity for syllables and the gap following. Gap is intersyllable interval. Okay, so the results are striking here that when we are looking at without gap selectivity, the pattern of encoding it's completely changes. It's completely opposite. So if you can see, it's clearly visible that on the top panel, typically that's TD is typically developed female, and VPA female typically developed mice, male, and VPA male. If you see that red denotes to random and the blue denotes for natural sequences, the selectivity for even in typically developed female, the selectivity for random sequences is higher than the natural sequences. So these are just the green are syllables. So these are just token we are just computing or cumulative responses of tokens. And the bottom panel, in the bottom panel, when we are including gaps, the selectivity for natural increases. And yes, so what we conclude from here is that the gap, the silence is an integral part of this sequence as much as the syllables are. It's the structure, the entire structure, what is important in the vocalizations of males, of mice, in this case males, and that is what is making sense to the female. The entire structure is what is making sense to the females. So and this is for the exposure. This is again for the exposure, exposed typically developed female and typically and VPA females. The results remain same even in that case. That the top panel is for without gap, and the bottom is for with gap. So the conclusion of my talk is simple, that I was trying to, I was trying to convince somehow that the vocalizations, the mice vocalizations can be used, or can be used analogously to speech, as they do have structure in it. They are not just random syllables produced by mice, and obviously there are contexts, but they have in that context also, we generally we look at only syllables, frequencies, frequency or pitch or counts, but no one has ever done the analysis for the entire sequences or tried to look into the structure of the vocalizations. So and we can use these mice models for any language disorder, to study language disorders or communicative disorder in future. I'll just end my talk here. Thank you.
So you showed that in VPA females, the selectivity was increasing even for random sequences, random syllables, right?
Here?
The previous panel itself.
This one.
Right, exactly. As compared to your control females,
yes,
the selectivity for random syllables was higher.
Right.
Right. So now my question is, if you were to give these mice, this VPA treated mice, a two-tone choice task, let's say, where these two tones are random syllables, would you expect these VPA treated
Two
females?
tones are random syllables.
Yeah, like the stimuli, audio stimuli is random syllables consisting
yeah,
of
two
random
stimuli are
syllables.
random. Okay. Yes.
Would you expect these VPA treated females to perform better on that task? What do you say? A correct and wrong choice task for random.
Both are random. Both are random.
Yeah, and one specific stimuli comprising of these random syllables has a associated reward, which is the right stimuli, and the other one does not.
Okay.
So in such a choice task scenario, would you expect the VPA treated females to perform better because they have higher selectivity in terms of responses?
Okay. I think first of all, if you're talking about experiments with reward, anyways, even if I think in VPA case, if I'll take natural syllables. Wait, first of all, syllables are with respect to natural and random are with respect to the sequences they are part of first, and secondly, if you're talking about reward, anyway, whatever stimulus you give, I think reward works with the reward is coming with the stimulus. They're associated with the stimulus. Doesn't matter what stimulus is. So that paradigm should not work. And secondly, that paradigm should work for everything. And
but would the performance be on par, or would you expect the VPA females to perform better? Is my question.
Okay. So I think, but if you're taking that syllable, see, first of all, learning, this doesn't show the learning processing of VPA and typically developed mice. Because then again, in that paradigm, the learning will play a role. That's first thing. And if you are, I think if you are interested in whether they are more inclined toward random sequences than the natural one, and they prefer random sequences, are you hypothesizing that?
No, it's just based on selectivity itself. Just that they have higher selectivity in terms of responses.
Yes.
To even the random sequences, whereas the control ones are only selective to natural sequences or more selective to natural sequences.
I think that's just for that is just the stimulus for them, right? And if you see the overall selectivity, it's not very much higher than the typically developed. And again, I think one more point here can be that VPA in VPA cases, or sorry, in ASD cases, there's inhibitory and excitatory ratio imbalance. I don't know how much that plays role here. there's auditory or sensory dysfunctions is also happening in VPA or in ASD cases. I even don't know how much that plays a role here.
Okay.
Am I making sense?
Yeah.
Uh,
So tomorrow we should start at 9 o'clock.